Good morning and a warm welcome to everyone joining us online for this year's Agronomy Conference. The conference is now in its 18th year, and whether you're joining from the office or the kitchen table, it's great to have you with us. Over the course of the sessions, we'll be diving into some of the most pressing challenges and exciting opportunities in agronomy, from integrated pest management to the latest tools and techniques shaping the future of sustainable farming. In just a few moments, we'll be starting with a first session, which will be on IPM and a focus on cabbage stem flea beetle and the UK cereal pathogen virulence survey. The second session will be all about fungicide response curves and also resistance in fungicides, herbicides and insecticides. After lunch, we'll have a session focusing on variety selection, and the final session of the day will be on nutrient dynamics, cover crops and biostimulants. But today is about more than just the technical presentations. It's about connecting us all as a community committed to pushing the boundaries of modern agronomy. And what makes this conference special is not just the caliber of the speakers or the topics and the sessions on the agenda, it's you. Your questions and insight are what's going to make the discussions richer and more relevant for us all. So I encourage you to make full use of Poll EV, and I think a QR code will appear on the screen, and there'll be a code for you to use also. So please make full use of that, and we'll be hosting Q&A sessions throughout today. So over to our first session. Let's get started. I'm going to hand over now to Henny Lowth, who spent the last few months tirelessly making this day happen together with the rest of the team. And Henny will introduce our first speaker.
Brilliant. Thank you very much, Anna. And yes, welcome to all. And yeah, we're really looking forward to a really great day of speaking sessions. So the first one that we're kicking off with, we're going to be looking at integrated pest management. So we've got 4 fantastic speakers for this session. So the first one is Mark Ramsden. He's an IPM principal consultant at ADAS, and he's going to be talking you through about getting farmers engaged with IPM. So over to you, Mark.
Thank you, Henny. Hello. I'll begin by saying something that you already know. Genuine engagement is not achieved by presenting information. Engagement comes from the interpreting and discussing and challenging that information, and then understanding how it applies onto a given farm. In this respect, integrated pest management is particularly challenging because there are lots of different approaches to managing lots of different pests, and understanding how those different approaches integrate with each other to form an overall IPM strategy. And that needs engagement across the sector to help farmers make the best choices for their farms. So what we're trying to achieve with this engagement is a step-by-step process of improving IPM over time. Now, over the last 10 years, we've seen huge advances in the advancing of integrated pest management in the UK. And that comes from this circular process of planning what's going to happen on a farm, implementing that plan onto the farm, adapting it to reflect the different pest priorities that emerge during the season, reviewing that process, and then updating it for the next plan year after year. This step-by-step cycle advances our IPM strategies, but it's important to understand that it's not going to bring about massive quick change. It is something that changes over time. It's also worth remembering that integrated pest management can be applied to farms irrespective of the strategies being implemented. So whether it's a regen ag farm, an organic farm, or if they're using precision farming or more conventional approaches, integrated pest management is a strategy that will help the farm and the farmer. Now, if we want to bring about change, we want to improve application of integrated pest management, then there are lots of different ways of doing that. We could have a more policy top-down approach that provides financial resources for implementing different aspects of IPM, or that has legislative mechanisms for achieving IPM. The problem with this approach is that because it's top-down, it becomes very generic and sometimes puts people into a position where they're making difficult application choices. We also find that once those policy incentives stop, then the action stops, whereas what we really want is genuine engagement of IPM that genuinely helps farms progress their crop protection strategies that protects the crop performance this year and into the future. So to do that, what we want is some tools that help us facilitate the engagement. So I'm going to talk through a few tools that are available and how they're used to achieve genuine engagement. The first one is a tool for developing IPM plans, and this is for creating a bespoke plan for a given farm. So this is a free tool to use. The link is at the bottom there, and you can use it to create as many different plans for as many different farms as you like. You can also copy those plans across. You can edit them over time, so you can copy and duplicate a plan into following years. And this helps you develop your IPM strategy for a particular location over time. When you walk through, you can select the crops that are relevant to a particular farm, and then the plan will give you a series of questions to work through together between you, the agronomist, and a farmer to understand what the general practices are that you want to implement, what they mean, what the kind of supporting resources you might need are. And you can also look at what other farmers are selecting in the tool by using the benchmarking. You can click on more details, and that'll take you through to additional technical details, for example, on the AHDB website. Once you've gone through those general practices, it'll also alert you to any particular control measures that you might be able to select. And again, you can look at the details of how those work and get more information. So this tool is designed to help you walk through with a farmer and get that genuine engagement to create a bespoke plan for a farm. At the end, you can review the plan and you can create a PDF report, which you can then sign off together, and that complies with the SFI Action IPM 1. So this tool helps you to get that initial plan, but obviously plans often go wrong and you need to make changes and adapt to the season. So the next tool I want to talk about is the IPM Decision Support System platform, IPM Decisions. And this I presented a couple of years ago when we launched it in 2023, and it's still going this year, and we'll keep maintaining this into the future. And it supports a whole range of different decision support systems across a range of different pests and crops. And the key thing with the decision support systems is that they don't give you an answer, a particular direction to go in, but what they do is provide a measure of risk and reduce the uncertainty about which pests are a priority, which ones potentially are not going to be a problem in a particular season. You see here on the home page that there is a risk map for Septoria, and I'll come back to that in a moment. But if you register an account and log in, you can get detailed outputs on a range of different pests. So this is just a bit of a clip from my platform account. And you can see I've set up a farm for our ADAS Botsworth site in Cambridge. And at the moment, there aren't many pests that are posing a problem to crops, but barley yellow dwarf virus is one that we need to keep an eye on. So this is the TSUM model that's also available on the AHDB website, and you can see on the right here that it's reporting periods of high risk when there's potential for winged aphids to be in the crop and spreading the virus. Again, it's important to say that this is not telling you this is what is happening in the field. This is telling you where there are periods of risk for you to consider whether an action is required, to monitor in the field, to see what other information you can pull together and what decision needs to be made. If you'd like to know more about BYDV in particular, we're doing some work on the AHDB Strategic Farm East, focusing on BYDV, and there'll be more details on that in the report coming out in the new year. There's also opportunity to join the BYDV Hub, where you can engage more directly in how these different support systems and the impact of different varieties can be used to create an IPM plan for BYDV. So again, this platform is designed to help you engage and discuss these risks with farmers. And I'll just go back to the home page here, which is where we can see the risk map. At the moment, there are two relevant risk maps for the UK. There's this Septoria one, and there's also a pollen beetle migration risk map that will go live in the spring. So this Septoria humidity model was developed in Denmark, and it's been validated in parts of Scandinavia. It's not yet been formally validated in the UK. However, the model is looking at the risk of septoria based on the relative humidity over a 72-hour period, which has been shown to be linked with a high risk of infection of the disease. And so while it's not been validated in the UK, it can still be used to give us an idea of the kind of thing, the kind of risk posed in the UK during the season. So we know that in the season just gone by, we started the spring with a high risk of foliar diseases because the winter had been wet and mild. And then we came into the season and the model started working. And I'm just going to run through a video which shows day by day the risk of septoria across the UK. And you can see at the start of April, there was a moderate to high risk of infection really through the first half of April, just as the crops were becoming vulnerable to the disease. And at that point, there was a lot of rainfall around and it was lingering in the crop, which not only meant there was a high risk, but there was also difficulties in actually getting onto the land to apply treatments. So we ended up with quite a high pressure at the start of the season. Then when we came into May, really the rainfall continued through the month, and that led to ongoing risk after the latent period, and we ended up with a really high disease pressure across the UK this season in what my colleague Jonathan Blake has described as a biblical year for disease. That gave us opportunity to learn a lot about septoria and other foliar diseases, but we also are able to look back through the season using these kind of risk maps to see what happened. And hopefully you'll be able to use this map again next year to follow through and help guide you on what decisions you could be making during the season. So alongside the IPM plan, we have these decision support systems that help us provoke discussion between you and farmers to identify what decisions need to be made. But this all needs to be built on a foundation of good knowledge about the overall management of all these different pest diseases in the crops. So I'm delighted to today launch a new video series that was funded by DEFRA, and these are freely available videos that we will be promoting over the next few months that walk through the science and practice of integrated pest management, focusing on disease management in cereals. But really, because they delve into the detail of the biology of the system and the different tools available, the concepts can be adapted across to other pests and diseases. So I do encourage you to take a look at these and work through. Now they are designed to be watched in any order, though we suggest you watch them in the order that they are in the playlist. And we also suggest that you use these as starting points for discussion. So they shouldn't be watched in isolation, they should form the basis of working through different concepts and ideas. So they start off with a short introductory video about what the series is about, and then they go through crop productivity and foliar diseases, methods of controlling disease, including both variety and fungicide applications. Also go through the details of fungicide resistance and how you can combine all these factors into an integrated approach, and also considerations of how these approaches also interact with the environment. So that gives us a good foundation of the knowledge, and there are lots of other training materials out there that you can use to build your knowledge base. But we also want to understand how these things get applied in practice. So it's really useful to see what other farmers are doing, and I've just highlighted here some videos that have been developed recently from an international network of farmers and agronomists looking to advance IPM strategies. Now, it might seem a bit odd to watch a video about a farmer managing disease in olive groves in Greece, when clearly that's a very different crop system to to systems in the UK. However, what we're really trying to learn from these videos is the process of change and how they were tackling different diseases and pests, what kind of considerations they had, how they went through integrating new approaches into their farm. So these videos are really there to provoke discussion. So what I suggest would be good is to watch these videos together with your farmers and then try and talk about how different aspects of that could be applied onto your farm. And this is what we really mean by engagement. By watching these things together, to discussing how they can be applied, that's where you start to interpret this and challenge them and find new ways of doing things on a particular farm. It can be a really daunting process to make changes to established systems, but only by really seeing what other people are doing to understand how the challenges and how they've overcome them can you start to really reframe your thinking on a particular problem. To support this, we've created a new IPM network, the IPM Net, and this is a network for sharing knowledge and experience to improve uptake and implementation of IPM. And hopefully this network will help connect people, and it's a free network to join, so you can register and you'll receive updates on resources, news opportunities and upcoming events, and we will facilitate discussion and help build the connections between people. We'll be putting out more resources over the coming months, and in February next year there will be an IPM conference. If you scan the QR code there, you can register your interest in joining, and there will be a more detailed agenda coming out in the new year. It's quite tempting to say that once you've established an IPM strategy, if it's not broken, why should you fix it? But we are farming now in an ever-changing environment. There are different weather patterns coming through. There are differences in what chemicals are available. There are new varieties coming through. Novel approaches to pest management are always being developed. And if your strategy stays the same, it will miss opportunities to improve the application of IPM, improve crop protection, and potentially you'll find that you're up against some big challenges in the future that you aren't necessarily prepared for. So this process of continually reviewing and updating IPM plans is really important to keep on top of the pressures and to make sure that you are making good choices throughout. So hopefully these tools will give you an opportunity to discuss and interpret and apply different IPM strategies with farmers, and hopefully they will provoke some good discussion, and hopefully we'll be able to support those discussions and greater engagement between across communities in the future. Thank you very much.
Brilliant. Thank you very much, Mark. That's great. It's great to know there are so many resources and tools available for both farmers and agronomists. So we really do encourage you to get involved with everything that Mark's just been talking about. Brilliant. So we're on to our second session of the integrated pest management topic. So up next, we've got Sacha White. So Sacha is a lead crop protection scientist at the AHDB, and he's going to be deep diving into the world of cabbage stem flea beetle, so giving an overview of where we are in terms of IPM. So over to you, Sacha.
Morning, everyone. So cabbage stem flea beetle, where are we with management of the pest, and where might the future lie? Now, I think it's always best to have a refresher of the life cycle of a pest. Knowing about the life cycle is key. It helps us understand where there might be weaknesses and that we can manipulate to our benefit. So starting at the top of the life cycle here, adult flea beetles start moving into emerging oilseed rape crops in August, September, usually towards the end of August, beginning of September time. They feed on the crop, they cause that classic shot holding damage, and if the feeding pressure is very high, then the crop can be wiped out. After a couple of weeks, they'll mate and start laying their eggs. That egg laying is temperature driven, and so it can continue all the way through the winter as long as conditions remain mild enough. Eggs are laid in the soil near plants, and those eggs start to hatch out really from sort of October times onwards, sometimes in September as well. And again, that rate of egg hatch is temperature driven, so it can continue through the winter, just like egg laying, as long as conditions remain mild enough. And it's fair to say that probably for the most of the last decade, winters have remained mild enough for that to happen throughout. Hatching larvae move from the egg to a nearby plant. They climb up, they burrow into a petiole, and they sit there and feed, and they're protected from all sorts of things, including pesticides. Come March time, some of those larvae will move to the stem and they'll sit and feed behind the growing point. And it's usually in the new year when we start to see them, the main damage from the larval feeding, and also crops can be wiped out then. Come April, May time, the larvae have finished developing, they'll drop from the plants into the soil, they'll burrow down and form a pupa, and in the pupa the new adult forms. Those adults emerge June, July time. They'll feed a little bit on the crop, will have no impact on yield, and then after a short time they'll move to a sheltered area where they'll enter what's called an estivation. It's essentially a hibernation in the summer. After a few weeks, they'll leave the estivation, usually August time, and ready to move into new crops, and the process starts again. Now, I think it's worthwhile thinking of the adults and larvae as two separate problems. They cause different sorts of damage at different times, and the ways that you might manage one might differ to the way you might manage the other, and sometimes actually make the problem worse. Now, the reason we have a problem with cabbage stem flea beetle is because we've lost effective chemical control. We no longer have neonic seed treatments, and all we have left are prethroid sprays. However, prethroid resistance is now widespread in cabbage stem flea beetle, and that is well illustrated with these two maps of surveys that were carried out by Rothamsted in 2018 and 2019. Every dot on these maps show where a population of flea beetle was collected and tested for resistance to lambda-cyhalothrin. The colour of the dot indicates the level of resistance. So where you've got a green dot, it means none of the population was resistant. And as you go up through the yellows, orange, reds, and into purple, you get to 100% resistant. Now, if you look at the map in 2019, on the right there, you can see very, very few green dots, maybe 3 or 4 across the whole of the country. Most of the dots are actually in the orange, reds, and purples. So most of the populations have more than 50% resistance, and certainly in England that resistance is very widespread, so the chances are you're not going to get control with a prethroid, but you will be knocking back your natural enemies. And speaking of natural enemies, what is there out there that will attack cabbage stem flea beetle? Well, carabids, we know there are ground beetles that will go through the crops in the autumn, hoovering up eggs. They might eat larvae, we don't know, but I'm pretty sure they would if they can find a larvae and get to it. There are parasitic wasps that also attack cabbage stem flea beetle, so there's several species that target the larvae in the spring. It's quite neat how they do this. They walk up and down the outside of the plant, they sense where the larvae is inside. When they find it, they put their ovipositor in through the plant and into the larvae. They lay an egg, that egg hatches out and eats the larvae. There is also a species of parasitoid that attacks the adults. This is Microtinus brassicae. That it does this is a relatively recent finding in the last 5 or 6 years. This is active in the summer and the autumn. It attacks the adults, lays an egg in it, and the adults will die off over a period of days and weeks. But we also think that as soon as an adult is parasitized, that it either stops laying eggs or drastically reduces its egg laying. So if enough adults are parasitized, then that can have a huge impact on the numbers of larvae you're likely to see later on in the season. There are other natural enemies, things like spiders, nematodes and fungi will also attack cabbage stem flea beetle. And actually the latter two are now being investigated to see whether they can be used as biopesticides to target flea beetle. Now we have all these natural enemies, but we've also had huge populations of cabbage stem flea beetle for the last few years. What we would expect is the natural enemies to start exerting an impact on those cabbage stem flea beetle populations. And you can see this is sort of known as the Lotka-Volterra predator-prey model. You might remember it from your sort of GCSE biology. Essentially what this says is that as a prey population increases, then in response a predator population increases because it's got lots of prey to eat. As the predator population goes up, they start to eat all the prey, so the prey population drops down, and then the predator population drops down. And it goes through a cycle like this, and it's well illustrated with that graph on the left there, which shows lynx populations responding to Lepus americanus hare populations. However, we haven't really seen this for cabbage stem flea beetle. And what might be happening here is what's known as the paradox of pesticide. So this is where a pesticide is applied to control a pest, but actually it's having a big impact on its natural enemies as well. And actually that means that in total, you're losing control of the pest. And that's made worse where the pesticide isn't very effective, as is the case with cabbage stem flea beetle because of the resistance issue. But we do need to know more about what contribution the natural enemies are making and how we can improve their activity. Now, earlier this year, AHDB funded NIAB to carry out some work monitoring the migration of adult cabbage stem flea beetles in 7 counties across England. This involved putting traps out in fields in these counties, so ground-based and raised traps. And here we can see the cumulative number of flea beetles caught at these sites in these counties. So what we can see immediately is big differences in terms of the absolute numbers of flea beetles, but also the timings of migration. So the Bedfordshire site had far more flea beetles than any of the other sites. Suffolk, Wiltshire and Yorkshire also had a few, but the others had far fewer. But also the start of migration started much earlier in places like Suffolk, early August there, whereas places like Hertfordshire didn't really start until early September, so big variations there as well. Now, how did 2024 compare to 2020? Well, in 2020, we had far more flea beetles, 3 to 4 times as many flea beetles. And also the pattern of activity, that migration activity, was much more constrained in 2020, really happening in bulk between the end of August and middle of September. When we compare this year to 2021, well, again in 2021, we had far more flea beetles, 2 to 3 times as many. And again, that pattern of activity was much more constrained, end of August to mid-September. In 2022, well, the numbers are much closer to this year, but still more across the board. And again, a much more constrained pattern of activity. Now, the question is why have we had so far less flea beetles this year compared to previous years? Well, it's difficult to know, but there are a few hypotheses. It could be due to the weather. We've had a much cooler, wetter summer than normal, and we know flea beetles don't like that. We've also seen big reductions in oilseed rape areas in recent years. So this autumn, it's been estimated around 215,000 hectares of oilseed rape in the ground. These reductions in oilseed rape might mean that because there's less oilseed rape plants for the flea beetle to complete their life cycle on, that's having an impact on the flea beetle populations. Also, it could be that the natural enemies are finally starting to exert an influence. A final possibility is that we now have this SFI for cover crops, and many of these cover crops include brassicas. And it may be that these brassica crops, these winter cover crops, could be acting as a trap crop, drawing flea beetles away from oilseed rape crops into these cover crops. It's something we need to find out more about. Now, what do we know about cabbage stem flea beetle? Well, there's a lot of AHDB funded work over recent years which has shown us what impact flea beetles have on the crop. We know that oilseed rape plants have a really good ability to tolerate adult feeding, more than 50% leaf area loss, and they can often survive. Once a plant gets to the 4-leaf stage, then they're very unlikely to be wiped out by adult flea beetle feeding. What we really need to avoid is flea beetles arriving into the crop en masse at the point at which the crop is emerging, because that's when the crop is most vulnerable to being wiped out. In terms of the larvae, well, we have a treatment threshold, not that we have an effective treatment anymore, but that threshold is 5 larvae per plant. It's based on good data from the 80s. We don't have a treatment threshold for spring larval numbers, and perhaps we ought to, because we're seeing far higher spring larval numbers than we once did because of these mild winters. More recent work has estimated that every larvae contributes to about 0.05 to 0.07 tonnes per hectare reduction in yield. But that data was based on trials, and that trial data showed that there are some crops that had a very high potential for tolerating larval damage, so much higher larval loads they were able to put up with. And what we think was happening with these crops is that these were crops which had much more biomass in the spring. So this can be down to varieties with good spring vigour, but also seed rate. If you've got a low seed rate, then that encourages much bigger plants in the spring that's better able to tolerate that larval feeding damage. So where are we in terms of managing the pest? We haven't really got a responsive treatment anymore because pyrethroids generally aren't working very well. So the moment is about minimising the risk of the pest being at problematic levels and being able to mitigate its damage. So for the adults, there's a range of things that have been looked at in recent years, and I've given them all traffic lights. Where you've got a green traffic light, it means that there's been a lot of research done here. I mean, we know a lot about the benefit this approach provides. Where it's orange, there is some research, there's some data that shows that there's a benefit. And where it's red, it may be that we have some anecdotal information that suggests it might be helping, but we need to do a lot more work to establish quite how much benefit we can get. So starting at the top here, ensuring we've got rapid, vigorous and even establishment, because where we do otherwise, and a crop is establishing slowly and patchily, that is ideal for flea beetles just to pick off the plants as they come up. So ensuring good establishment is about making sure you've got good soil moisture, having a variety which maybe has good autumn vigour and matching that to sow date, nutrition, all of these sorts of things are important. Sow date has a huge influence on the level of pressure you're likely to get from flea beetle. So for adults, you've got two options. You can either sow early, so this is before mid to late August, and what you're doing here is hoping that you're going to get a crop that's well established, it's got a good few leaves on it by the time the flea beetles arrive, as I say, usually towards the end of August or mid-September, and therefore well able to tolerate that feeding damage. The other option is to sow late, so after mid-September, The idea being that these crops are going to emerge after the bulk of flea beetle migration has passed, so flea beetles are going to be elsewhere, and you're going to suffer much less adult damage. Companion crops. There's now a really good body of evidence that companion crops provide reliable reductions in flea beetle damage. Maybe 30 to 50% reductions, and it might not be the full answer, but it's part of the puzzle. Trap crops, so these are crops that draw flea beetle away from your oilseed rape crop. And there was good work done by ADAS 5 or 6 years ago to show that leaving oilseed rape, a whole field of oilseed rape on your farm until at least the end of September, draws flea beetles away from your cash crop into the volunteers, and therefore you see less damage. Stubble management, cultivations and organic amendments, there is some evidence for all of these to show they're providing some benefit, but we need to do more work. For cultivations, by the way, I mean here min-till approaches. There's a little bit of anecdotal information that's showing that this can result in less adult damage. For larvae, sow date again is very important, and the message here is really clear. The later you sow, the lower the larval pressures you're going to get later on in the season. Now, for sow date in general, there's obviously risks associated with any sow date, and so a possible approach to spread that risk is to spread your sow dates on farm. Companion crops and trap crops, both of these have been shown to also reduce larval loads. Seed rate, again, as I mentioned earlier, lower seed rates tend to produce larger plants better able to tolerate that feeding damage in the spring. Defoliation, so going into a crop and topping it or grazing it with sheep in the autumn or early winter, this has been shown to significantly reduce larval loads. The challenge is making sure that you don't see a yield impact. And that's mainly down to the severity of the defoliation and the timing of the defoliation, but also the spring weather can have a very big impact on this, and obviously you won't know what that's going to be like when you're doing the defoliation. Again, with cultivations, min-till approaches, there is a small amount of information that seems to be suggesting this also results in lower larval loads, but we do need to do more work here. So there's a lot of other work going on. So biopesticides, so RNA, nematodes, fungi. NIAB have also finished some DEFRA-funded work that seems to suggest that lightly cultivating fields that come out of oilseed rape lowers the numbers of flea beetles emerging from those fields. Rothamsted is doing work looking at how cabbage stem flea beetle find crops and how we can manipulate this. BIOS, Harper and ADAS have done some really good work on trying to build models to help us understand the migration of the pest. They've made a good start there. There's more work to be done. And JIC are doing good work on breeding for resistance, which Rachel will be talking about after me. So what next? Well, here I like to draw the analogy with blackgrass resistance. So in the 80s, we started seeing resistance to blackgrass, and it took a good few years to identify what cultural control approaches we needed to use to manage blackgrass. And that was reviewed by Lutman et al., and you can see the results of that review there, which show the percentage control different cultural control approaches provide for blackgrass. And the green bars show all the things that you can do if you wanted to have a winter crop. Now, by stacking those green bars, like ploughing, delaying draining, higher seed rates, that sort of thing, you get additive levels of control, and you put them all together, and you get a control that might be similar to a herbicide. And I think that's where we need to go with cabbage stem flea beetle. Understanding how we can stack the approaches we already know about and get a good and reliable level of control. Obviously, there is increased complexity, maybe additional cost in terms of time and that sort of thing. What we really need to do next is improve how we communicate the work that's already been done so that you can put it into practice, and also carry out further research. And I'm sure that AHDB will be funding more flea beetle research next year. Please submit your ideas for where you think we need to be doing work to the letterbox. Anna will be talking about that in more detail later, but we want to integrate your ideas. So thank you for listening. Thank you to all the people who have been involved in the work that I've talked about here, and also thank you to all those organisations on the right there that have been involved and co-funded the latest piece of AHDB 4-year work, and that final report of that work is going to be available online in the near future.
Fantastic. Thank you very much, Sacha. What a great comprehensive review of where we are in terms of cabbage stem flea beetle research. So just a reminder for those of you at home, We would really like to keep your questions coming in, so we will have a Q&A session after all of the speakers. So yeah, please do submit your questions onto Poll EV. We'll then be putting those to the speakers later on today. So coming up next, it's keeping on the same theme of cabbage stem flea beetle. We have Rachel Wells from the John Innes Centre. She's a senior scientist there, and she's going to be deep diving into varieties and variety performance against cabbage stem flea beetle. So over to you, Rachel.
So thank you very much. It's a great pleasure to talk about the work we've been doing on breeding for cabbage stem flea beetle resistance. So we've had a couple of talks about IPM, and we know that knowledge at all these levels is required to help us combat our pest. So I thought I'd start with the IPM triangle here, which shows we should start with those agronomic practices at the bottom there, go through monitoring and forecasting and warning systems, as we saw presented by Mark, before moving into mechanical and physical controls, biological controls, with those chemical controls as a last resort. But really, to effectively use this triangle, we need to understand four things: the pest and the crop, so the biology and the life cycle that is there, the current control measures, what and how it works, those natural controls, so those controls coming in, what is the efficacy of that, and the regulatory landscape they sit in, and then we can use that knowledge to direct our research and development. So from my interest in here, I'm very interested in the susceptibility of the crop to the pest, and then with natural controls, genetic resistance. So how can we make this work? So one of the things I'm often asked about is it possible to select for insect resistance? Well, actually, we've got a long history of breeding for a lot of traits, so if you think of quality and yield, and these are things that are quite easily selected for. However, when we look at breeding for disease resistance, again, we've been doing this for a long time. This was first mentioned in wheat by Biffin in 1905. So, and these are all things that are quite easily selected for in field trials or under controlled conditions using a natural or deliberate infection. Breeding for insect resistance is less well developed, and mainly because we've had all these chemical controls to support us. So if we look into the field, actually it's that spatial and the temporal variation that makes insect research challenging. We've seen, we can't guarantee when those flea beetles are going to migrate in. Are they going to come early? Are they going to come late? But actually, if you look at distribution across the field, that's variable. So the image on the crop right there, at top right there, shows slug damage within a crop, and you can see this is present in patches across the field. So if you're trying to do a varietal trial, actually there are some patches and some varieties that would be highly affected, and this would be nothing to do with genetics, it's just distribution. On the bottom right, you see a distribution map of cabbage stem flea beetle females. And again, this varies across the field, so very hard to do a varietal trial. We know we've got multiple pests in there, so we've seen an incidence of cabbage stem weevil larvae, so they will impact on the scoring we do. We know when you have differences in developmental stage between your plants, actually we'll have differences in infestation. And also, we've got that availability of the pest. We've got an annual life cycle, so that makes it very hard to study things more than 1 year at a time. So field trials are important, but actually looking at laboratory bioassays allows us to perform controlled infestation studies and also study plant pest and pest environment interactions. So what I'm going to do first is just talk about some of the differences in resistance types. And I'll begin with avoidance and tolerance. And these are really things that have been selected during breeding and under recommended list trialling since we had the emergence of the cabbage stem flea beetle issue in 2013. So if we look at avoidance, actually in this process, we're trying to minimize the pest effect by completing the vulnerable life cycle stage before or after we see that main attack. So breeding varieties with rapid germination and growth following sowing, all those that can grow despite adverse environmental conditions. So if we're sowing early, if it's dry, do we have varieties that deal with drought? If we're going late and it's not quite warm enough, do we have varieties that still perform and grow away? It's harder to avoid the larval infestation, going for late sowing so we're not having the adults within there. Again, we need to push those varieties that grow away quickly. Sacha also touched upon tolerance. So this is where we can mitigate the negative effects, fitness effects caused by herbivory. So do we have varieties that grow away from the adult stem flea beetle damage, so still established despite the adults? And do we have the varieties that continue to yield despite larval infestation? And this is really where those breeders are coming in to help us. So for me, I'm more interested in the true genetic resistance, and we can divide this into two types, antizoonosis and antibiosis. So if you look at antizoonosis resistance, actually this comes down to preference. So is there a genetic trait in the plant that affects the behavior of the insect pests? So does it want to eat one more than the other? Do they want to lay eggs on one more than the other? Then we have antibiosis resistance. And this was where the host plant constituents cause adverse effects to the pest life cycle. So you may get eggs laid by the plant, those larvae may go into the plant, but actually you see a reduction in maybe the fecundity, so you've got delayed development of larvae, reduced size and weight. And we can see this in this picture on the bottom right, where our resistant soybean varieties have much poorer larvae condition than the ones in the susceptible lines. Actually, we can look into orange blossom midge resistance in wheat, and we see exactly this. You've got the SM1, a single major resistance gene, and there's antibiosis resistance that slows larval development. And this is now bred into 40% of the UK varieties. So for my research questions, I'm interested in, can we screen for resistance? Is there variation? How do plants defend themselves? And how do we deploy this variation into the crop? So cabbage stem flea beetle, we get one generation in a year in the field. So that's univoltine. Not very good for studying genetics. So we have captive colonies that we rear in-house, and this gives us a constant supply of beetles. So we've always got adults and fresh larvae to do our trials with. We can put these beetles into our feeding screens. So we've got a typical chamber on the left before feeding. We add our beetle, and 24 hours later, we can see the amount of damage that's been done. If we look with our breeders across some varieties, we've got the estimated mean percentage eaten on the left here, but we can see between varieties we've got a range of variation, with some performing much better than others. We can also go through and perform larval screens, so actually growing plants up to a certain stage, adding a number of larvae, and then evacuating these from the plants to see how well those larvae are performing. And these are tests that we've shared with our breeding partners so they can select themselves. So yes, we can screen for resistance, and there is variation in susceptibility. But how do the plants then defend themselves? So within our project, we've screened a diversity set to look at the genetic basis of resistant, and we can identify resistant and susceptible plants on the left and the right here. Those differences are actually retained, so if we replicate for those lines in our non-choice situation, we always get the same outcome. If we give the beetles a choice between resistant and susceptible, they always prefer the susceptible. And then actually, if we go into the field, you'll see the resistant variety on the left, the susceptible one on the right, and hopefully you can notice there are still green seedlings on the left and not on the right. So the number of plants per plot directly indicates the difference we see between these lines. Luckily, with the population we've screened, we have that phenotype data, but also we know a lot about the underlying genetic variation. And this allows us to look at an association between the two to really go down and identify the genes that are underlying the differences. So this is what the Brassica napus genome looks like, so oilseed rape at the genetic level. The black and red bars represent the different chromosomes, and each of those dots on the graph represents a genetic variant within that plant. the higher up the dot is, the stronger the association between feeding and the genetic variation that's there. And hopefully you can see some peaks on the graph there indicated by the yellow arrows. So we're interested in what are the genes underlying these associations. Testing things in oilseed rape is difficult, but luckily we have a model plant that's closely related, takes up a lot less space, grows a lot more quickly, and we can do the same kind of feeding studies that we do with oilseed rape. So taking mutant lines for our peaks there in the graph, we've got our wild type lines on the left, and we've got our mutant lines on the right. And basically what this shows is we get more feeding in the mutants, which means the presence of the active gene is providing resistance within our plants. And we're now doing further work on this. Now we're also trying to understand a little bit more about those plant-insect interactions, and this can really help us learn how to make the plant less susceptible. So we've got chemicals within the plants that can attract and deter pests and beneficials, and the beetles can also scan the leaves, so they use their feet, their mouth parts and their antennal tips, and they can use this to tell them whether actually something is edible or not. We also know that the plant will respond to attacks by pests and pathogens to activate defense responses. So what do these look like? So for our susceptible and our resistant line, we can say, oh, do we have differences in feeding behavior between our lines? And so for this, my postdoc Ryan looked at a lot of beetles. He was monitoring feeding at 2, 4, 8, and 24 hours on plants, and checking every 5 minutes for the first 4 hours and every 20 minutes for the next 4 hours to see what was happening. And what he found is between the resistant and the susceptible lines here, shown in blue and yellow, there was no difference in time to first feeding. So the beetles were feeding on plants no matter what. But when he looked at the level of damage across time, actually he could see they were eating that susceptible line much more quickly. That suggests there's some induced defense going off in that resistant line. So we know that plants can sense damage, and I'm showing you here a beetle biting into the plant, and you can see this glow of fluorescence. That's the plant sensing it's being damaged. But can it actually sense it's being damaged by a beetle? So in this experiment, we looked at trying to see gene expression changes, so the activation of defense pathways. So on the right, we've got a normal seedling, no damage. In the middle, we've got one with mechanical damage, so we stabbed it with some scissors. And on the left, we've got a seedling that's been fed on by beetles. But can we see any difference in genetic response between these treatments? So the graph on the left shows general gene expression response. And we can see it divides into 3 different sections, ones that are no feeding, ones that are to do with mechanical damage, and ones that can do with beetle feeding. So actually, we've got a very specific response. If we look at the graph on the right, far right, we have the genes that are changing in response to mechanical damage. Then the middle, genes that are changing in response to beetle feeding. And actually, you can see there's much more changes going on with that beetle feeding, suggesting the plant is sensing those beetles and responding. Now, we can zoom in and have a closer look. A bit of a complicated graph here, but I just want you to focus on that purple box. So we have our susceptible line on the right and our resistant line on the left. If our gene expression is low, so no real response, it's blue, and if it's red, we've got a high response, okay? So for our susceptible line, all our panels there for our different treatments are blue. No difference in response. But if you look on the left-hand side to the resistant line under beetle feeding, BF, you see suddenly we get this flush of red coming through. Again, we're seeing that it's upregulation of defense responses in response to beetle feeding. We can see this if we look at individual genes within this category. So blue is our resistant line, again, BF for beetle feeding, and we see this massive uptick in response. And these candidates have previously been implicated in disease and insect response. So these genes may actually be influencing our feeding levels. So I've shown you we know how plants defend themselves, But how do we deploy this variation into the crop? So if we're looking to breed a flea beetle resistant variety, actually conventional breeding, we're taking our resistant, crossing it with an elite, and then going through a period of back crossing and marker-assisted selection. So normally something like this will take 8 to 10 years once we have the knowledge of what we're looking at. Now we can, if we know that our gene is resulting in susceptibility, go forward and look at mutation breeding or modern genome editing techniques. So in both these, we're looking at knocking out a gene to prevent it from working, to prevent it making that plant susceptible. But again, you're looking for 6 to 7 years for mutation breeding and 4 to 6 years if we were to go forward and use genome editing. Now, within NAPIS, actually, the variation is quite small, so we only expect this to have a partial effect, and we expect to, pyramid this with multiple other IPM approaches. But actually, we can look outside Brassica napus, and if we go into Sinapis alba, so white mustard, something you'll consider being, growing as a condiment, actually we can see a much higher level of resistance here. So our Sinapis alba cotyledons are there on the right-hand side, Brassica napus on the left, and this is the level of feeding after 48 hours. And you can see that Sinapis has been barely touched compared to the Brassica napus. So it offers us a promising source of genetic resistance. However, trying to breed this in is really, really difficult. So we're looking at wide introgression here. You can cross these plants, but then you have to go through a rescue of the offspring that come through this. And quite often the offspring aren't fertile, so there is a number of manipulation techniques we have to use. You then go into a period of back crossing to get rid of all those things in the mustard that we don't want within our oilseed rape. So though promising, this can take anything from 8 to 20 years. So looking forward at genetic breeding solutions, actually, we're long-term solutions. But this is definitely something we're thinking about for the future and something I think is going to be important to combat crop pests. Now I'm going to finish by touching, as Sacha did, on novel controls and RNAi-based approaches. So we know these can be powerful and targeted specifically towards our pests. So it is possible to engineer our plant to produce RNAs that will be complementary to certain RNAs within a beetle. And actually, this can knock out the function of core proteins within that beetle, causing up to 100% mortality. So we're focusing on also looking at developing these tools, hoping farmers are going to be able to integrate these technologies. But from a GM side of things, and this doesn't really fit with legislation, so uptake and general opinion is difficult in this area. As Sacha said, we can look at chemical applications, including this technology, and I think it's a good tip for the future. So hopefully I've shown you that we have screening methods for adult and larval resistance. We can identify genotypes that are more resistant and susceptible, and we've passed this methodology to the breeders so they can select during their trialling. We're developing that knowledge on plant-insect interactions and the factors controlling resistance and trying to deploy this into crops. And we've also got promising results in Brassica relatives, though it's complex to transfer these between species. So the breeding itself is a long-term approach, but actually that RNAi-based modification offers a potential genetic route to targeted crop protection and could be utilised much more quickly. So with that, I'd like to end. Thank everyone involved in the project, everyone from Team Flea Beetle that's made this possible, and thank you guys for listening.
Brilliant. Thank you very much, Rachel. And I think, a real positive look forwards there in terms of cabbage stem flea beetle and oilseed rape, albeit slightly longer term solutions perhaps than what we would like, but great to know that there are opportunities out there for improvements. So we're about to head into our last presentation of the Integrated Pest Management session. Just another quick reminder to get your questions in via Poll EV. We'll be heading to the panel session after this next presentation. So up next, we've got Hugh Davis from NIAB, and Hugh is going to be talking us through the UKCPVS project and talking about where the project is going to be going over the next couple of years. So over to you, Hugh.
Good morning. So thank you. I'm just going to give you a sort of background about the UKCPVS and some of our aims, some of the research over the past couple of years, and then just sort of the take-home messages. So for those of you who don't know, the UKCPVS was founded in 1967 after an outbreak of yellow rust on Rothwell Perdix, which was a previously resistant wheat variety. Since then, the survey has monitored a range of different diseases from yellow rust in wheat and barley, crown rust, different mildews, rhynchosporium and leaf spot. So the main aim of the UKCPVS is to identify changes in pathogen populations within the UK, and if we have any new races that are appearing that agronomists and farmers need to be made aware of. We rely heavily on the samples that we receive from you guys, basically. So anyone that's agronomists, farmers, trial operators, breeders and researchers. And the project is funded by AHDB with a contribution from AFA. So some of the other project objectives is to monitor the pathogen populations in respect to the virulence, and this is done by testing on differential lines and currently grown wheat varieties. We're trying to identify novel pathogen pathotypes and how these are significant towards the disease resistance of RL and candidate varieties and how this potentially might be breaking down over the years. And then we're continuously trying to feed this information into knowledge transfer activities such as these, and provide our isolates that we've collected over the years to other researchers where they can develop some extra research for the project. Year on year, we're also trying to continuously develop our methodologies to try and get the most out of this project and get the most information for the agronomists. So the past couple of years, the UKCPVS has focused on 3 different pathogens. So we start off with wheat yellow rust, which is also known as a stripe rust, after the stripe sort of characteristics as on the leaf, and wheat brown rust, and barley powdery mildew. So what happens when you send us in a sample? So every sample we receive, we will put this onto our system and send you an email just acknowledging that we received your sample and what isolate code that sample has been given. For all the samples we receive, we will bulk these up. So for yellow rust, we will select a single stripe, and this will be damp chambered overnight. And the next day, we'll put this onto a pot of a susceptible variety. And then this will be grown up in the growth rooms. And after about sort of 14 days, we'll be able to harvest the spores and put these into long-term storage. For brown rust, it's a slightly different process. So we will harvest a single pustule from the sample that you have sent. And then this will get put onto a detached leaf box. And after about 10 days, we'll then put this onto another susceptible variety of wheat. And again, grown up in the growth room to harvest those spores and put into long-term storage. So every isolate that you send in, we will have a sample of that in our long-term storage here at NIAB. Depending if the sample was a good enough quality to, get a sample from. So from every sample we receive, we select 25 samples, and this is depending on sort of geographical location and the variety. So we try and get a good range from across the UK and from a variety of varieties. Those 25 isolates then get tested in the growth room on a set of differentials, and this is to understand the pathotype of the isolate that we have received. From those 25 isolates, we then select a subset of 5 isolates, and these will go for testing at growth rooms. and these will be tested on the RL and candidate seedling tests. And you'll find the data for that in the young plant rating in the recommended list booklet for oilseed and cereals. And those same 5 isolates up until 2024 were also put on adult plant trials, testing on the RL and candidate varieties, and that is used to validate the yellow rust watch list. So since 2019 up until 2024, we were also working on genotyping yellow rust isolates. So we've genotyped 120 isolates over those years, and that was done by Helen Bates and Anne Webb at NIAB using the Marple pipeline that was developed by Diane Saunders at the John Innes Centre. And the aim of this was to categorise these isolates into their different genetic groups. So from this, we found that predominantly the red group was dominant across the whole of the UK. And this can also be seen across Europe, where, again, the red group dominates. We also found 5 pink isolates over the years and 1 purple isolate. Again, this is sort of similar to what we see in Europe, where about 2.5% of the isolates belong to the pink group, but still dominated by the red group. If you want to find any more information about this, please have a look at the UKCPVS annual reports, and you'll get loads of detail on what we found. So as I mentioned earlier, we're also looking into powdery mildew. So it's sort of a similar process about how we would receive the samples and isolate. So each sample gets isolated from the leaves, and we select up to 25 isolates that we've received, and we'll test these on detached differential leaves in the incubator. And again, this is just to work out the pathotype. So we're going to have a look at the deliverables now. So we're trying to gain an understanding of the race structure within the UK for each of these pathogens, so yellow rust, brown rust, and powdery mildew, and trying to get an understanding of how the disease resistance of these pathogens relate to the RL and candidate varieties at the seedling stage. We're trying to rapidly identify novel pathotypes that have the potential to break down these resistance in the RL and candidate varieties, which is of course vital for the industry. And year on year, we're trying to increase our pathogen isolate storage, which we have going back to the start of the project, and we're trying to just get a range of geographical locations and as many different virulent profiles as possible. And yeah, again, improving methodologies and trying to attend as many knowledge transfer activities as we can. So let's have a look at the 2023 data. So for yellow rust, for the seedling stage, we didn't see any major variety breakdowns. And as we had expected, it was dominated by the red group, which is also known as the warrior group. But we did see a proportion of these isolates falling into the pink and the purple groups. We didn't see any virulence detected for the yellow rust genes 5, 10, 15, and 24. And a low level of virulence was detected in the population for YR8. The virulence was fixed for the following yellow rust genes there, 1, 2, 3, 4, 6, 7, 9, and so on. And only one variety out of all the varieties we tested was resistant to all the 5 isolates we tested, and that was the variety swallow. And we actually found that several of the RLs that had a high resistant rating of either 8 or 9 were actually susceptible to 1 or more isolates that we tested. For example, Graham. So the adult plant trials, many of the RL varieties that we tested were resistant to all the 5 isolates that we selected. And the candidate varieties that were added to the workbook for 2024 and 2025 actually performed quite well. Is that going on? There we go. I've gone back, sorry. Brown rust, skip that one. So we had 15 samples in 2023, and we found a virulence was fixed for the leaf rust genes 1, 10, 13, 14A, 15, 16, 17 and 37. We found the frequency for the leaf rust gene LR24 increased from 12% in 2022 to 40% in 2023. The pathotype here was the most prevalent pathotype we found in 2023, and we managed to identify 5 new pathotypes in 2023. All but one of the RL and candidate varieties was susceptible, and that was RGT Goldfinch, and that was resistant to all the 5 isolates that we tested. For the brown rust adult plant trials, the varieties performed pretty much as I expected when you look at the RL ratings, and the isolate 22024, which had the virulence for LR24, did infect Warrior and Theodore. So now moving into sort of the 2024 season. So over the winter we had quite a mild winter, which meant the brown rust was able to overwinter well. So really we need quite a hard frost for a fair amount of time for the spores to not really continue to the next season. We also experienced a very wet spring, so that meant farmers were struggling to get onto the field with the machinery and give the sprays that we needed to, keep the disease levels down. And this was then followed by quite an early season epidemic of brown rust. So usually you would see sort of samples coming into the UKCPVS around like mid to end of May, but we're seeing them arrive at the start of April, so quite early on. And then we also found a really, really high disease pressure of brown rust later in the season, which I'm sure you're probably all aware of. And we saw quite a few sightings of yellow rust and septoria across the UK, but no stem rust this year. And we think this is probably down to the mild summer that we experienced. So how's the project changed from 2023 to 2024? Well, we stopped performing adult plant trials in 2024 and instead put the focus on the seedling stage, growth room-based studies for yellow rust and brown rust. We introduced a new reduced differential set, which we used to pathotype the rust isolates, and we increased our isolate numbers and testing on the full set of recommended list and candidate varieties. And where it was possible, we did try and improve our in-season reporting. For wheat and barley powdery mildew, we didn't have any unusual sightings or reportings to indicate any sort of major changes within the UK for the wheat mildew populations. And although in 2023 we did see some susceptibility in spring barley up in Scotland, we didn't actually receive any samples in 2024. So now let's have a brief look at the 2024 results for yellow rust. So we received 68 samples from 16 counties across the UK and 25 different varieties. The graphic here just shows you a heat map of the locations where we received samples from. So you can see most of them came from Lincolnshire, Norfolk and Cambridge, but we have also received samples from Scotland and Northern Ireland. So we had a good geographical range of samples this year. So from the 2024 tests, we haven't seen any major variety breakdowns in the seedling stage growth room trials, and we didn't see any virulence detected for the yellow rust genes 5, 8, 10, 15 amongst the 25 isolates that we selected. 17 of the RL varieties and 8 of the candidate varieties were resistant at the seedling stage, and you can see the ones here in blue are your recommended list, and the ones in green are your candidate varieties that were resistant. This table here just shows you the varieties and how many isolates they were susceptible to. So for yellow rust, 14 of the RL and 15 of the candidate varieties were susceptible to 1 but up to 16 of the isolates we tested. So you can see KWS Sciate was susceptible to 16 of the isolates we tested. So for wheat brown rust, we received quite a high number this year, predominantly down to the weather conditions that I was speaking about earlier. and we received 65 samples from 11 counties and 31 varieties. Again, mostly sent in from Lincolnshire, Norfolk and Cambridgeshire. So the results for the 2024 brown rust so far, the seedling stage growth room trials showed that we analysed 10 different isolates from 7 counties and from 10 different varieties. So we had a good geographical range and tried to use as many different varieties to get a good representation of what's going on across the UK. We found virulence for the genes LR1, 3Ka, 13, 10, 14A, 15, 16, 17, 26, 28 and 37. And we found a single isolate with the virulence for LR24, and this was sent in from Lincolnshire. Most of the RL and the candidate varieties were susceptible to at least 3 of the isolates that we tested. The only variety that was resistant to all 10 was the candidate RGT Goldfinch, but we did also see that the candidate variety Diamond was resistant to 9 out of 10 of the isolates. So again, this table here shows you the varieties and how many of the isolates we tested were susceptible. So most of them were 3 or above, and you can see quite a few were susceptible to all 10 of the isolates we tested. So sort of just your take-home messages from today. Over 2023 and 2024, we didn't see any major breakdowns in the yellow rust, and the red group remains dominant across the UK, and our population of yellow rust is quite diverse. So, we encourage you to monitor your crops and spray if it's required, or send us in some samples if you see anything unusual. For brown rust, out of all the RL and the candidate varieties, only RGT Goldfinch was resistant at the seedling stage, and we saw increase for virulence of the LR24 gene rose from 22% to 40% in 2023. Please refer to your AHDB booklet for your resistance ratings, so your young plant and adult data, and have a look at the AHDB watch list to make your variety choices. And if you're using a variety that's got a good resistance rating, try and reduce your fungicide use and have the confidence to do so. Pathogen populations within the UK are remaining quite diverse, so keep a close eye on your crops and spray if needed. And send us in a sample, which I'll get into next. So always got to try and get a plug in to get some more samples for the next year. So when you're sending us in a sample, please try and use a brown envelope so we get the sample in at the best sort of quality, and make sure you only use the words free post UKCPVS. If you use anything else, sometimes it can get held up by the Royal Mail, and by the time it gets to us, the sample isn't the best, and we struggle to get a sample from that for isolation. I also want to make you aware that on the 13th of January, we also have the annual stakeholder meeting. So we'll be going into more details about the 2024 season and how the project is going to be sort of changing in the next couple of years. And we've also got some guest speakers, so we'll be covering topics about ergot and BYVD. So if you want to find about how to register for that, go on the AHDB website and look for the UKCPVS stakeholder meeting, and you can register there for the event. So I'd just like to say thank you to the following people, in particular Kostya Kanuka, who's the current UKCPVS lead. And I'd also like to say a special thank you to Amelia Hubbard, who sort of did all the research for the past few years, but retired earlier in the year. So thank you for your time, and hopefully I'll see some of you in January. Thank you.
Brilliant. Thank you very much, Hugh. It's really great to see the UKCPVS project adapting to those changing challenges in terms of disease resistance. And yeah, just another plug, would really encourage you to send your samples into them. It's really important. The more samples they get, the better they can map that resistance. So yes, please do send them your samples. And so that is the last of our speakers in the integrated pest management session. So we're now going to move over to the Q&A. So thank you to all of you who submitted your questions via Poll EV. There may still be time to submit a few more, so please do get involved with that. So I'm now going to hand back to Anna Reynolds, who's going to be chairing the Q&A session for you today.
Brilliant. Thanks, Henny. We've already got quite a few questions coming in, which is fantastic, so keep them coming. To kick us off there, I've got two questions. One is for you, Hugh, and really I'm thinking back on about our farm in the previous season. I think actually our milling weeds did quite well in proteins because of brown rust, because the yield was so affected last season. How do you see those dynamics coming into the 2025 year? What's coming up out there on the ground?
Well, I mean, it depends on sort of the weather conditions we experience. If we're going to have another mild winter, you're going to see probably another outbreak of brown rust next year, potentially. So we'll see.
We'll see. Okay, yes. Brilliant. And the other question I have for the whole panel is kind of thinking 5 years from now, what development are you most excited as you think is going to make the most difference on farm across all the things you've just mentioned throughout your sessions? And whilst you think of that, I'm just going to encourage everybody to keep sending questions in through Paul EV, and you should have a QR code on the screen that you can scan for that. So 5 years from now, what excites you most?
For me, for flea beetle, I'd say it'd probably be the development of good decision support tools, help growers and agronomists work out what's happening with the pest, when it's happening, and what they can do to sort of inform them a little bit about what they can do to help manage it a little bit better as well. And hopefully maybe we'll also start seeing some more products coming on that people can use as a responsive treatment as well.
Thank you. Mark, Rachel?
I think that there's a huge opportunity for better integration of the different approaches, and I think that's what we'll see more of over the next few years. So where we're managing varieties according to their benefits, and then we can build that in and complement those with other approaches. I think we'll see more of that in the next few years.
If you think about excitement, maybe worried is more what I would say is the whole, with oilseed rape and cabbage stem flea beetle, we've got that first glimpse of what happens when you withdraw a chemical from the system. And actually, you can see the chemicals dropping off and us having to move towards these different ways. And I think that full integration is going to be necessary to make things, work. But I think it's very exciting in terms of cabbage stem flea beetle and oilseed rape, because we're at the forefront of pulling together this knowledge and applying those different techniques. I think RNAi offers us a really amazing potential tool, because you're looking at targeted pesticide products then. So, only affecting the beetle. So I think, there's some very interesting results that are going to come out there.
Thank you. Got a question about drilling date. Is it not time that we got less hung up on date of drilling when it comes to cabbage stem flea beetle? Is our first question here. In our experience, temperature, moisture and wind speed have far greater effect than date. The conditions post-drilling will have far greater effect on activity, and last time I saw flea, it didn't have a calendar. What do we think to that?
I mean, I think with all of these sort of culture control approaches, there's not going to be a hard and fast answer. There's always variability in the system. There's always biological variability, and absolutely right. That does come down to things like temperature. They are insects after all. But I think there's been a lot of work done over the last decade or so that shows that sow date has a big influence, and you can reduce your risk by choosing your sow date carefully and matching that to conditions as well, so soil moisture and all these sorts of things. But there will be occasions when it doesn't work, and that's why where we start looking at stacking these culture control approaches, we can improve our chances that if one part of it doesn't work, that another part of it will. So there will be some certain times when the sow date, you sow late or you sow early and you still lose a crop. But if you put some other things in there, then you might also further reduce that risk.
Yeah, So again, go back to that holistic
Yeah,
approach. And it's interesting on your graph, there was a much more sort of sporadic emergence in recent years, like
Yes.
2024.
This year, yes.
So it'd be interesting how that develops into the future. Are we going to see
Exactly.
more of that variation?
Yeah, yeah. And it's the data, so the monitoring that we did this year is good because it kind of gives us a picture of what happened this year, but also that's a useful data set for further developing decision support systems to help predict that migration.
I think it's always a challenge that where you've got good experience that sow date is important, and you can see that drives early or late drilling, but it oversimplifies the complexity of what's going on. And then as we get more data, we can produce these more complicated models that incorporate that moisture, wind speed, and all these other factors. But then, because they're more complicated, they have more assumptions, and they need more testing to be robust and applicable. So that's where we are in that journey. We've got a simple rule, and we want to get to a more advanced decision support system that supports more accurate decisions, but that just takes time to get there.
Okay, thank you. Going back to UKCPVS, a question for you, Hugh. What samples are you after? Is it plants or leaves? Give us a bit more of an idea.
Leaves would be, leaves are ideal. Okay. Perhaps a small handful with some sort of varying disease levels on there, some quite high, some a bit smaller, so it's easier for us to sort of harvest that single stripe of yellow rust or the single spore of pustule, of brown rust. And if you go onto the AHDB website, you'll find a registration form. So if you can fill in that with all the details of the crop, the variety, any treatments it's received, and sort of the disease level, and then we can process the sample from there.
Thank you. And on cabbage stem flea beetle again, a question from one of our viewers is, do we think the number of the beetles in trailers at harvest could be used as a forecast for the next growing season?
I think it can be an indicator, for sure. I mean, if you're not seeing many there, then maybe you're in for less pressure. But again, I don't think it's a hard and fast rule, just like we've been talking about with sow date. We had a situation a few years ago when very few flea beetles were seen on the trailers, and a lot of people asked me exactly the same question. Are we in for a good season ahead? And I kind of I said, be cautious. And lo and behold, it was a bad season. So, it could be that the flea beetles have moved already from the crop by the time the harvest has happened. For whatever reason, they might be slightly ahead, a warmer spring, a warmer earlier summer. And so they might have already moved into their sort of sheltered areas for the estivation. And so you're not seeing them as much on the grain trailers. So there's all sorts of things that could be happening. Even if you do see lots, it could be that, autumn is very wet and very cool, and therefore that's not very good for the migration. And for adult flea beetles, they don't like being very active in those conditions. So again, you might not actually see the high level of pressure that you would expect.
Thank you. And would you expect cabbage stem flea beetle resistant genes to offer long-term durable resistance, or would you expect that they'll keep breaking down to resistance? How does that dynamic work?
I think that will understand that the basis of the underlying resistance will relate to that. So we're looking at identifying resistance in a number of different ways and trying to understand the interaction and the mechanisms there. And that way you can select for multiple paths to get to resistance. If we can pyramid multiple forms of resistance or act on multiple pathways in the plant, then we can prevent that resistance breakdown. So there's still a lot we don't know about why they prefer certain plants, about why larvae do well, maybe in some better than others. So, just gaining that knowledge, I think we can work towards something that's much more durable than deploy a one-hit, one-shot effect.
Yeah, absolutely. And you mentioned antizoonosis and antibiosis. Ideally, are you looking for both, or is one or the other, you think, more, offers more of an answer?
I think you need to look at both of those. It's quite obvious we've got anti-zoonosis when you look at our difference in feeding preference, because they prefer to feed on one and not the others. Getting to the antibiosis resistance is much harder, but we know if we go out into that Synapis alba, the larvae do less well than they do in Brassica napus. So there is something there that's preventing the growth of those larvae. So two very different types of damage. We need to combine everything we can to protect the crop.
Thank you. A question for Mark on the screen. Do you know how many farmers are currently implementing SFI IPM action on farm? And do you think the payment is enough to encourage people to use it? Do you think it's really making the difference?
I don't know exactly how many people are making the claim for the SFI action. I do know that in our tool that I gave a highlight on, over the last 12 months we've had in excess of 1,500 plans made, and I know that there are more other tools available that are also being used. So we know that there are a lot of plans being made that are compliant with the action. In terms of whether the payment is a big enough incentive, for me, I think that an IPM plan is kind of the cornerstone of how you bring all these things together and how you identify which tools and resources you need to bring into your farms. So really, the incentive is there to get people to use them, but the benefits are going through that process for each farm. So the benefits are there for the farmers already. That IPM incentive is really on top of that to make the time to sit down and go through the plan.
Thank you. And another question, really touching on economics of growing oilseed rape, but also on some of the things that you touched on, Sacha, around companion crops, trap crops. We talked about sowing dates already. Do you think we currently know enough when we're stacking enough of those to really make a difference in a particular farmer's scenario?
I think we need to know more, but we've done a lot of research on companion crops, trap crops, sow dates. There's other areas we want to do more research on to see if we can build those in, stack those things in as well. There's no guarantee. You can put all these things in place. There's no guarantee that the crop will survive. But I speak to a lot of growers who are trying these things and do get a successful crop year on year. Maybe they're lucky, but maybe they are doing the right things. It's about reducing the risk at the moment. Our focus is about reducing the risk of the pest being at problematic levels. It might be there, but is it at a damaging level? And also about mitigating against the damage. So we've got varietal choice in there as well. In terms of the characteristics, the varieties, good autumn vigour, good spring vigour, these things, we think that they're helping as well. I would like to think that people can still try growing oilseed rape. This year, I would say, there's a lot of healthy oilseed rape crops out there, and we've got low flea beetle pressures. But even in the areas where there are high flea beetle pressures, I haven't heard too many reports of crops being badly damaged.
Thank you. And going back to that, just burnt into my sort of retina is the red dot next to cultivation on your slides. So a question from someone here is they believe that post-harvest cultivation can lead to massive reduction in cabbage stem flea beetle, perhaps 90%. Shouldn't this be widely adopted? Is there evidence? Is this right?
Yeah, well, so firstly about the cultivation thing and the red dot, this is something that AHDB tried to research in the latest project, but it's very difficult to get people to carry out cultivation trials on a sort of a plot level, because it tends to be what people don't want to have is different levels of cultivation going on in their field. But it is something we definitely want to look at more. The NIAB work was about cultivating post-harvest, and absolutely there were some really encouraging results there. They were sort of their initial findings, so we definitely need to do more work to establish how much benefit it's really providing. And the reason, I think people maybe should give it a go, but the reason I'm kind of giving a word of caution here is because a lot of the parasitoids that are effective against cabbage stem flea beetle, they are overwintering as pupae in the soil. Okay. And those pupae will be in the soil at the time when those cultivations are happening. So we do risk actually damaging the parasitoid populations. So we've just got to make sure that it's working and it's not having counterproductive effects as well.
Absolutely.
That's the other thing.
Yeah, I think going back to what we talked about is really that IPM, your plan for your farm, really having a look at all the different options and stacking them and thinking about how that applies to your particular context on farm and making that decision for your context and your cropping. Thank you for that. And a question going back to the UK cereal pathogen virulence survey. We've got samples being sent in. Obviously, people are looking around, if something looks unusual, you're relying on somebody's definition of that. Is that the best way to do it, do you think? Are you getting a really good sample scientifically? And also, are you receiving samples from the RL trials? And have you made any changes to the sampling this season?
So we haven't made any changes to the sampling, but we could do with a better range of geographical locations of them having samples received from. So perhaps in the next meeting in January, we could, suggest to the RL operators that perhaps they could send us in samples if it's possible, because we understand, farmers, agronomists are very busy at that time of year. It's difficult to get samples and spend the time doing that. In terms of looking for something unusual, if it's a variety that's got a good disease rating and you're finding yellow rust or brown rust, then that's something we're quite interested in finding.
Thank you. And I think we're going to take one more question about cabbage stem flea beetle. Low numbers of cabbage stem flea beetle adults this autumn and a small area of crop. Do we think the pressure will be low for the next few seasons? Should we be going in? If we've been taking a break from it on our farm at home, should we be going for it next season?
Well, as I say, this could be the reason why we've got lower pressure this year, because the actual areas of oilseed rape have been lower in recent years. But these cover crops with brassicas in them, if the flea beetle are able to complete their life cycle on those, then that's going to keep the populations high, because they will just be arising out of those crops. If those winter brassicas in those cover crops are being removed early enough before the flea beetle, the larvae, can complete their life cycle, so for instance maybe February time, then you will you will break the life cycle. So that way we might get the best of both worlds, but it remains to be seen.
Okay, thank you. And a final question more generally. We've got various types of IPM plans, and if you have one, we've got a few of them online. Are they sufficient? And do you think there's anything missing currently from the current variety of IPM planning tools that we have available?
The main purpose of an IPM plan is to think through what you're doing and to identify the tools and resources you need for your farm. So to a certain extent, any plan is good. So you can use whatever plan really suits your approach. The tool I presented is structured so that it kind of prompts you to different resources and tools and technical information, but that doesn't mean that that's the best one for you to use. So I would just encourage everyone to find the tool that best suits what they want to get out of it. It helps them engage with their farmers and find the best approach for that farm.
Okay, and how do we do that? How do we find? Do we have to try them all, or is there anywhere? Do we have a summary somewhere that highlights the differences, or anything like that?
At the moment, there's only 2 or 3 extensive planning tools around that I'm aware of. So there's the one that I presented. There's the NFU one, the VI one, and then various agronomy institutes will have their own. I think there's a Leaf Sustainable IPM guide as well. So there's a handful of them that are out there. So it shouldn't take long to have a look at them and see which one works for you.
Okay, brilliant. Well, you named quite a few there, so we'll, go and have a look at those and select the one that works best for your situation and your customers. We're going to finish the Q&A here. What we'll do is we'll collect all the questions at the end, and we'll disseminate some of the answers on anything that we haven't touched in the Q&A sessions live today. But now we'll go to a comfort break, and we will be back at 10:45 with our next session looking at plant protection product doses and also resistance. Thank you very much, everybody, and we'll see you at 10:45.
Hi everyone and welcome back to the Agronomy Conference. So our second session of the day is going to be looking at pesticides, so we're going to be looking at resistance and performance. So our first session looking at pesticides, we're going to be looking at fungicide performance and resistance. So we have Stuart Knight from NIAB, who's going to be talking about wheat and barley, and then we've got Philip Walker from AHDB, who's going to be talking us through oilseed rape. So kicking us off, we've got Stuart Knight, so handing over to you, Stuart.
Thank you. Good morning, everyone, and welcome to the 2024 Fungicide Performance presentation. As mentioned, this is a bit of a joint effort between myself and Phil Walker of AHDB, who will be covering the oilseed rape. But the project itself is a collaboration between all the organisations whose logos appear at the top of the slide. Before we launch into looking at any of the data, a little bit of background around choosing fungicides. So matching fungicides to the disease risk is really important, and that's a function of the variety, the sowing date, the location, and the local weather. Mixtures and alternations of fungicides with different modes of action, different fungicide groups, are often the most effective way of tackling disease and reduce the likelihood that fungicide resistance will develop within our pathogen populations. So we shouldn't underestimate the threat posed by resistance to the performance of fungicides, and it's essential that we take resistance management seriously when planning our fungicide programs. And there's more information about that on the Fungicide Resistance Action Group page on the AHDB website. So let's kick off with wheat and barley. And before we look at any of the charts, just a quick reminder for those who aren't familiar with it, of the trial methods that we have for this work. So in order to maximise the difference between the treatments for each disease, we pick high-risk locations, we pick highly susceptible varieties to the target disease, and we look at applications at a single spray timing, which depends on the disease. The dose response graphs that I'm going to show you will show average performance measured across a range of sites, a range of seasons, or a range of leaf layers. And sometimes we apply cover sprays that are not active against the target disease, and those are used to reduce the effect of other diseases in the trial. A good example this year was the effect that brown rust crept into some of the septoria trials, and we used some cover sprays to control it. Now, some of the diseases I will show you will be protectant and eradicant activity charts, and this is what we mean by this. So on the left-hand side of your screen, you have a picture of a cereal plant being sprayed where leaves 2, 3 and 4 are emerged, 2 most recently, 3 and 4 obviously for a little while. When we come back to assess that crop 3 or 4 weeks later, the flag leaf will be out, leaf 2 will have been out for some time. So the disease that's controlled on leaves 1 and 2, the flag leaf and leaf 2, will largely be protectant activity. The disease control we measure on leaves 3 and 4 will be largely eradicant activity. Sometimes we have a combination of those two, and that's when we described it as mixed activity. Before we launch into the actual control data, a reminder, if you needed it, of the 23-24 cropping season, and in particular the impact that the weather had on both on crops and on disease pressures. So as you will well remember, it was a very wet autumn, winter and spring last year. The left-hand chart on the slide shows you the Met Office winter 2024 rainfall amount as a percentage of the 30-year average. And you can see that pretty much the whole of the UK, with the exception of Northern Ireland and Western Scotland, experienced very, very wet conditions, up to double the normal rainfall over the winter. And that meant obviously problems with soil wet conditions, but also meant that a lot of the crops were either drilled quite early or drilled very late. There wasn't many crops in that middle window. Not only was it very wet, it was also a very mild winter, with in particular very few night frosts. So the right-hand chart on the slide is the Met Office winter 2024 minimum temperature, mean minimum temperatures, and that's the anomaly against the 30 years again. So you can see a huge area of red, pretty much covering the whole of the UK except northern and central Scotland. And there, particularly in East Anglia and central England, we saw temperatures anything up to 2 degrees above the normal. And that led to, for example, in wheat, some very high levels of septoria and brown rust. So let's move on to our wheat trial sites in 2024. There were 10 of them, 7 focused on Septoria tritici, 1 on yellow rust, 1 on brown rust, and 1 on fusarium. The Septoria tritici trials, some had T1 spray timing, some were T2, but the data can be combined. And then for the yellow rust, it's a T1 timing, for brown rust, a T2 timing, and the fusarium obviously is a T3 timing trial. And the varieties that we chose, again, for those trials match the diseases that we're targeting. Now, in the fungicide performance trials, we test a vast range of products, both in any one year and over time. And this table summarises the products that were used, and which I'll be showing today in the presentation. So I'll go through the list. So on the left-hand side, we have the product name. On the middle column, we have the active ingredients, and then on the right-hand side, we have the mode of action. So starting at the top, Arizona and Proline, our Arizona being our multi-site, our Proline being our sort of standard azole. We've tested those over many years against Septoria, and they present a bit of a benchmark for us when comparing the newer products against. We only test those now in the Septoria trials at a single full dose, 100% dose. Moving down the table, we have a couple of azoles that we've tested, mefentrifluconazole or Marisa, and then tebuconazole Toledo. The tebuconazole was only tested in the yellow rust trial. Then a series of other straights, so Pectiga, the fenpicoxamide, a QOI fungicide, 4 SDHIs, Elatus Plus, Imtrex, Vimoy, and Miravis Plus. And that's the first time at the Agronomy Conference we've presented data for Miravis Plus. We released some earlier this year because it came out just too late for last year's conference. So this is the first time we've put it up on the screen. And then 4 mixture products, 3 of which you'll be very familiar with, Revistar, Ascarex Pro and Univoc, as mixtures of DMIs and SDHIs, or in one case a DMI and a QII for Univoc. And 1 new product, recently approved, Ipresso, which is a mixture of isoflucipram plus prothioconazole, so essentially Vimoy plus Proline. In addition to the products that I've got on the screen there, a further 6 unregistered products were tested in 2024, and we will release data from those as soon as they become approved. There is also another product we've tested in the fungicide performance trials, which we won't be showing today because it wasn't in the 2024 trials. That's a mixture of isoflucipram and fluoparam and prothioconazole with the name Plaxium, and we will be releasing some data on that after the conference. A reminder that regardless of how we test the varieties, test the products, many of them we test as single actives. That's not how they're designed to be used. We should always use those products in a mixture with at least one other fungicide from a different fungicide group that has activity against the target disease. So, on to our wheat Septoria triticide efficacy data. So I'm going to show you some data from 5 trials in 2024, and that's going to be protected data. We did collect some eradicant data and some mixed data from a couple of the trials. That goes into the over-year data sets, but I'm not showing that today. So let's move straight on then to the Septoria protected data from 2024. As I say, 5 trials, and I'll take the opportunity on this first slide just to run through the layout of the chart so you're familiar with the rest of the presentation. So you'll notice there's 2 charts on this slide. It's the same data sets. We've simply split them into 2. On the right-hand side chart, you have the straight active ingredients. On the left-hand side chart, you have the mixture products. If you look at the bottom axis, the x-axis on the 2 charts, that's the percentage of full label rate. And you will notice that we test the products at quarter, half, full and double dose. Now, just to say from the start, double dose obviously is not something you can do commercially in the field. We use that dose in order to help us plot accurate curves. And when we put these charts up onto the AHDB website after the conference, you will notice we limit the axis to 100% to reinforce the fact that's the highest dose you can use commercially. And obviously on the y-axis, on the left-hand axis, we have our measure, in this case, percentage disease. So let's move back to the right-hand slide and start with the straights. So you can see the 2 single points, the Arizona and the Proline, which are our sort of benchmark values, giving us about 40% control, 40% protection against Septoria. And then our new products, which are all the curves, and you see the standout product there is the Miravis Plus, giving the highest level of control. Very close behind that is Pectiga, our QII, also giving very good level of control. And then we have the Marisa and the Vimoy, giving us better control than the Proline or the Arizona, so around 60% control, something like that, but not quite as good as the Miravis Plus or the Pectiga. And then on the left-hand side chart, our mixture products, so we have Ipresso there and we have Univoc. Those are giving us very similar levels of control to one another and marginally better than the straight actives that are their component parts. Moving on to the yields for 2024, now I've split the 5 trials that I just showed you in terms of Septoria protection into 2 sets for the yields. The first set of 3 trials is those trials where we had little or no brown rust in them. So this is really reflecting the Septoria control fully. And again, looking at the right-hand side chart, first of all, our straights, you can see that all the new products giving us, newer products rather, giving us better yields than the Arizona and Proline. The Marisa and Vimoy, almost inseparable as they were in terms of septoria control, but the star performers, again, really the Miravis Plus, and very close behind that, the Pectiga in terms of the yield response. Obviously, with the yields, it's up the other way, the curves, because the higher the dose, the higher the yields we get. And then on the left-hand side, our mixture products. Again, some benefit from the mixtures compared to the straights. The difference between the Univoc and Presso in terms of yield, slightly bigger than it was in terms of disease control, but nevertheless, both products performing well from a yield perspective. So that's the 3 trials without any brown rust. Now here's the 2 trials that did have some brown rust present, as well as obviously a lot of septoria. And you can see a couple of differences here. One is that the curves are a lot closer, particularly on that right-hand chart where we have the straights. You can see the separation between those straight products is much less where we had the brown rust. And you will notice that the Miravis Plus, which was the top performing in terms of yield, is now only on a similar line to Marisa. So the presence of extra disease has impacted on the relative yield performance. If you look at the left-hand side, you can see, again, the Univoc and the Presso, slightly closer than they were in the septoria-only trials, but again, the Univoc just having the slight advantage. But again, the mixture products giving us high yields at any of the straights from their yield components, from their active components. So that's data for 2024. I'm now going to show you the over-year data for septoria. So the first set is the septoria protection from 16 trials over the last 3 years. So on the right-hand side, again, we have our straights looking very similar to the 2024 data, with the Miravis Plus closely followed by the Pectiga as the most effective, and then the Vimoy and the Marisa giving us good control, better than the Arizona and the Proline, but not quite to the level of the Miravis Plus and the Pectiga. And then on the left-hand side, we have an extra mixture product now on this chart, which wasn't in 2024 trials, but was in the other 2 years, and that's Revistar. And you can see the Univoc just about leading the pack there on the mixtures, Revistar a little way behind, and Vimoy roughly in the middle between the 2. And again, some advantage compared to any of the straights. So we have some eradicant data from the 3-year overview as well, 4 trials of eradicant data. And the key thing to point out here is that all of the products giving us less control in this eradicant data set than they were in the protectant set. The rank order is very similar. So again, the Miravis Plus, the Pectiga, the strongest, the Marisa and the Vimoy very similar to one another, and all of them better than the Proline. We don't include the Arizona here, the Volpet, because it's not an eradicant fungicide. So, but the main difference is the products are all giving us slightly inferior control. And I think it's an important message that timing is critical regardless of how good the fungicide that you are using. And on the left-hand side, again, the rank order performance of the products, so the mixture products on an eradicant side, very similar to the protectant performance. Yields, again, as you'd expect, this is just septoria-only trials now, so not any trials that had any brown rust in them. And again, the products that were really good against septoria typically are giving us the best yield, so the Miravis Plus, closely followed by the Pectiga, and again, the Univoc on the mixture side leading the yields. Now, in addition to using the fungicide performance trials to track the efficacy of fungicides, we also use them as a source of samples to monitor sensitivity of the pathogen populating to fungicides, led by my colleague Nicola Hawkins at NIAB. And this, we've been doing this now for quite a few years, so we have some long-term data sets. So what you have here is a couple of charts. The left-hand one, which is described as prothiodesto, think of that as prothioconazole, and then the right-hand side, mefentrexfluconazole, that's obviously Marisa. So we have taken samples selected from Rothamsted from the early season before any fungicides have been applied. We have about 20 years worth of testing now. And the charts you have in front of them are looking at the sensitivity of the isolates in the population. So the x-axis is the sensitivity, the EC50, and basically the higher the EC50, the lower the sensitivity. And the right-hand axis, the vertical axis, is the proportion of those isolates in the population. So when we tested isolates from 2003 and indeed 2008, they were all very sensitive, all with EC50s of less than 0.01. But over time, that population has shifted left to right. And you can see that now, for the last 3 years, the majority of the population has had EC50s of more than 0.1, and increasingly, a number of isolates have EC50s dosages of more than one. So we have seen that left to right shift, in the decreasing sensitivity over time. The good news is, in the last 2 or 3 years in particular, we haven't really seen much change. It's more or less in the same place. For mefentrifluconazole, we have a slightly shorter data set, but again there you can see even between since 2019 through to 2024, a slight shifting of the population and moving of the population from left to right towards slightly lower sensitivity, higher EC50s, and we see some of those isolates appearing in the last 1 or 2 years, even for mefentrifluconazole with EC50s above 1. Another way we can track the performance of fungicides over time is directly from the fungicide performance data. So we've got a number of charts here looking at now percent septoria control. This is not percentage disease, it's percent control, so that's where it goes up to 100. And for the last 6 years, and we've put some products on here that have been in most of those 6 years of trials. I think because there's a lot of data on this slide, just focus on the top curve, which is Miravis Plus, which has been in all 6 years. You can see that in the first couple of years in that data set, 2019, 2020, that upper line giving us roughly 90%, 95% control, and still very good control in 2021. In the last 3 years, we've just seen a little bit of reduction in that average level of control, more down to around the 80-85% control mark. And in fact, if you look at most of the other curves, which have been in, most of the other products that have been in several years, you can see again, there's a tendency as we go left to right across the screen for the curves to be lower and the levels of control to be lower. There is monitoring work on the isolates that takes place to back that up. And again, similar to what I showed you with the azoles, Bixafen, which is the SDHI in Aviator, you can see that between 2003 and 2024 on that left-hand chart, the isolates moving left to right, moving towards higher levels of EC50, lower sensitivity. But again, not so much change in the last 2 or 3 years. Most of that change happened around that 2017-2018 period. And then for the pitiflumethafen, again, shorter time scale, but even for that, for the Miravis Plus, we've seen some shift evident in the last 2 or 3 years compared to first testing in 2020. Okay. That's Septoria, that's our big chunk of data. Now let's move on to the other diseases. So I'm going to cover, first of all, the rust trial data for wheat in 2024. 1 trial at Terrington on yellow rust, 1 in Cambridge on brown rust, and we have yield data from both of them. Let's start with the yellow rust. Again, similar layout of charts as before. On the right-hand side, we have our straights, and actually all of the products here giving very similar levels of control in 2024. Very difficult to separate out those curves, really. And you'll see that the green dotted line here, that's the Toledo, the tebuconazole. So that's the first time we've had that back in these trials for a while. But one of the reasons for putting that in was to give us some idea of how the newer fungicides perform against what many people consider as a sort of a bog standard, standard for yellow rust control. And then on the left-hand side, we have our mixture products. So we have Ascarex Pro, we have Univoc, and we have Impresso. And again, all those products performing very well, better than any of the straights. Impresso, in particular, being a new product, encouraging to see the yellow rust performance there. Looking at the over-year data, so that actually spreads the products out a little bit more than the one-year data. So on the right-hand side, our straights, you can see that the products pulling apart, the curve's a bit pulled apart there, with Latus Plus, the standout SDHI performer, the Marisa and Vimoy giving us intermediate control, quite similar to one another, and the Pectiga giving us useful control, but not to the level of the SDHIs. And then on the left-hand side, we have our mixture products, and here we have Revistar in as well as Univoc and Impresso, and you can see again, Univoc and Impresso very similar in terms of control, better than any of the straights. The Revistar also giving us good control, but not quite to the level of the Univoc and Impresso. And if I show you the yield data from those 3 trials over 3 years, as you'd expect, that very much reflects the level of yellow rust control. So we have Latus Plus giving the best yield responses, the Marisa and Vimoy quite similar in the middle, and then the Pectiga, some response reflecting that a level of control, but not as good as the other products. And then on the left-hand side, our mixture products. Again, the 2 products that gave us the best control over the 3 years, the Univoc and Impresso giving us the highest yields, and the Revistar slightly behind that. But again, some advantage in terms of yield from the Revistar compared to, for example, the Marisa, showing the benefit of the mixture compared to the straight. Now I'm going to move on to brown rust. So now there's a slight change in the slide. Now all the products are on a single slide. So there's only one mixture product in here, which is the Impresso, the blue dotted line. and all the other products on this chart are straight, so they're on one chart. And there's quite a lot of curves here, so I'll take a moment to talk through these. So if you look at the bottom curve, the lowest curve, the one that gets us down to under 5% disease, that's the Imtrex, so our fluxyperoxide, and that was a very good performance in 2024, which was a very, very difficult year for brown rust control, a lot of high pressure. Now, I wouldn't necessarily expect Imtrex to perform as well as that in every trial. This is just one trial, but it did perform very well in this year's trial. And then above the Imtrex, there are 3 products that are on pretty much the same line. So the Oppresso is one of them. That's the blue line with the dashed line. And you can see that's just deviating away from the other 2 above between the 100 and 200% dose, so above the full dose. And there are 2 products that are on identical line, Marisa and Vimoy. So that line above the Oppresso line is actually 2 products. You just can't see it because they're on exactly the same line. So they're very similar in performance. And then above that, you have the Pectiga, giving us a modest level of control, and then 2 products which were giving us a relatively low level of control and a bit variable in case of one of them. So that's Miravis Plus, and the other one is Elatus Plus. Now, the Elatus Plus is the one that's giving us quite variable control there. And this is interesting because when we first tested Elatus Plus against brown rust many years ago, it was extremely effective against the disease. And what we've seen over the last few years is a decline in its efficacy, which we're still trying to understand what's behind that, whether there are changes taking place in the brown rust population that can explain that. Because the Elatus Plus performance is changing over time, we've excluded it from the 3-year overview because if we were to present that now, that 3-year overview wouldn't necessarily be representative of the performance today. But we do have all the other products on the 3-year trials, and actually we have one other change here, which is Imtrex. Rather than Imtrex in the 3-year overview, we have the Revistar, which is, again, the standout performer on this data set. The bottom line, the Revistar obviously containing fluxyperoxide, as well as the mefentrifluconazole, giving us very good levels of control in that 3-year data set. Again, the 3 products, the Oppresso, the Marisa, and the Vimoy, very, very effective and very similar to one another. You can just about see the lines separating this time, but very similar levels of control. And then the products giving us lesser control, Pectiga and the Miravis Plus. So useful activity, but not anywhere near the levels of the other products. Our final wheat disease is head blight, and that's a combination of Fusarium and Microdocium. And we have 3 trials in that data set from those 3 years. And we have 3 products we've tested or can present today, Marisa, Proline, and Miravis Plus. And you will notice the Miravis Plus in this trial set, the full dose of the Miravis Plus is 3.2 litres a hectare. So it's higher than what we use in the foliar diseases, which is only 2.65. So Miravis Plus giving exceptionally good control of the head blight symptoms, even down at relatively low doses, and superior to what many people would think of as a bit of a standard for the head blight control, which is thiaconazole Proline, which is giving us around 50% reduction compared to the untreated. And then the final one, which is the Marisa, again, giving us a useful reduction, around 40% control relative to the untreated. So I say that data set this year is a mixture of Microdocium and Fusarium symptoms, and we didn't get any usable DON data for this current season. So, quick summary for wheat then. So in terms of Septoria, Miravis Plus and Pectiga, the 2 straights, and then the 2 mixtures, Oppresso and the Univoc, achieving the highest levels of Septoria control. The Vimoy and the Marisa, good levels of Septoria control. We've seen that over several years, but not quite to the level of the products in the bullet point above. We saw good yellow rust control from the azels and the SDHIs we've tested, but over the years, it's tended to be the case that the mixtures, so Ascarex Pro, the Oppresso, the Revistar, and indeed the Univoc have been more effective overall in terms of the disease control than any of the straights. It was a very, very challenging year for brown rust control. Imtrex, fluxyperoxide, gave that very good level of control in 2024, and we've seen similar levels of good control from the Revistar in previous years. But we also have Oppresso, Vimoy, and Marisa, which were very, very effective again in 2024. We did see some further small shifts in fungicide sensitivity, although the Rothamsted data didn't show much change. We know that 1 or 2 sites have shown some interesting isolates, a little bit less sensitive isolates, so things are still moving, but quite slowly, and we've not really seen any dramatic changes this year, so nothing too much to worry about in a single year. But the message really remains very simple, which is that mixtures of fungicides from more than one fungicide group are really, really important. They provide the most robust disease control. They provide better yields, highlighted by the sectoral trials where we had a bit of brown rust. And it's really important to use a combination of fungicide groups to reduce the risk of resistance development, not just in Septoria, but in other pathogens as well. Okay, onto the barley. 7 trial sites in 2024, 3 targeting Rhynchosporium, 2 targeting net blotch, 2 targeting Ramularia, and unfortunately we had a good spread of those 3 diseases across our sites. As with wheat, we test a whole range of products. Some of those are approved, and that's the ones I can talk about today, and that's in the table here. So again, the Proline and the Marisa that you'll be familiar with, and then a new product, which is ReviPro, which is a mixture of mefentrifluconazole and prothioconazole, so a mixture of Proline and Marisa. That's the first time we've presented any data for that, so it's brand new out. And then some other straits, the Comet, so the strobilurin, peroxystrobin, Imtrex, fluxaperoxide, which I mentioned on the wheat brown rust, and then the Miravis Plus, SDHI product. And then a couple of mixture products, Ascarex Pro, which you'll be very familiar with, been around for some years. And again, we have the Apresso in these trials. So that's the isofluricipram prothioconazole mix. As with wheat, we tested a further 4 unregistered products in 2024, and we'll be releasing data on those as soon as we can once they are registered. As with wheat, it's just as important to make sure you check your labels before use and to make sure that where you're using products, you use them in mixtures with at least one fungicide with an alternative mode of action to ensure that we have that has efficacy against the target disease, to ensure that we have robust control. So as with the brown rust, all the products now are on a single chart. The reason there's 2 charts on this particular slide is because the left-hand chart is protectant activity and the right-hand chart is mixed activity. It's a combination of protectant and radicant activity. So we had 2 trials on Rhynchosporium in 2024 where we got protectant data, 2 trials where we got mixed data. So we start on the left-hand chart with the protectant data. You can see we have a couple of mixture products, Apresso and the ReviPro. Remember that ReviPro is a mixture of 2 azoles. It's not a mixture of 2 different fungicide groups. So those mixture of 2 azoles can often mean that we get better disease control in terms of the spectrum of diseases, but it isn't as an effective anti-resistance management tool as, for example, two different fungicide groups. So bear that in mind. So we have those 2 mixtures and the 4 straits. The star performer, again, in terms of the protectant activity, was the Miravis Plus, closely followed by the ReviPro, and indeed the Apresso. So those 3 really giving us very good levels of protectant activity, and then good activity too from the Marisa, the Imtrex, and the Proline. So we've got quite a lot of choice there in terms of protectant control. In terms of the mixed activity, once again, the Apresso and the Miravis Plus giving very good levels of control, as did the ReviPro, and then the other 3 products giving good levels of control, but not quite as good as the others. And in particular, the Imtrex perhaps falling away a little bit in that mixed data set. Moving to the 3-year overview, so we have 5 trials over the last 3 years of protectant activity, 4 trials over the last 3 years of radicant activity on the right. Deal with the protectant first of all on the left, and again this time we've taken out the ReviPro because we only had it in 1 year of trials this year, and we've got Ascarex Pro now in this data set, so that's a mixture as well. So our mixtures are the Ascarex Pro and the Apresso, both of those giving very good protection, as of course did the Miravis Plus, and then similar levels of control from the Proline and the Imtrex. The Marisa over the 3 years, perhaps the least effective of those over 3 years, but still giving us useful control. On the radicant side, it's a very similar picture, except perhaps the Miravis Plus didn't stand out as much on the radicant data set as it did on the more protectant data set. If we look at the yields from the 6 trials over those 4 years for Rhynchosporium, again, the standout products really for yields, Miravis Plus and the Apresso, and then perhaps the weakest of the yield responses coming from the Marisa, which we kind of saw in the disease control data, the eradicant side, perhaps not quite up to the level of the other products, but still giving us some response. In terms of net blotch, I'm going to go straight to the 3-year overview data set. So 3 trials, protectant data on the left-hand side, 3 trials of eradicant data on the right-hand side. And looking at the curves, again, we've got a mixture of straight products and mixtures. The main mixtures being Espresso and Ascarex Pro. So the bottom line on the left-hand side is our Miravis Plus, the best net blotch control, closely followed by Marisa and our Ascarex Pro and our Espresso. So some good options there for net blotch control. And then good activity too from the Pro line and the Imtrex, but not to the level of the other products. On the eradicant side, again, similar story, possibly a little bit more separated the curves there, but the Marisa, the Miravis Plus and the Espresso, again, and the Ascarex Pro giving us good levels of control. The Imtrex, again, on the eradicant side, probably the weakest of the bunch. Our final set of data for winter barley, is Ramularia. So we have 3 trials from 2024 on Ramularia, just 4 products on this particular chart. So 3 straights, the Marisa, the Proline, the Miravis Plus, and then 1 mixture product, which is the Espresso. And you can see that the highest level of control we got was from the Miravis Plus, extremely good control of Ramularia in this data set. The Marisa and the Espresso both giving us better control than our standard, the Pro line, which was giving only a modest level of control in this instance. So we have a number of options that are giving us better control than the Pro line standard. The over-year data, which is 9 trials from the last 3 years, again, very similar to 2024 data set. Miravis Plus, the most effective of the products we've tested there. The Marisa and the Espresso, very, very similar to one another. Good levels of control, not quite as good as Miravis Plus, but better than the Pro line over that 9 trial data set. So, summary then on the barley. So the Miravis Plus and the straight, and then the mixture products, the Espresso, the Ascarex Pro, and the Revi Pro have given the highest levels of Rhynchosporium control in our data set, both individually and over the 3 years. But we've also had very good activity from the Imtrex, from the Marisa and the Proline. So there are quite a lot of options available to us on the barley for our Rhynchosporium control. In terms of net blotch, the Miravis Plus has shown the highest levels of control in our data set. And the second best really to that is the mixture products, so the Ascarex Pro and the Espresso, which are maintaining good levels of efficacy as well. So we have, again, a number of options for net blotch control. In terms of the Ramularia, the Marisa and the Espresso, more effective than the Pro line, but the Miravis Plus gave the highest level of control in both the single year and the over year data set. Finally, a reminder that just as with the wheat, and very important here because many of you will be thinking that the Miravis Plus is obviously a very broad spectrum product, giving us good control, but even there, mixtures always offer the broadest spectrum control and always offer the most robust control. So no matter how much any of the individual actives may look good, it's really important to use them in appropriate combinations in order to ensure robust disease control and minimise the risk of resistant development. And you see there's quite a lot of options there across those different diseases to get that combination. So that concludes the presentation on wheat and barley, and I can now hand over to Phil Walker of AHDS to cover the fungicide performance update for oilseed rape.
Thank you for that, Stuart. Yep, as mentioned, Philip Walker from AHDS, and I'm going to present the fungicide performance update for oilseed rape 2024. So the fungicide performance series in oilseed rape is a 4-trial series. So we have 2 trials looking at foamy stem canker, so that's Tarrington in Norfolk and Rosemont in Herefordshire. We've got some good stem canker index data and yield data from those trials for this season. We then got the Hymofort trial for light leaf spots, that's North Yorkshire, so that's hosted by AHDS. Fortunately for that site this year, we had a late light leaf spot epidemic and it occurred after the T2 timing. So the light leaf spot just came in too late to see any relative fungicide performance for light leaf spot control or yield reduction in relation to those spray timings. But at the Midlothian site, so that's near Edinburgh, hosted by SRUC, we had a good early light leaf spot epidemic. So we've got some leaf severity data to show from that site. Unfortunately, yield was compromised at that site due to some pigeon damage and overly wet soils at pre-harvest. So for the products which we're going to present results for today, so these are some of the main azoles and SGHI coforms which are available for use in oilseed rape. So there's Proline 275, prothioconazole, Aviator 235X Pro, so that's a bixafen prothioconazole coform, Plover, difenoconazole, and then Propulse, that's prothioconazole plus fluplyrand coform. There were a further 4 unregistered products which were tested in the trial series for 2024, but the data for these will not be released until they're registered for use on the market in oilseed rape. So we'll move on to the trial results. So this is going to be the results for Tarrington. So this is the foamy stem canker site in Norfolk. So it's variety Flamingo, so septal variety for foamy stem canker. We had a relatively late FOMO epidemic at that site, so we was getting first leaf spotting at the end of November with the T1 spray applied at threshold on the 6th of December, as well as a follow-on T2 sprayed at reinfection at the end of January. Even though it was a late epidemic, it was a relatively severe epidemic and a high instance of FOMO leaf spot, as well as quite a short crop, so we got some good high to moderate stem canker index and relative yield responses. So these are the results for that site. So we'll start with the disease index chart on the left. So just to give some context, so the scale is stem canker index. So this is an untreated stem canker index of 65. So that's 65% of plants dead from foamy stem canker. And then normally when we look at these charts, we're looking to see where the products give a stem canker index reduction to sort of close to 30 or below, because that's where we normally see an economic yield uplift. And as you can see from the products which are included in this trial, those products are giving that stem canker index reduction of close to 30 or below at that 50% dose rate, with only marginal improvement of increasing that dose rate above 50%. So the top line there, that's the Plover. We've then got the prothioconazole and the Procross giving a similar level of control. And the bottom line there, giving stem canker index reduction just below 20. That's the bixafen plus prothioconazole coform and the Aviator. When we look at the relative yield uplifts on the chart on the right, so we've got an untreated yield of 2.75 tonnes per hectare. So we're getting close to optimal yield uplift at that 50% dose rate, with only a marginal improvement increase in dose rate above that. However, I'll read the doses out at 75% dose rate because we get a little bit of product separation there. So the bottom line there is the difenoconazole, the Plover. That's given a 0.3 tonnes per hectare yield uplift. Propulse 0.35. The Proline is given a 0.45, and that top line there, that Aviator, is given a 0.5 tonnes per hectare yield uplift. When we look at the second site, so this is Rosemond in Herefordshire, so variety Aspire, so also septal variety to foamy stem canker. So we had an early FOMO epidemic at this site. It was occurring on the first and second leaves. So the T1 sprayed was applied on the middle of October, so the 16th of October at threshold, as well as a very quick reinfection period. So the T2 spray was applied on the 22nd of November. So as a result of that early leaf infection, we've got some good moderate stem canker indexes and some good relative yield responses. So when we look at the charts for the Rosemond site, so we start with the disease chart on the left. So we've got an untreated stem canker index of 47, so moderate stem canker index. And as you can see again, all those 4 products tested, they're given that stem canker index reduction of 30 or below at that 50% dose rate, with little or just marginal improvement of increasing that dose rate above 50%. So that top line there, so given that stem canker index reduction just below 30, that's the Plover, the difenoconazole. We've then got the next line down, given that stem canker index reduction just below 20, that's the Proline, so the prothioconazole standard. We've then got the Propulse, and then given that stem canker index reduction just below 10, the bixafen prothioconazole coform in the Aviator. When we look at the yield uplift chart on the right, so we've got an untreated yield of 3.95 tonnes per hectare. Again, 50% doses giving close to optimal yield uplift. For consistency, I'll read the yield responses at the 75% dose. So that bottom line there is Plover, that's given a 0.1 tonnes per hectare yield uplift. The Proline's given a 0.3 tonnes per hectare yield uplift, and those 2 SGHI coforms, so the Propulse and the Aviator, they're given a 0.4 tonnes per hectare yield uplift. When we take the average stem canker indexes and yield responses for those 2 sites together for 2024, we get a moderate to high stem canker index of 55, and then we see where those products are maximising out disease control at that 50% dose rate. So again, top line there, so getting that stem canker index reduction to 30, That's the difenoconazole in the plover. We've then got next line down below that is the profioconazole in the proline. And given that stem canker index reduction just below 20, we've got the propulse. And the bottom line there is the aviator, again, that bixfen coform, given that stem canker index reduction to 10. When we look at the average yield uplift from that site, so again, 50% dose giving very close to optimal yield uplift. I'll read these out at 75% dose for consistency. So that bottom line there, so that's the difenoconazole, that's in the plover, that's given a 0.2 tons per hectare yield uplift. We've then got the proline and the propulse giving a similar yield uplift of 0.4 tons per hectare. Then the top line there, that is the aviator, so the bixfen coform, that's given a 0.45 tons per hectare yield uplift average across those two sites. So we're going to have a look at the over years data set now. So we've got quite a good long-term data set now for foam stem canker results from trials going from 2015 to 2024. So this shows the average product performance across those 16 trials, taking into account high pressure and moderate pressure seasons. Just to note, Toledo, that was only included for 1 year in this data set. Propulse has been included for 2 years. All our products have been included for at least 3 years within that data set, and the ProLine, the profioconazole standard, has been included in all those 16 trials since 2015. So we start with the stem canker index disease control chart on the left. So we've got an untreated stem canker index of 46, so moderate sort of severity. Top line there is Toledo, so that's straight tebuconazole, so that has given a reduction in stem canker index, but it's very dose rate dependent in comparison to the other azole products which have been tested within the trials. When we look at all the other products, we're getting a stem canker index reduction of at least 25 or below at that 50% dose rate, and you can see those product performance is very much maxing out that 50% dose with very little or marginal improvement or increase in dose beyond that 50%. So that second to top line there, given that stem canker index reduction of just below 25, that's the plover, so difenoconazole. Then got two products below that performing quite similarly, the ProLine and Priory Gold. So Priory Gold being a difenoconazole azoxystrobin coform. We've then got Filan and Propulse performing similarly, Filan being straight boscalid. And then we've got 3 products below that, giving that stem canker index reduction of below 10 of the Aviator. We then got Architect, so that's a plaxostrobin-based chemistry. Then that orange line on the bottom, that is Shepherd, so that's a plaxostrobin plus boscalid coform. When we look at the average yield uplift from that site, so we've got an untreated yield of 3.56 tonnes per hectare. Again, we very much see those yield uplifts maxing out at that 50% dose rate, with very little benefit of increasing that dose rate beyond 50% to maximise yield. I'll read these yields out at 75% dose rate because we get a little bit of product separation there. So the bottom line there, that's Toledo, so tebuconazole, so that's given a 0.15 tonnes per hectare yield uplift. And then got the Plov and the Filan, they've given a 0.3 tonnes per hectare yield uplift. ProLine, Architect and Priory Gold, they've given a 0.4 tonnes per hectare yield uplift. Propulse, 0.45 tonnes per hectare yield uplift. And then there's 2 other SGHI coforms, so the Shepherd and the Aviator, they've given a 0.5 tonnes per hectare yield uplift on average across those 16 trial series. So we're going to move on to the light leaf spot data. So this is the Midlothian site hosted by SIUC near Edinburgh. So it's variety campus, so it's susceptible variety to light leaf spot. So we had a sort of early light leaf spot epidemic at this site. So there was detecting light leaf spot in incubated samples over October. So the T1 spray got applied at the end of October, on the 31st of October. Then that T2 spray is timed for when the light leaf spots first starts becoming visible within the crop, within the spring period, looking around that sort of pre-stem extension timing. So in this case, it was applied on the 10th of April 2024. So very much trying to replicate what would be done in a commercial situation, an autumn applied timing followed by a spring applied timing when light leaf spot first becomes visible. So we've got some good light leaf spot severity data. Unfortunately, as mentioned, yield was compromised at that site due to some pitcher damage and some overly wet soil conditions pre-harvest, so there was just some inconsistent yield results. So when we look at the light leaf spot disease data for this site, so we start with the chart on the left. So we've got an untreated leaf severity at just above 6%, so sort of moderate to high leaf severity. So this is leaf coverage of light leaf spot on the average across the whole of the leaf canopy. And this is on 100% of plants, so, you know, a high pressure situation, so 100% plants affected. moderate to high leaf severity infected. And as we see from those 3 prothioconazole-based products tested, Proline, Propulse, and Aviator, they've given a similar level of efficacy at that 75% dose rate. So it's becoming quite evident in these high-pressure situations that high dose rates, so sort of 75 to 100%, is required to achieve that minimum of that 40% efficacy for light leaf spot control, where we normally see that economic yield uplift from the application of the fungicides. When we look at the timing treatments in this trial, so the chart in the top right-hand corner, so for each year we include a timing treatment in a light leaf spot trial, so that's either half full rate Proline applied once in the autumn and then half full rate Proline applied once in the spring, and this is to see the relative contribution of those separate timings into the light leaf spot control. And as we can see for this season, both of those separate timings have given pretty much an equal contribution to the reduction in light leaf spots. So the efficacy that you see in the chart is the result of both of those spray timings, and this is important when you consider that autumn applied timing, which is normally over the latent period when light leaf spot might not necessarily be in the crop, but we still see where it might not necessarily be visible in the crop, but it's latent within the leaf tissue. We're applying a fungicide at that timing, we're seeing the benefit of control even though the light leaf spot might not be visible. As we didn't have any yield data for 2024, we're going to go back and look at the 2023 data where we did get some yield responses. So this is the High Mothorpe site, so that's North Yorkshire. So again, this was a high pressure disease site, so we had light leaf spot detected in incubated samples over the winter, as well as when at that spring spray timing, we had 100% light leaf spot visible in the crop when that spring applied timing was put on, so that was a highly curative situation, so it was high pressure and it was a very strong test of the products. So as a result of this being quite high pressure, we saw that all those products tested, they all gave a relative level of performance and efficacy against light leaf spot at that 100% dose rate. So again, high pressure, high dose rate required to see that level of control. So we've got Priory Gold in there, so the difenoconazole oxystrobin coform, Toledo, so straight tebuconazole, given the same level of performance at that high dose rate compared to those prothioconazole-based products also included within the trial. For the timing treatment for that season, for that 2023 trial, as mentioned, that spring applied timing was a highly curative situation, so a lot of visible light leaf spot. So we saw a greater contribution to light leaf spot control and yield uplift from that autumn applied timing during that latent period. However, we did see a small contribution to yield from that spring applied timing, so that likely protected that yield contribution from the autumn timing going into harvest. When we look at the yield uplift from that site, so we did get some product separation in this high pressure situation, so untreated yield around just above 2.7 tonnes per hectare. So if we look at those yield uplifts from that 75% dose, so we've got that Priory Gold and Toledo, they've given a 0.2 tonnes per hectare yield uplift. We've got the Proline and the Propulse, that's given a 0.3 tonnes per hectare yield uplift. And that top line there, that's the Aviator, so that Bixafen prothioconazole coform, giving a 0.4 tonnes per hectare yield uplift. With that Bixafen SGHI coform, we've seen that additional yield contribution in the absence of an improvement in visible light leaf spot control, and that's likely as a result of either a physiological boost from that SGHI coform or from some contribution to latent non-visible light leaf spot control, giving some benefit to increasing yield. So this trend of these SGHI coforms giving an increase in yield in the absence of visible light leaf spot control, we saw this also in some of the historic data sets. So this is light leaf spot disease data going from 2019 to 2021 across a 5-trial series. So we start with the disease control chart on the left. So again, sort of high moderate leaf severity, so just above 5%. And we're seeing that all those products tested within this trial are giving that 40 to 50% efficacy at that 75% dose rate. There's 2 products there on the top line giving a similar performance. That's the Flan and the Priory Gold, so Boscalid and that difenoconazole oxystrobin coform. Do note those products don't actually have a recommendation for light leaf spot on the label, so it's always worth checking labels for any product restrictions, but they are included here as they're likely or could be used at the similar times of year for the control of other diseases, namely foam and leaf spot. And so we're just seeing the relative secondary control of those fungicides used at these timings. There's 3 other products included there showing a similar level of efficacy, so the Proline, the Shepherd and the Aviator, they do have a specific recommendation for light leaf spot control. When we look at the yield uplifts, so if we go looking at that 50 to 75% dose, we've got those 3 products there, so the profluconazole, the Boscalid and the Priory Gold, giving a similar yield uplift of 0.3 tonnes per hectare. And then above that, we've got those 2 SGHO co-forms, so the Aviator, so the Bixen profluconazole, and the Shepherd, the Boscalid plus the Plactostrobin. They've given an additional yield uplift of 0.2 tonnes per hectare, so that's 0.5 tonnes per hectare over untreated. So again, we were seeing that co-form SGHI included within another mode of action. We're seeing that additional benefit of increasing yield in the absence of visible light leaf spot control, either through a physiological boost from those fungicides or through some latent light leaf spot control. So we're going to go back and look at some of the historic data for the Sclerotinia trial series. So this is trial series from 2015 to 2017, so across 5 trials. So if we look at untreated control, so we got a moderate Sclerotinia index of around 18. And when we're looking at these charts, when we consider this is a single application timing at mid-flowering, where you're looking to get that maximum level of fungicide persistence, so you want about that 3 weeks worth of protection from that single application. You're looking at the high dose rate, so that's 75 to 100% dose rate, where you're going to get that high level of fungicide persistence only for using the high dose rates. And as we can see, all those azoles, SGHIs and SGHI co-formulations at those 75 to 100% dose rate, they're all given a very high level of efficacy, all performing very similarly. 2 products there that's given a slightly lower level of efficacy, but still very highly effective against Sclerotinia. So that line just below that is the Amistar, so that's straight azoxystrobin. And then we've got the Amistar difluconazole azoxystrobin co-form in the Priory Gold, also giving a good level of efficacy, but just a little bit lower. And reasoning for that is because the Priory Gold has half the amount of loading azoxystrobin in it as what you find in the Amistar. And then those relative levels of efficacies is reflected in the yield responses, so that single apply timing at that mid-flowering period. So for 75% dose, so we've got the Priory Gold giving a 0.2 tonnes per hectare yield uplift. We've got the Amistar giving a 0.3 tonnes per hectare yield uplift, and all those other products giving a similar 0.4 tonnes per hectare yield uplift. So in conclusions, we've got effective azole, non-azole options for foam and stem canker control. Yield responses range from 0.2 to 0.55 tonnes per hectare in 2024, and that's on separate varieties, part of this 2-spray program. Like leaf spot, we've found we've got azoles, non-azoles, co-formulations which are effective, providing similar level of control of disease and yield uplift, though we do get some regional variation like leaf spot control, and it's worth just checking product restrictions for any products which may not have recommendation on the label, as well as considering your time of application. And then Sclerotinia stem rot, we've got a range of modes of action available, so that just helps you consider where different modes of actions can be used in the program compared to where different modes of actions have been used during your foliar blade primings, so where you can alternate those modes of actions just to help prevent the build-up of resistance to fungicides. So I just want to finalise and acknowledge the project partners for fungicide performance, as well as the field teams and trials teams for those relative organisations who put a lot of work and contribution in producing results for these trials which are presented today. So that concludes the Fungicide Performance Series 2024. Thank you for listening.
Brilliant. Thank you so much, Stuart and Phil. That is a huge amount of information and a huge amount of data to be presenting, so thank you for doing it so eloquently. And just a little reminder that Poll EV is still open, so if you do have any questions for Stuart and Phil so far and for any of the other speakers in this session, please do submit them to Poll EV to be asked to the panel a little bit later on. So coming up next, we've actually got a familiar face. So Sacha White is joining us again, and Sacha is going to be talking to us about insecticide resistance guidance. So handing back over to you, Sacha.
Thanks, Henny. Morning again, everyone. So I'm going to be presenting some AHDB funded work looking at re-evaluating insecticide resistance management. So it's clear that insecticide resistance poses a serious challenge to our ability to sustain high yields, and there are several resistance management strategies that we can use. The probably most widely used are things like rotations and mode of action. So your first spray might be mode of action X, second spray might be mode of action Z, and you continue to cycle like that. Alternatively, you can use mixtures, so 2 different modes of action mixed together in a spray, applied at the same time, and you spray that mixture at each spray occasion. Or you can reduce your dose, so you might just use mode of action X, but you're reducing the dose, so you still get effective control, but it's a resistance management strategy. And you can mix and match these actually in a program. Now, IRAC and IRAG UK recommend that the optimal approach for insecticide resistance is to use rotations of mode of action at their full label rate. And I should say IRAC is an international organisation that provides guidance on resistance management. The membership is AgChem companies. IRAG UK is a UK version of that, essentially. It focuses on UK issues, and it has a wider membership, so it includes AHDB government and independent research organisations. Now, it's fair to say that there remains a lot of uncertainty over which is actually the most effective resistance management strategy. So to try and answer this question, AHDB and DEFRA funded some modelling work. This was about 10 years ago, and that was work that was carried out by Rothamsted and ADAS. And that modelling work compared different resistance management strategies for their ability to slow the build-up of target site resistance. And the general conclusion to that work was that reduced dose mixtures most often resulted in the longest effective life of insecticides, i.e. they slowed the build-up of resistance more than the other resistance management strategies. And I should say that a reduced dose mixture is a mixture of two modes of action, but this time you're reducing the dose of those modes of action, so you're still getting effective control. Now, there were some exceptions to this general finding. That included where the insecticide efficacy was very high, where the pest was haplodiploid, and also the reproductive strategy could affect the rank order of the resistance management strategies. But nevertheless, this overall finding that reduced dose mixtures was most often the most effective approach contradicted with the current guidance. But before it could be integrated into current guidance, it was felt that more empirical evidence was needed to test the model. And that's where the work that I'm about to explain comes in. So 4 years ago, AHDB, BBRO and Corteva funded ADAS and Rothamsted to carry out experiments to test the model. These were randomised replicated cage experiments, and they involved peach potato aphid. There were 4 clones of peach potato aphid used, and between them these clones had all combinations of 2 resistance mechanisms. There was a super KDR that confers high levels of resistance to pyrethroids, and mace that confers high levels of resistance to carbamates. And all of the clones, peach potato aphid, I should say, is asexual in this country, and they were asexual in these experiments, so there was no sexual recombination happening. So those clones were 800F that had neither resistance mechanism, clone D that had super KDR, 5191A that had mace resistance, and then clone A that had super KDR and mace resistance, so it would be highly resistant to both carbamates and pyrethroids. So the experiments involved treatments that were essentially insecticide resistance management strategies, sometimes with different levels of efficacy. Each cage in the experiment had the same aphid population at the start and the same resistance frequency. Every cage was sprayed at the same time, approximately weekly, whenever there were enough aphids to justify doing that. And then also there was a weekly immigration of aphids arriving into the cage. So that was just to simulate a sort of a real life scenario, but also to ensure that none of the clones died out during the experiments. After each spray, a few days later, each cage was counted, the aphids were counted to assess the population, and also a representative sample of aphids were taken from each cage, so about 100 aphids from each cage. These samples were then tested using a novel qPCR assay that was developed as part of the project. What this assay allowed us to do was determine the change in the resistant frequency of each resistance mechanism in each cage throughout each experiment, and therefore compare the resistance management strategies. And the cages were kept in an environment-controlled glasshouse at ADAS Boxworth, and here you can see one of the sprays going on to the aphids in one of the cages. So experiment 1 had 3 treatments and 9 reps of each treatment. Treatment 1 was a high dose rotation, so a carbamate was applied first, then a pyrethroid, and then a carbamate, and so on and so on. And you can see in the tables the parts per million of each of the insecticide used and the percentage of the field rate. And you can see the percentage of the field rate is much lower than we would use in the field, and that was because we needed to reduce the dose so that we got the appropriate mortality for these cage conditions. The second treatment, or I should say that the carbamates used for all of these experiments was pyramecarb, and the pyrethroid used was lambda-cyhalothrin. The second treatment was a high-dose mixture, so a carbamate and pyrethroid mixed together at the same doses as were used in treatment 1. And treatment 3 was the moderate dose mixture. So again, the mixture of carbamate and pyrethroid together, but this time the dose was reduced to achieve about the same level of mortality as was achieved in treatment 1. You can see the starting ratios of the cages on the right there. So every cage had 60 aphids. 54 of the aphids were the fully susceptible clone, and the remaining 6 aphids were split evenly between the 3 resistant clones. So what did we find? Well, firstly, looking at the aphid numbers in this experiment over time. Here we've got the mean aphid populations per cage over the course of the experiment. This experiment went on for about 80 days. And we had 6 sprays being applied, and you can see the sprays are indicated by the black arrows on that graph there, and the different coloured lines show the different treatments. So what we can see is after the first spray, we saw a reduction in the aphid populations. That's to be expected. The populations are largely susceptible at this stage. But from that point onwards, from the second spray onwards, we've lost control of the aphid populations. They just continue to rise. And eventually we reached, well, the aphid populations plateau. Essentially, they reached the carrying capacity of the cages. So each cage could sustain about 4,000 aphids. Now, there were very few differences between the treatments in terms of the aphid populations, and indeed none of these were significant. But more importantly, what do the treatments do for the selection for resistance? Well, looking at super KDR resistance first, here we've got the percentage of individuals that had that resistance at each time point. What we can see is after the first spray, we're selecting very strongly for resistance. By the time we get to the fourth spray, almost all the treatments have 100% resistance in them. We can see some slight differences between the treatments. So the high dose mixture appears to be selecting for resistance most rapidly, and the moderate dose mixture most slowly, but these differences weren't significant. When we look at the same, but for mace resistance, again, that first spray is selecting strongly for resistance, maybe not as strongly as for super KDR, but again, by the time we get to the fourth spray, we've selected for almost complete resistance in all of the treatments. Again, there's a slight trend for the moderate dose mixture to select less rapidly for resistance than the other treatments, but these differences weren't significant. You'll also see at the end, for those 5th and 6th sprays, we see a reduction in resistance. And that might seem surprising, but what we think is happening here is because the cages have reached carrying capacity, that selection is happening for reasons other than the insecticide. So probably due to the relative ability of these clones to compete in a resource-scarce environment. Going on to experiment 2, this time we had 6 treatments, 6 reps of each treatment this time. So first 2 treatments are high-dose rotations. They just differ in terms of whether the carbamate is applied first or the prethroid. The second treatment is a high-dose mixture, so again, the prethroid and carbamate together at the same doses as used in treatment 1 and 2. Treatment 4 is a moderate dose rotation where a carbamate is applied first. This time the doses are half those used in treatment 1. Treatment 5 is a moderate dose mixture where the doses are half those used in treatment 3. And treatment 6 is a low dose mixture where the doses are half those used in treatment 5. The starting populations and resistance frequencies are exactly the same as were used in experiment 1. So what do we see in terms of aphid numbers in this experiment? Well, this time I've split the data across 2 graphs just to make it easier to interpret. We've got the high dose treatments on the left and the moderate and low dose treatments on the right. So looking at the high dose treatments first, that first spray strongly controls the population. Again, these populations are largely susceptible. These are high dose treatments. But from the second spray onwards, we've just lost control of the population, and it rapidly reaches that carrying capacity of 4,000 aphids. On the right-hand side, the moderate and low dose treatments, that first spray again is controlling the population, moderately slow this time because these are moderate and low doses. And the build-up of the aphids do still get to the carrying capacity, they just take a little bit longer to get there. Now, overall, there were no significant differences in the change in aphid populations over time between these treatments. Again, more importantly, what do these treatments do for super KDR selection? Well, again, the first spray selects looking at the high-dose treatments on the left first, that first spray selects very strongly for resistance. By the time you get to the third spray, you've got to almost 100% resistance in all of those treatments. Looking at the graph on the right next for the moderate and low-dose treatments, that first spray is also selecting strongly for resistance, perhaps not as strongly as for the high-dose treatments. But by the time we get to the 4th or 5th spray, we've got to pretty much 100% resistance in all of the cages. There is a trend for the low-dose and moderate-dose rotation treatment, the low-dose mixture and moderate-dose rotation treatments to select less rapidly for resistance than the other treatments, but these differences weren't significant. Now, for the selection for mace resistance, this time the high dose treatments and the moderate and low dose treatments appear to be doing something pretty similar. The first spray is selecting very strongly for resistance, and we're getting to almost complete resistance by the third, fourth, fifth spray. The biggest difference really here is with the high dose rotation treatment, where the pyrethroid was applied first. So here, that's on the left-hand graph, that green line. After the first spray, we're not selecting very strongly for resistance. And the reason for that is because this is a pyrethroid spray, and we wouldn't be expecting it to select strongly for mace resistance. By the time the carbamate does get sprayed at that second spray, we've pretty much caught up with the other treatments. But overall, there were no significant differences between these treatments in this experiment. So looking at our final experiment, this had 6 treatments with 6 reps. The first 2 treatments were solo carbamate and solo pyrethroid treatments. So essentially, these are treatments where, for instance, for treatment 2, a pyrethroid was applied again and again and again. So these treatments were included to give us an example of what is really a bad resistance management strategy, and is actually often what happens really for pest control in the field. Certainly for cereals and oilseeds, pyrethroids tend to be the insecticide that's used again and again. The third and fourth treatments are high dose rotations, just different in terms of whether the carbamate or pyrethroid is applied first. They are being applied at the doses used in treatments 1 and 2. Treatment 5 is the high dose mixture at the same doses as used for treatments 1 to 4. And treatment 6 is a moderate dose mixture where the doses are half those used in treatment 5. The other big difference in this experiment was that we dropped the clone A, which was the double resistor, the clone that had both resistance mechanisms. This was done because we felt that this might be why resistance was being selected for most rapidly in experiments 1 and 2, and why we might have been more difficult to tease apart the differences between the resistance management strategies. This time we've still got 60 aphids, 56 of them are the fully susceptible clone, 2 are the carbamate resistant clone, and 2 are the pyrethroid resistant clone. So when we look at the aphid numbers, there's a lot going on in this graph, so I'll just drill down into the salient bits. The first thing to say is that we retained a level of control of these populations throughout the experiment, so none of the cages, none of the treatments reached that carrying capacity. That meant that the experiment could go on for longer, 4 months this time, and we had more sprays going on as well, so 10 sprays throughout the course of the experiment. Overall, all of the treatments are doing something pretty similar. They're all providing a relatively good level of control of the aphid populations. There is some variability over time, but generally they're doing something similar. The big difference is that red line at the top. So that's where the solo pyrethroid treatment, where the pyrethroid is applied again and again. What was found here was actually this provided very little control throughout the experiment, and that was surprising. It wasn't expected to do that. The batch of insecticide was tested. It was tested against culture aphids, and those experiments showed that it was providing good control. So this was an anomalous result with this solo pyrethroid treatment. Now, going on to look at the selection for resistance. Now, because we don't have that double resistor in here, I can present the data slightly differently. So what we have is bar charts for each treatment. So treatments 1 to 3 across the top, and treatments 4 to 6 along the bottom. Each column is a different time point. So we've got the start, we've got the S1s and S2s, etc. They mean spray 1, 2, 3. So start, pre-spray 1, post-spray 1, so on and so on. The blue sections of the bars show the percentage of the population which is the susceptible clone. The red bit is the pyrethroid resistant clone, and the green sections are the carbamate resistant clone. So if we look at the solo carbamate treatment first, we're selecting moderately quickly for carbamate resistance, that mace resistance. So by the time we get to the 6th, 7th spray, we've got about 70-80% resistance. Interestingly, we were also selecting for pyrethroid resistance here. That wasn't expected, but what this probably reflects is the relative susceptibilities of the susceptible clone and pyrethroid resistant clone to carbamates, and also maybe some slight differences in their basic life history traits. So one of them might be a slightly higher reproductive rate than the other. The treatment 2, solo pyrethroid treatment, we were expecting to see selection for pyrethroid resistance. We saw a small amount, but much less than expected. And this is probably because, as I mentioned in the previous slide, this treatment didn't provide very good control at all. So where you get poor control, you're probably not selecting for resistance very much at all. But what we do see here is the carmate resistance being well controlled and removed from the population. And actually that applies for treatments 3 to 6 as well. All of these resistance management strategies are working well for managing the carmate resistance. Treatments 3 and 4 are the high dose rotations, just differing in terms of which insecticide goes on first. And they're both doing very similar things. They're both selecting moderately quickly for pre-throid resistance. Treatment 5 is the high dose mixture, and here we see the most rapid selection for pre-throid resistance. And treatment 6 in the bottom right, this is the moderate dose mixture, and here we can see the slowest selection for pre-throid resistance. And in fact, here we saw significant reductions in the rate of pre-throid selection in this experiment. So just to conclude, well, the findings certainly aren't clear-cut, but certainly experiments 1 and 2, whilst we didn't see significant differences, there was a trend for reduced dose mixtures to slow selection. In experiment 3, we did see the reduced dose mixture significantly slowing the selection. Overall, this does provide some support for the model findings and suggests that reduced dose mixtures are at least as an effective resistance management strategy. The IRAC positional mixtures has softened slightly. They now see that it does have benefits for resistance management, but they still say that those insecticide that go into the mixtures should be applied at their label rate, which obviously differs from what we're finding here, both in the experiments and the modelling work. Now, the findings of this work and the modelling work are yet to be included into the IRAG guidance. So for the moment, I think you should still be following the IRAG guidance. That IRAG guidance can be found on the AHDB page. We host IRAG, so if you just Google AHDB and IRAG, you'll find it. And there's also lots of useful, digestible guides on there as well. The other thing to say, from this work, we found that where you have a pest which has resistance to both mechanisms, mode of action mechanisms that you're using, then you have a serious issue for resistance management, because you're going to select for resistance no matter what you do quite rapidly. And that's well illustrated by the differences that we saw between experiments 1 and 2, on one hand, where we saw rapid selection for resistance, where we had the double resistor in the population, and experiment 3, where that double resistor wasn't included in the experiment, and the selection for resistance was much slower. What is really critical is we put resistance management strategy in place as soon as we can, because by doing that, we avoid the appearance of double resistors for as long as possible. And the sooner we do these things, the longer we're going to have insecticides being effective for. The last thing to say is IPM is a really effective resistance management strategy, because the less you have to spray, because of all the other things you're doing, the less you will be selecting for resistance. And the final thing to say is that all of this has been about target site resistance. And there is a good possibility that guidance may differ for metabolic resistance, so we may need to do similar work for this type of resistance. So thank you for listening. Thank you to ADAS and Rothamsted for carrying out these very difficult experiments. Thank you to Bieber and Corteva for co-funding the work, and all these people for their involvement.
Brilliant. Thank you very much, Sacha. Just like the fungicide performance, a huge amount of data, a huge amount of information, and really well conveyed. So thank you for that, Sacha. And do make sure that you look up the IRAG information on the AHDB website. So up next, we have another double act. So we have Katy Hebditch and Lynn Tatnell from ADAS, and they are going to be talking today about herbicide resistance. So yeah, I'm going to hand over to them.
Brilliant, thank you. So the last AAB paper on the current status of herbicide resistance in the UK is now about 10 years old. So I'm here today to talk a little bit about where we are in terms of resistance currently in the UK, what we see as the future risks, and also present some research that's being done by different organisations in this area. So I think unsurprisingly, the resistance landscape continues to be dominated by the arable annual grass weeds, so particularly our blackgrass and our Italian ryegrass on the top 2 photos there. So in addition to seeing an increasing number of samples becoming resistant to our Group 1 and Group 2 post-emergence herbicides, we're now also seeing widespread resistance to our pre-emergence herbicides. So that's things like pendimethalin, flufenicet, and prosulfocarb. And that's likely linked to that shift to our kind of autumn programs of pre-emergence herbicides, which we've had to force or sort of be forced into because of the post-emergence resistance. So back in 2014, it was estimated that resistance occurred on greater than 20,000 farms in 37 counties. And we're now beginning to see ryegrass follow a similar pattern to that. So we're seeing samples come in for testing from sort of western counties like Shropshire, Herefordshire, and also down from sort of Yorkshire as well. So we're seeing the spread of ryegrass resistance in a similar fashion to what we've seen with the blackgrass. In terms of our wild oats and our bromes, we're not quite seeing that large-scale pre-emergence resistance problem that we've seen with the other two, but we are seeing an increasing number of samples come in, again for ALS testing mainly, but also ACCAs, so things like phenoxyprop and pinoxidin in our wild oats. So there's not yet been a published case of glyphosate resistance in the UK, but it is a real risk, and that's something that Lynn will cover a little bit later. But we do have reduced sensitivity in a couple of populations, so that's an Italian ryegrass and a sterile brome population, and it's something we continue to monitor very closely. Brilliant. So quite a lot of people are working on resistance at the moment, so I thought I'd present a couple of results from recent years. So this is from a paper led by Sarah Cook at ADAS that collated blackgrass resistance testing results from the past 35 years to essentially look at trends for particular actives. So just quickly to explain this graph a bit, we present resistance results as these R rating. So you can see in green there we have sensitive samples, but then once you're getting up to RR and RR, this is essentially resistance is confirmed in that sample and herbicide performance is highly likely to be impacted. So what we can see, so this was a graph for mesosulfuron and iodosulfuron, so an Atlantis type product. And you can see in 2001 and 2, when testing began, actually 100% of those samples were still sensitive. By the time we're reaching sort of 2011-12, we're seeing 63% of those samples now rated as RR or RR. And actually by 2019-2020, so that's when this sort of paper's collate data up to, we're now seeing 90% of these samples being RR or RR. And actually, if you turn that into kind of a regression, that's a decline in herbicide efficacy of this by 3.7% a year. So really just seeing that decline in control quite quickly. But then we also did the same thing for pendimethalin. And actually what you can see is that number of RR or RR actually seems to fluctuate a lot more. So there is sort of a general increase over time, which corresponds to a 25% decline in control over the testing period. So that began in 97 and again ended in 2020. But that is still sort of a decline of about 1.1% a year. I think what's really nice about this paper is it kind of backs up what we've been saying, is that those post-emergence groups are the highest risk, and we will see resistance develop a lot more quickly, but it's important not to ignore these pre-em groups. And another nice thing they did with this paper is actually correlate it to PUSTATS data, so pesticide usage. And what you could see is with the post-em, so that ALS graph on the back one, we saw usage decline as resistance increased. However, with the pendimethalin graph, actually we saw that usage is still very high, so it's just important to consider that resistance risk. And another thing they did as well was able to look at the geographic spread of the data that was coming in. So we can see that in the 1980s, in that dark green colour there, resistance to at least one herbicide was restricted to just a few counties, mainly in the southeast. But actually, by the time we got to the 1990s, resistance is now confirmed in much of central and southern England. And again, by the 2010s, we're seeing that increasing spread northward and increasing spread westward. So it's sort of really nice that we can use that data to show that geographic spread over time. So moving on, so this was a study that was led by NIAB. So they screened 197 brome samples for ALS resistance, and that was sort of the 5 main brome species in the UK. And actually what's really interesting from this is that we seem to have quite a high prevalence of ALS resistance in the rye and meadow bromes, so highlighted there as 28% and 25%, whereas actually previously, before, we thought sterile brome might have been a bigger problem. So that's sort of really interesting results. And you can see sort of on the picture as well that all these samples were also screened against glyphosate. There were no signs of resistance in any of these. So again, they mapped where these cases came from, and actually what's really interesting is that we observe these very distinct clusters of resistance. So this might actually point to a more sort of biosecurity issue and that possible spread of resistant seeds across fields and across farms, and also highlight how important integrated weed management is as well. So again, sort of another really nice example of how we could use that geographic data for resistance. And then finally, so Rothamsted are coming to the end of their Blackgrass Resistance Initiative programme. So this is where they've monitored set fields yearly to map blackgrass levels, and they now have 10 years of data from this. So their plan is to retest for resistance of all the initial samples, and that's for both target site and non-target site resistance, to see essentially how much those populations have shifted over the course of the project from 10 years ago to now. And as they've mapped those same fields, they also have lots of field data, so they might be able to sort of correlate this to potential drivers of resistance, or perhaps see what practices have kind of worked to prevent that resistance from coming on. So they're now moving away from focusing kind of on blackgrass alone towards broader weed issues with the One Crop Health project, and I've just included a link there in case anyone's interested to look at that. And then moving on to our broadleaf weeds. So generally testing's been a lot less extensive than for the grasses. So back in 2014, it was reported that we had ALS resistance in poppy, chickweed and mayweed. That's in those first 3 photos there. And since then, we also have had a case of ALS resistance in prickly sow thistle. So again, sort of testing is quite sort of not as extensive as the grasses. So at ADAS, we'd say test about 7 to 8 samples a year compared to the 50 plus we're getting in for sort of blackgrass and ryegrass. But interest does seem to be increasing. So we've got 15 plus samples this year. And again, all of these requests are to ALS herbicides. And I've just put in there as well about sort of the group 5 triazonones. So there was quite a historic case of groundsel resistance, but this doesn't seem to sort of recurred again, and perhaps not something we're too worried about compared to the ALS herbicides. And this is just sort of a little picture just to show how complex it can be. So both fluazulam and metsulfuron are ALS herbicides. So you kind of think that if it's resistant to one, it would be resistant to both. But actually, in the case of this sample, we see extremely high sensitivity still to fluazulam, but actually quite high resistance to metsulfuron. So even within those mode of action groups, you might see sort of a range of resistance depending on the actual sort of chemical structure of those herbicides. So again, just kind of highlighting that importance of testing. And in terms of broadleaf weed resistance and what kind of we think are the future threats, so again, it says ALS herbicides, they do remain the highest risk because they're very widely used across the rotation. So this year as well, we are having requests to test a greater variety of species, so things like groundsel, cranesbill, sow thistle as well. We have seen alternative modes of actions come in, so like the phenoxy and haloxafen herbicides, and these have really helped support the herbicide portfolio. But we also do need to make sure that we're not putting too much pressure on these herbicides. We've also seen the introduction of several herbicide-tolerant crop varieties. And again, sort of these have been great for weed control, but also it's very important to consider the resistance risk of kind of repeated use of similar herbicides, and also really important to follow label advice in these situations. And again, sort of our main message is just sort of consider that ALS use across the rotation and kind of minimize where possible. So a quick summary of kind of resistance. So resistance testing is really important as an early warning. So one thing to consider is that it will overstate the degree of resistance at the field scale. Because you're taking seeds only from survivors, you might say get a triple R result, and actually in the field, you're not quite seeing that level. But we think it's really important as kind of an early warning so that something can then be done to minimize the risk going forwards. So again, so our arable grass weeds, particularly our blackgrass and ryegrass, do continue to pose the greatest resistant threat. Italian ryegrass in particular emerging as a significant concern, as what was once quite a southeast centralised problem does now seem to be spreading. And again, so our resistance in broadleaf weeds has been reported less frequently, but we are now seeing sort of increasing attention in terms of species requests for testing, and again, really looking into the importance of ALS in that. So that's quite a quick summary of sort of the resistance status in the UK. And now I'll pass on to my colleague, Lynn Tatnell, who will cover a bit more about management systems. Thank you.
Good afternoon. So my name's Lynn Tatnell. I'm a weed biologist at ADAS. So I'm going to cover production system changes and with a particular emphasis on weed management as part of that. So we've seen a lot of changes certainly over the last decade, but within the last 5 years there's been a lot that's really changed. So from a weed perspective, we're going to explore some of that. So what is changing and why? So I've tried to put these into different categories. So this is certainly not an exhaustive list. There's lots of other things, but I've tried to make these as the kind of key points and things that came to my mind while I was considering this topic. So starting with farming systems, regenerative agriculture is obviously one of the biggest things now that is very popular and extremely high on people's agenda. So we're going to touch on that a bit in a moment. Things like SFI payments, they're perhaps making a big difference now to how people are considering what they might do across their rotations within their cropping systems. So again, from a weed point of view, they will actually be making quite a change going forward. So that may include things like taking fields out of production and putting them in a cover crop or a living mulch. So again, all having an impact on the soil seed bank and the weed seed bank. Also now is an increase in cover crop use and living mulch use. So again, this brings a whole different species diversity into the system. So these may be species that we're not used to in our arable crop rotation. So again, they could be fantastic from a weed management point of view or bring challenges. So we'll talk about those in a moment. And we see a big change in decreased tillage. So perhaps the use of the plough is now becoming less popular and less in favour. So now that will bring a big change in our systems. And with that, we have wider rotations and things like agroforestry. That's fairly new in the farming system area, but now, again, quite widely adopted. If we think about the pesticide change category, so we've just heard from Katy the increasing risk in resistance, and then now grass weeds in particular are a big challenge still, so they must be taken seriously. And we do have over-reliance on the few actives we have. So we don't have a lot of new actives coming through, and this is the same across weeds, pests and diseases, but we are then slightly over-reliant on the actives we have. So with this pesticide loss, this is something we really, really need to think about. And finally, but last but no means least, the wider context of the environmental issues. So we're thinking about carbon concerns. So are we looking, if we're using alternatives to some of our herbicides, is that putting more carbon in the atmosphere? Have they got a bigger carbon footprint? Is actually a herbicide a lower carbon footprint? So these are all things we really need to think about in the future going forward. Soil health is obviously very high on the agenda. So again, with the regen ag movement, that is very much driven by soil health and people thinking about their soils. And an overall reduction in pesticides. So again, your IPM strategy is obviously a great way forward, but we really do need to think about how that is impacting on our weed management and weed communities. So I'm going to start a little bit touching on regen ag and think about the principles of that. So these are probably very, very familiar to people. So the 5 principles of keeping the soil covered, maintaining living roots, minimal disturbance, integrating livestock, and maximising your diversity. So these are all things that can give us kind of positive and negative sometimes impacts on our weed communities. So if we think about things like covering the soil, maintaining your living roots and integrating livestock, they could all be fantastic for weed suppression, for that competitive effect of weeds in the seed bank. So these are things that could really, really benefit weed management going forward. But minimising soil disturbance, again, not having the plough in your system could be very dependent on your weeds, your seed bank, and knowing what is within your seed bank. So again, if you have perennial weeds in your system, this could be a benefit if you're not chopping up those rhizomes underground and spreading that problem. So it should be a positive, and we need to just consider how we use it and how our weed seed bank is and what we're doing to make that beneficial for us. So there are now a lot of strong voices of change, and this is quite exciting. It's really exciting times, it's really different. So things like the Groundswell event, so this is now the Groundswell Festival, many people will have attended this. It's now extremely well supported, and these are just a few of the associations I've picked out here to give people an idea of who is kind of talking and singing from the same hymn sheet. So Nature Friendly Farming Network, you'll see posting a lot online, ORC, CRT, Leaf, so these are all big movements and people that are coming together and saying the same messages. So, and then there's important brands like Wild Farmed. This is something that's reaching the public a bit more widely than our rest of our industry. So it's important that we do share these messages really widely out there with public groups and use these platforms as well as we can. So thinking about weed management particularly then within this challenges within the regen ag. So as I said, we think, within this, mainly we're looking to reduce the use of plough or remove it altogether with the trend towards minimum tillage. So the main consideration is thinking about your soil health and your quality. So that's obviously all based around getting your productive, healthy crop. So with this reduction in soil tillage, we do really, really need to think about where your weeds are in your seed bank. So if you have a very high grassweed burden and you have seeds that unfortunately were shed into that crop that year, then maybe you should be thinking about a very decent stale seed bed or a different cropping regime altogether, or going into a spring crop that year, because you know you're putting a lot of seeds back into the seed bank that year. So that transition phase of thinking about how your system might go into the more sustainable, perhaps regen ag situation, knowing where your weeds are, and particularly the grass weed burden must be really high on your agenda. But really, what is your starting point? And that's going to be different for everybody. So there is no set rules. You need to know your land, your weeds, your situation, and then you can plan your strategy and what would work for you. So I think for this, each field could be different on your farm. So it's really knowing where your weeds are and what you're planning to do across a rotation that's really important. But quite often in these systems, we are more reliant on glyphosate. So this is something we really need to think about going forward. So without the plough in the system, we are seeing an increase in glyphosate use. So that may be for a number of different reasons. It could be to control or terminate cover crops that we've had in a break crop, to use as that stale seed bed before we drill a cereal, or for perennial weed control. So it's all different uses of glyphosate. Mechanical weeding is often seen as one of the tools, as an option, as an alternative to glyphosate. But if you consider that against glyphosate, you're suddenly already moving the soil more. There's more soil disturbance, there's perhaps more compaction. So this is all a balance and weighing up the pros and cons, and again, knowing your land and your weeds. So striking this balance is really important. So I just want to touch a little bit more on glyphosate. So we can see from this graph, this is taken from the PustStats data from 2022. From the last 30 years, we have seen a very high increase in glyphosate use in UK cereals. So you do have to look at these data with a bit of a context here. So this is data that could have been used pre-drilling of the crop or pre-harvest of the crop. So it doesn't specify in which phase of the crop these are being used. But this is, obviously, you can see a big increase over time. So we had a bit of a plateau during the naughty years. There was not much of a change, but now we are seeing a big increase. However, I don't necessarily think this is a bad thing. This could be an increase because we're actually getting the messages out there of having that really effective stale seed bed before you go into your crop. So we're perhaps then not putting the pressure of the weed control within our cropping cycle and putting too much emphasis on the few actives we've got left in that part of the cycle and actually getting it right in our stale seed beds. So that's something to think about, and this isn't necessarily a negative thing that the rate of use has gone up. So obviously I haven't got a lot of time to talk about this today, but I would like to highlight two quite important papers, and if this is something that's of interest to you, there's a current paper that's just been released this year from Neve et al, and this is the current and future glyphosate use in European agriculture. So this paper is freely available from Weed Research. And there was a survey done, it's the Endure survey that some people may know of this, so Antia et al. But again, if this is of interest to you, I would highlight both of these references are really important and really worth a read. So the Weed Resistant Action Group, RAG, produced these guidelines nearly 10 years ago now, and these were minimising the risk of glyphosate resistance. So you've just heard from Katy that we've obviously got quite a shift in resistance in the UK in other modes of action, but this was set about as an industry initiative to be proactive to try and minimise the risk of glyphosate resistance coming into the UK, because we do know now across Europe we do have a few populations of grassweeds that are resistant in arable cropping systems, and we don't want that to be the case here. So this was very much an industry-led collaboration, and the guidelines were put together using data that were through research projects funded by AHDB and industry, and we quantified a lot of the detail that are in these guidelines. So again, you can find the guidelines online on the AHDB website, but these are the 4 key principles. So we really must thinking about preventing survivors. So if you've got a few survivors that have had exposure to glyphosate at perhaps a low rate or a rate that has not killed them, we do not want those to go on to produce seed and put those seed in the seed bank. So preventing survivors is always key. Maximising efficacy of the glyphosate product you're using, so think about the dose and the timing and the size of the weed. It's really important to get the right dose for the right size weed. think of alternatives. So whatever alternatives you may like to use or have as an option on your farm. So that could be a cultural control technique. So maybe within a stale seed bed, you could have a glyphosate application and a cultural control option within that. So please do bring in those alternatives as an IPM approach. And then monitoring success. So again, pay attention, go back, to have a look at how successful it was, and really do prevent those survivors coming through. So really going forward, I think it has to be about diversity. So we've got to be able to adapt to these situations and not put all our eggs in one basket and rely on one thing. So I think it's really, really key that we do reduce the reliance on glyphosate where we can. So use it wisely, use it at the right rates when you need it, and then we can come back and keep using it because we'll have it. Whereas if we develop resistance to it or have other issues with it, we will not have this as a tool in the future. So it's really important that we use this as wisely as we can. But think about your crop rotations, and they are already getting more diverse. We've talked about spring drilling as a option for blackgrass control or grassweed control, but now we are looking at wider rotations. We're bringing in the cover crops, we're bringing in different species within perhaps a wider lay for a couple of years. So it really is starting to change, and we're seeing the benefits of that change. And again, I've mentioned it before, but think about the weed seed bank. It really is important. The weeds don't go away. They're there. Some of them will be there for a long time. Something like a poppy seed will be viable in the seed bank for 50 plus years. So it is really important you don't keep bringing those seed back up. Cultivar choice is something I think we're going to see a lot more of in the future, and this is something that you'll hear about this afternoon from Jed and from Dominic. But hopefully, again, for weed management for the future, this is something we can start perhaps picking varieties. We know that some of the hybrid barleys are much more competitive and good at suppressing some of our grassweed species. So cultivar choice should be high up there on one of your cultural control options. And competitive crops, make sure your crops are as strong as they possibly can be. So we need to think about drilling dates, seed rates, how you can get the best out of your crop, because you're reducing your weed burden and lowering that whole weed productivity. So if the weeds are not then going to maturity, you're not getting the seeds coming back into the seed bank. So all of this does need planning, and it needs time, and you do need to think about the rotation. So I'm afraid it isn't a quick fix, but it really will be worth it, and it really will pay you back. So finally, just to conclude there for both Katy and myself, so we've seen that herbicide resistance in grass weeds still does remain a big challenge. Broadleaf weed resistance must be considered, and particularly the use of ALS is often quite high across the rotation, so this is something that we are watching closely as researchers. Remember the importance of resistance testing. It is a really useful tool, so perhaps consider it within an IPM plan. Monitoring your weeds and identifying problem patches, and then remove those patches. So if it's something that's easy to remove or a small scale, then obviously remove it before it's setting seed in your seed bank is really important. But unfortunately for weed management, it is a longer term approach. So do know and understand your own seed bank, and it will reward you. So you really need to get to know the fields that you're farming or the fields that you're involved with. So do use glyphosate wisely. It is a really important tool for all of us, and we would like it to stay, so we all are responsible for that. And again, as Sacha said, IPM, integrated weed management in this context must really be followed, because it's really important for weed management. So I will leave it there, and thank you very much for listening.
Brilliant. Thank you so much to Lynn and to Katy for really laying out where we are in terms of herbicide resistance, but also talking about the implications of these changing management practices on future weed management, but also on sort of future herbicide resistance and what steps we can take to kind of, reduce that resistance going forward, and making sure that we still have those tools that we rely on, like glyphosate, going forward. So that was the last of our speaker sessions for this pesticide session. In just a moment, I'm going to be handing over to Ollie Johnson, who is Head of Agronomy and Engagement at AHDB. He's going to be leading the Q&A for this session. Before we do, just a quick reminder to get involved via Poll EV, so you can submit your questions there that will then be sent to the panel. We've also had a few questions from some of you about your basis and Neroso points and how to collect those. So after the event today, all of those of you who have registered will be sent a follow-up email, and that will give you the information required to register your basis and Naroso points. So yeah, just a thank you to all the speakers for this session. I'm just going to hand over now to Ollie to do the Q&A with the speakers.
Thanks, Henny, and thanks to the panel. Some really interesting stuff there. As Henny says, don't forget to get your questions in on Poll EV. You'll have seen the QR code over the course of today's presentations, I'm sure. One that's come in just now relates to light leaf spot curves. And should data be presented when the disease level is only being reduced from 6 to 4%? What is the LSD of the trial? Surely this is within the margin of error and data is effectively meaningless, especially when taking into account the pigeon and water damage.
I suppose I'll take that one on. Yeah. No, look, the curves we produce for fungicide performance are always within a statistical mean, so to speak. So there is a statistical reduction there for that control. If there wasn't a statistical reduction, it wouldn't have been presented. So first it's within a statistical mean. As mentioned, we're getting that minimum of that 40% efficacy from that level. And as I said, we're getting that reduction at that higher dose rate. So that was the caveat there for saying, yeah, we are seeing the reduction where we normally see an economic yield uplift for light leaf spot. So there is value within producing that data because it shows the usefulness to any sort of farmers or agronomists if they want to make a decision based on what they're seeing on there. Now they have to then particularly see that data and then make a decision within themselves, knowing the regional levels that you get with light leaf spot control. It can vary from region to region if they feel that there is a gross margin used from using that data. So we are providing a strong sort of service and a strong recommendation by putting those results out there as they're presented.
Okay, thank you. And I guess this, there's a question from me really, and from my experience, was around the sort of fungicide dose response curves. It's similar to that in cereals and in oilseed rape. We get a lot of questions relating to why we use responsive varieties when perhaps in the field we wouldn't use those. We would always be aiming to use a more robust variety. Why is it that we use those for the fungicide dose response curves? What's the methodology behind that?
Yeah, the main reason we choose varieties that are susceptible to disease or very susceptible to disease in some cases is to obviously partly to ensure that we provide a robust test of the products. And also to make sure that we generate some disease on the efficacy of those fungicides. I think if we were to use varieties that were relatively resistant to disease, obviously we'd find that some seasons we wouldn't generate enough disease. I think the other thing to say is that although we are using very susceptible varieties, often when we're doing trials we're able to be more prompt with our timing. And we recognise that actually out there in commercial practice there's lots of other pressures and people can't always get on as promptly. And so sometimes if you are treating even a moderately susceptible variety but treating it rather later than you should be, you will find that control is quite difficult to achieve. So I think what we're doing there is hopefully giving growers information about what is likely to be the most effective where they're trying to target those diseases.
Sure, and that's about bringing out those graphs and showing the differences. Great. Got a question here for Katy. Why do you think we are seeing more resistance in rye and meadow brome species compared to other species?
I think, well, one of the things we probably love and hate about bromes is that so much of their biology is still unknown. But I think what's come to light recently is actually in rye and meadow, we're seeing more spring germination perhaps compared to some of these anastanthus species like sterile. So perhaps whilst those sort of sterile bromes are being targeted by those autumn residuals that you're using for blackgrass, ryegrass, actually you might see a lot of spring germination of these rye and meadow bromes, and then perhaps you're a lot more restricted in what herbicides you can use. And again, you're more likely to use those ALSs. So I think from our perspective, it's probably that germination, but there is a lot more research to do on brome biology.
Sure, and you were talking about farming systems. Do you think that the rise of direct drilling, for example, has that had an impact in terms of the brome populations and the spectrum? Because certainly from my career, I've seen that really take off on farms where we have that very direct drilling approach.
Yeah, definitely. And again, it's all, as Katy said, it's to do with the biology, but now we are almost moving our species around a bit more with the farming techniques we're using and the changing in the kind of switch to spring drilling. We are seeing a shift in the species adapting to that. So weeds are obviously quite clever and able to adapt to these situations. So definitely we will see more changes over time as we change these kind of processes even more.
And it's certainly something that I'm concerned about as an agronomist, as we cut down more and more on the amount of oilseed rape that's being grown because of things like cabbage stem flea beetle, and that was an opportunity to control some of those growing species, and it is very difficult to control those in, well, really any crop except for wheat, and even then it's a challenge. So I think it's going to be a problem going forward as well.
Yeah, definitely.
We've got a question here, and this is one for the whole panel perhaps. Why has the data from the Scottish sites not been included in this year's data set? I assume this means to the fungicide dose response. No shortage of septoria in Scotland in 2024, as probably for the rest of the UK as well.
Yeah, so I can answer that one. So as Phil mentioned, we have some very rigid, strict criteria on data sets that are included in the data sets that we present, in the curves that we present. And they have to comply with a series of tests in terms of the level of disease, the statistical significance of the control over and above the untreated, and so on, and be robust data sets. So where we have data sets that we feel do not comply with those requirements, we don't include them essentially. Sometimes we may get yield data from a trial, but no disease data, or vice versa. And obviously they can be included in the wider set, but it's really down to making sure we have high quality control on the data sets we use. It's not because there wasn't a lot of septoria in Scotland, because there was.
Yeah, as many of our Scottish agronomists on the call today will no doubt relate to. Question here for Sacha. This chap says, or person says, if I understood correctly, the cages to which difficult insecticide treatments were administered were located in the same area. How can you be certain a treatment did not impact or reach aphid populations in other treatments?
Well, we only opened the cages when we actually had to spray that cage. We also had a boom designed especially for the project, so that we could get full exposure as quickly as possible when we were spraying a cage. We didn't have to open it we only had to open it for a short amount of time. There were very few winged aphids being produced in the experiment, so the chances of them moving between the cages was further reduced. I mean, you can't exclude it happening, completely, but there were processes put in place to minimise it as far as as feasibly possible.
Sure. Great. Okay, thank you. And this is a question for Lynn and for others. Regen Ag is anti-IPM in terms of herbicide reliance and resistance management. Discuss.
Where do I start? I'm not sure I agree with anti-IPM. I think we need to embrace the whole of IPM with this. So obviously with my weeds management hat on, I think actually as we go away from the reliance on herbicides, obviously that's a benefit for herbicide resistance and the impacts. We're not putting the pressure then, but glyphosate we have to be incredibly careful. That's the one I think we've got to worry about. But I think, as long as we are integrating lots of different tools, and we need to do it, we can't just put all the pressure on the herbicides, because particularly within, we've got very few actives anyway, so we are putting all our eggs in one basket. So I think for me it's all, as I said at the end, all about diversity, thinking about what you can integrate with that. So if you're using glyphosate in a stale seed bed, make sure you've got the dose right for the target weed, and you're bringing a cultural control option in there as well, cultivation. So for me, it's trying to maximise everything and all the tools we've got. So I hope it's a positive thing, and IPM should be a positive thing, that it's, we put labels on things, so Regen Ag could mean different things to different people. So I think we've just got to be careful labelling things and putting them in compartments. If we could all do a little bit of everything, then it's bringing an integration in.
I think one of the concerns that agronomists would have about, when we spoke today about the seed bank and about regenerative agriculture and cover cropping in SFI, is about some of those species within the SFI mix, and I'm thinking in this point particularly around Phacelia, and a lot of agronomists and farmers will be perhaps thinking about the weed return from that, but also other species that are growing and weeds that are growing within the canopy that we aren't controlling on these rotational type programs. Do you think there's a risk of adding more seed to the seed bank and building resistance through that?
There could be, and it is something we are watching closely, and we get a lot of questions about controlling certain species within cover crop species, so that's something that we're looking at the moment as well, because we need to understand what our herbicides will and won't do if we need to do herbicide control, and obviously we don't want the pernicious weeds to be going to seed in the cover crop that we think we're using for the benefit of other parts of our rotation. So it's certainly something to watch, but I think for me, again, having the diversity, you've got to, some level of species there, some level of weeds are actually good for the environment. So there could be some chickweed at a low level could be a bird feed source. So we've got to have that kind of diversity, get away from the monoculture and too many of the one species of grassweed. But if we could have a little bit of biodiversity in there with certain species, that would also be good. But it is certainly something we all need to watch going forward. And again, monitoring your fields, knowing if you've got particularly bad thistles, you might have to be very careful in your cover crop not to make the thistles worse in that situation, for example.
Okay, yeah, thank you. A question here. Is there an over-reliance on prothioconazole as an anti-resistance partner in new products, considering its own resistance status?
Yeah, I mean, I think it's really important to say that we should not rely on any single active heavily in any program. So whether it's a case of looking at different partners to add to your SDHI or to your QII, for example, other than prothioconazole, whether it's using mefentrifluconazole instead of prothioconazole, it's really about the range of different active ingredients and fungicide groups you use across the program. So yes, we wouldn't want to design programs that are heavily reliant on any single active, whether it's prothioconazole or anything else. And I'm sure that the industry is very conscious of the fact that that we do need to have other effective partners compared to prothioconazole. I mean, in some cases, prothioconazole is still very effective. For example, as a yellow rust partner, it's very good, very effective against that. Yes, its level of septorial control is not as good as some of the other products, but there we have a range of other choices that we can use. We have the SDHIs, we have multi-site, for example, which we can put in there as well. So I think choosing the right partner for the right disease, not over-relying on any individual active ingredient, either from a resistance point of view, also for overall disease control point of view, is really important.
And on that point as well, I mean, I noticed on the graph there that, of course, previously we used a lot of chlorothalonil, a very effective product, very cost-effective as well. People are now moving towards Folpet in Arizona, for example. And in a protectant situation, if I remember right, the Folpet was delivering around about, was it 15%?
40% control
yeah.
at the full dose.
And actually, when you looked at the protectant levels from, say, Adepidin and Inatrec, seem much more effective. Is there still a role for Folpet? Do we still need to put in that in the tank, or are these other products happy to stand on their own in other mixes with other things, of course?
Yeah, absolutely. I mean, I think, again, it's important to remember what does Folpet do? It's a protectant. So if you were in a situation where you were really chasing disease, you're not going to get that level of efficacy in Folpet. And we didn't even include it on the eradican because it's not an eradican fungicide. In a protected situation, again, as part of a program that uses fungicides from across multiple groups, it's very sensible to use it as part of that program. It won't suit every situation. It might not suit every combination, but it's one of a number of options you've got in the program to provide you some control. Because it is multi-site, it isn't selecting in any particular direction. So even though it might only be controlling 40% of the population, that 40% is not selecting, which is a useful thing to do. And really, as was talked about, as Sacha was talking about in terms of insecticide resistance, we try and minimise the reliance on any particular active, but in particular on any actives that are highly, highly effective, because those are the ones that potentially will select the fastest. So we try and use the non-selective fungicides as much as we can, and then use the others as an addition to that.
Okay, thank you for that, Stuart. We've got a question here just around biological fungicides, and they've used which I'm not entirely sure, I've never heard of it myself, polyversum based on the Pythium oligandrum. It has a range of serial registrations apparently. Has it been included in recent trials? And I suppose on this, are we looking to do more in terms of biological fungicides or biological controls as part of our fungicide work? This is for both of you, really, I guess.
Yeah, I know from oilseed rape work we have looked at polyversum previously in sort of small agar-based tests, lab-based tests at the very in vitro level, and some pot trial screening work, and there is potential for biologicals. It does need more further testings, more further trials, more on the field scale, more on the field scale trials, just to see where they can be used and where they can fit into fungicide programs to complement conventional fungicides which are available. I'm not sure from the cereals world if you...
We certainly are aware of the number of biological options. I think the challenge we have within the current fungicide performance trials is that they are set up to test conventional fungicides, and often the biologicals need a different approach. They might need a different timing. They might not perform well when they're in a trial that's got a lot of conventional chemistry in there. So in order to understand the biologicals properly, we need to have properly designed experiments that test those in that way. So I think it's certainly a need to have that, but probably not as part of the same trial series, as part of a different experiment.
That's really good. Thank you for that. And thank you again to all of you. That's been a really interesting session, and there's a lot. I mean, I've been taking notes all the way through, and I'm sure people have been at home as well. We're going to stop now for 30 minutes, and we're going to be coming back after that break for after lunch. So do join us again. Don't forget to look at that QR code. Get your questions in as we go throughout the day, and look forward to seeing you soon. Thank you.
This afternoon, we're going to have two sessions that are focused around sort of sustainable farming, and the first one of the afternoon is specifically looking at varieties. So kicking us off for this session, we have Paul Gosling, who is our Senior Crop Production Systems Scientist at the AHDB, and he's going to be talking about the recommended list. So over to you, Paul.
Thanks, Henny. Yes, in this slot I would normally give an overview of some interesting results that come out of the yield trials this year, things that don't always make it into the booklet. But this year we'll do something slightly different. I'm going to talk just about the RL fungicide programme and hopefully convince you that it's the appropriate programme for what we're trying to do with the recommended list. So what's the issue? Well, if we look at the cost of the RL wheat fungicide programme, and I will be focusing on wheat this afternoon, is that it costs about £260, obviously depending on where you buy your products and the exact rates you put on. But if you compare that with the cost of the average wheat fungicide programme according to the NIC's pocketbook in 2024, it's £113 a hectare. So you can see there's a huge difference in those two values. And the conclusion that most farmers and many agronomists make that the RL treated yields are, as a consequence, irrelevant to farm practice because no one would ever spend that much money on fungicides. So why do we spend so much money on fungicide for the RL programme? What are we trying to do? Well, the recommended list trials use what we call a disease exclusion principle on our treated trials, and that's designed to keep all disease levels below 10%. The reason we do that is it allows a variety to show its yield potential over a range of environmental conditions, from Cornwall to Cambridgeshire to the north of Scotland, without the effect of disease confounding the results. And it also means that all varieties are on a level playing field. So a variety like Crusoe, which is susceptible to brown rust, isn't disadvantaged in the trials, because we know some varieties which have disease susceptibilities can be commercially successful. But you may be thinking, the yield's unrealistic. If you look at the new recommended list that's just come out, the average yield of the wheat control varieties is 10.8 tonnes per hectare. If you look at the DEFRA survey, 2024 wheat yields are estimated at 7.7 tonnes per hectare on farm. So a huge difference. But trial yields are always going to be elevated compared with farm yields. There's a number of reasons for that. We avoid fields that are weedy. That's because the weeds just confound the variety effect. When we select the best parts of the fields, we don't put a trial on a headland because, again, it's confounded by compaction effects. Individual plots that are gappy or poorly established, we can take them out if we think it's not a varietal effect. And pest damage causing to plots, we've had everything you can imagine, badgers, swans, rabbits, deer, you name it, we've had it. We would take those plots out as well because that's not a varietal effect, that's just a random effect. There's another important effect that I have to take into account in trials, and that is the trial alleyway effect. Plants at the edge of the plot grow better because along those gaps, they have less competition for water, less competition for nutrients, and more sunlight, so they yield higher. So trial yields always tend to be higher than commercial yields. But remember, the world record yield for a semi-commercial scale is nearly 18 tonnes per hectare with Tim Lamyman. And we commissioned a review in 2023 with ADAS to look at the fungicide programme on the recommended list and see if we should change that programme. And one of the main conclusions they came out was the current system of robustly treated and untreated trials is the best way to show how to select and manage varieties when you combine them with the disease resistance scores, given the constraints of a trialling system where we're trialling lots of different varieties with different strengths and weaknesses. I'm going to say something a little bit controversial. The RL fungicide programme produces yields equivalent to good farm practice. And I'm sure many people out there won't believe that, but hopefully over the next few slides I'm going to show you some data that will convince you that this is true. And the data I'm going to use comes from the AHDB Fungicide Margin Challenge. This was a competition that was run over 3 years from 2019 to 2021 on some of our monitor farms. At each site, there was a single variety grown. There were several sites around the country. And it was a fully randomised plot trial with 3 replicates. The competitors in that were farmers and agronomists. We had an ag college team at one site. And they could put any fungicide programme together they wanted, up to 4 sprays. And the aim of this competition was not to maximise yield, it was to maximise margin. And that's an important point. Luckily for our purposes, the RL fungicide program and an untreated treatment was included within these trials. And they were dynamic programs. So each of the competitors received a report on the disease pressure in the trial prior to spray timings, so they could change their program to match disease pressure. All the timings were the same, T0, 1, 2 and 3, although not all the competitors used a T0. So the only thing that changed was the product and the rate between the competitors. The trials were run for us by AHDAS. They applied all the fungicides, and all the other applications of fertilizers and pesticides were the same for all the competitors. So I'm going to show you just some a set of results, or 3 sets of results from the 2021 year. And the first slide I'm going to show you is the results from the West, and this was Graham, perhaps understandably from the West. It's a very popular variety out there. And the chart on the left-hand side shows the yield on the vertical axis of each of those competitors. And along the bottom we have the fungicide spend for each of those competitors, excluding application. On the right-hand side, the red bar is the recommended list yield, and on the left-hand side is the untreated. So at this site, we had a yield response of 1.83 tonnes per hectare between the untreated and the RL. So a moderate response, which you'd expect with a moderately resistant variety like Graham. The RL program yielded 10.73 tonnes per hectare, and the average spend on the commercial programs, the competitors, was £82 a hectare. Average yield from the commercial programs was 10.3 tonnes per hectare, so a 0.4 tonne difference between that and the RL, suggesting that yes, the RL program does produce high yields. But the highest margin on this site, indicated by the green arrow on the chart, was from a £79 spend, and the yield from that was 10.62 tonnes per hectare. So actually the difference between that and the RL program was only 0.1 tonnes per hectare. Moving now to the East Anglia site, a very different variety, Skyfall, obviously very susceptible to yellow rust, and a very large yield response as a consequence, nearly a 4 tonne yield response between the untreated and the recommended list. The RL program yielded 10.98 tonnes per hectare. The average spend on the commercial programs was £79, and the average yield from the commercial programs was 10.01. So nearly a tonne difference, again suggesting the RL program does produce unrealistically high yields. But the highest margin from this site, again indicated by the green arrow, was £114, 10.76 tonnes per hectare, and only 0.2 of a tonne difference between that and the RL program. And third, the Northeast trial, KWS Parkin. KWS Parkin never actually made it onto the recommended list. It came very close, but it did take a chunk of the commercial market over a couple of years. So the chart shows the same thing. Very big yield response at this site, 3.63 tonnes per hectare, and a very high yield from the RL program, just over 12 tonnes per hectare. The average spend on the commercial programs was higher at this site, £97, reflecting the high disease pressure. And the average yield of the commercial program was 10.76 tonnes per hectare. So a very big difference there, 1/3 tonnes between the RL program and the commercial average. But again, if we look at the highest margin, again, with the green arrow, £115, 12.04 tonnes. And so the difference between that and the RL program, in fact, was only 0.03 tonnes per hectare. So a very small difference between those two. And on this chart, I've combined the data from all the 6 sites in 2021. On the vertical axis, we've got something slightly different. So we've got yield as a percentage of the RL fungicide yield. So at 100%, we have the red line, which shows that yield. And we've got the fungicide spend across the bottom. This time it includes application costs, so that the numbers are slightly different along there. But if you look at all the black dots, they are all our competitor programs, and you can see lots of the programs came very close to, equaled, or in some cases actually exceeded the RL fungicide program. And we're often told we should move away from a disease exclusion system to something like a farm standard or a commercial program. But if you look at this data, we could see different programs which people chose, which were aimed at being the highest margin, i.e. a commercial program, gave a huge spread of results. If we pick any single price point, so we picked £100 per hectare to spend on the RL program, you can see there would be a huge difference in the result we would get depending on the program we used. So in fact, we would be looking at not a variety trial, but a variety by fungicide trial. That would make it hugely more complicated to interpret. Just looking very briefly at the 2020 data, there were 3 sites in 2020. 2 of those sites, there was actually no statistically significant response to fungicides. As a result, as you can see, lots and lots of the programs there actually equaled or exceeded the recommended list trial. Recommended list program, I should say, sorry. So again, our farm standard would have given us a huge range of potential yields. Again, it would have been a variety by fungicide trial, not a variety trial. So what do these results tell us? Yes, the RL program is extreme in its cost, but not in the results it achieves in terms of yield. It adds very little extra yield compared with what the best commercial programs can achieve in normal circumstances. The best commercial programs aimed at maximising margin can yield as high or higher than the RL program. Why is that so? Why is we not getting this extra yield from the extra spend? Well, we have one program for all varieties in all locations. That means it has to be comprehensive to control diseases across all locations in all circumstances. That makes it very expensive. But a significant part of the RL program is not required for all varieties or at all locations. What do I mean by that? Well, if we look at the core wheat program, we can see on here a number of products that aren't really going to be doing very much in terms of boosting yield in different circumstances. So we look at the T0 timing. We start with cyflamid, a mildewicide. It's expensive, it's going to add a lot of cost to the program, but actually it's going to give you a very small yield response or no yield response at all in many situations. We've got tebuconazole and Comet in there. Yes, they're going to have an effect on varieties which have got susceptibility to rust, but varieties which have high rust resistance, that really isn't going to add you any yield benefit. Our T1 is pretty standard, Revistar plus Arizona. Again, we've got a mildewicide in there. Again, is that really giving us any yield increase? In most situations, I would suggest not. T1.5 is optional, but it's not particularly strong. If you look at T2, Univoc plus Arizona, again, pretty standard, what you'd expect on farm. I'd question how much the Arizona is boosting yield when you've got such strong alternative products in there. And if we look at the T3, Prosaro, it's relatively expensive, but mainly targeting Fusarium. It doesn't have much impact on Septoria. And again, you've got Comet there. Yes, useful for rust-susceptible varieties, but actually it's not going to do very much for rust-resistant varieties or in situations sort of the north of Scotland where you'd very rarely see any rust. The other thing to take account of is the declining benefit of increasing rate of products. So this is some work from the AHDB fungicide performance curves. You saw some of these earlier. And we've got on this chart the Revistar and Univoc, which was in our program. And if we look at the area which I've called farm practice, sort of between a half and 3/4 rate, and compare that with the RL, which is around 3/4 to a full rate, actually the difference in disease control at those very high rates is actually very small. You're not really getting very much better disease control from going higher in terms of rates. So, what are the conclusions? One fungicide program that aims to control all the diseases in all situations is fair to all the varieties. We're not advantaging a certain type of variety. But no variety is vulnerable to all diseases. No location will be vulnerable to all diseases. And something I haven't really talked about yet, no year will experience high disease pressure from all diseases. Not every year is going to be a brown rust year. As a consequence of that, parts of the fungicide program are not increasing yield, at least not on all varieties in all locations. Therefore, the fungicide program we use in the RL produces yields that are similar to what can be achieved by a good farm program, and I hope I've shown you data that convinces you that's the case. The treated and untreated yield plus the disease resistance ratings provide all the information you need to select and manage varieties. So, I would advise you stop worrying about the RL fungicide program. The data is there. Use the RL. Thank you.
Brilliant. Thank you very much, Paul. A pretty key takeaway message there, and thank you for that great sort of deep dive into the RL fungicide programme. So up next, our second speaker for this session is Diane Saunders. Diane is the Head of Department at the John Innes Centre, and she's going to be talking to us today about the Delivering Sustainable Wheat project.
So thank you very much, Henny, and I'd like to start by, of course, thanking the organisers for putting together this absolutely fantastic meeting. I hope you're all really enjoying it so far. And so now I'd like to shift gear a little bit and talk to you and give you a bit of an insight into one of the largest and coordinated wheat improvement programs here in the UK called the Delivering Sustainable Wheat Programme, also known as DSW, and this is funded by BBSRC. And this DSW program is just the latest in a series of wheat improvement programs that began over 12 years ago in 2012 with the introduction of the Wheat Institute Strategic Program. And when this was introduced in 2012, the aim of this program was to act as a comprehensive pre-breeding program to bring together experts in wheat genetics and also plant breeding to ultimately guarantee the sustainability of future UK wheat production. Then in 2017, this program morphed into the Designing Future Wheat program. Again, the aim of this program was to develop new wheat germplasm, which included some of the next generation of key traits. And then in 2013, finally, the Delivering Sustainable Wheat, this DSW program, began. And of course, at the heart of this program was, again, of course, the important aspect of safeguarding the future of UK wheat production. But to do this, the team decided to divide this program into 4 key areas which would address some of the key challenges in this kind of area of research. Firstly, looking at how we can safeguard future and increase future wheat yields and quality, also address those biotic threats to plant health, and then to increase the nutritional value of our wheat varieties, whilst also ensuring that as we go along, all of the vast array of data that we generate through this program remains accessible to the widest proportion of the community. And so to achieve these goals, Simon Griffiths, the head of the DSW program, brought together again a whole array of wheat researchers from across many different organisations within the UK, and he brought them together with the plant breeding community so that we could work together to achieve these united goals. And just some of the key questions that we've been working on within this program are, of course, looking at how we can increase wheat yield with the same nutritional input, looking at how we adapt to those rapidly changing agricultural systems, integrate pest management and increase wheat nutrient density, whilst ensuring all along the way that our data that we're generating remains fair, so findable, accessible, interoperable, and reusable to ultimately achieve our goal of producing a sustainable wheat production for UK farmers. And so this DSW program is very much a forward-looking program, but today I also wanted to share with you an example of a project which reconsiders an older question where we're applying now some of the latest technological advances in wheat research to address this older question, reconsidering the origin of modern wheat. And this project I'm going to just briefly talk about is very much led by Simon Griffiths, the head of the DSW program based at the John Innes Centre. And as many of you will be aware, of course, when we think about the introduction of modern wheat breeding practices, these were introduced in Europe at the beginning of the last century, where at this time Sir Roland Biffin introduced wheat genetics into the plant breeding sphere, where he realized at this time that some wheat plants contained constitutions that could be passed on to their offspring. And in particular, he focused on constitutions which provided resistance to certain attack by certain fungal pathogens. And in this case, one of the first varieties that he developed through the crosses that he generated was through a cross from a variety that was actually resistant to yellow rust infection and another variety that was high yielding, crossing them together in order to bring these traits together in one wheat variety. And this new variety that was generated was termed Little Joss, and of course farmers were really pleased with the production of this new variety, and they hailed him the Wheat Wizard. However, at this time, of course, the introduction of genetics into wheat breeding also brought along with it inadvertently a potential issue, which was the introduction of a potential genetic bottleneck as the diversity of pedigrees started to reduce. Of course, the plant breeders wanted to utilize the best possible varieties in all of their crosses, and a lot of those varieties had the same genetic background, and so this started to then reduce the kind of diversity among the wheat varieties that we now rely on. But at this time in the 1920s, another very forward-thinking geneticist, Arthur Ernest Watkins, realized that these modern plant breeding practices could potentially start to reduce the diversity of our wheat varieties here in the UK and elsewhere. And so he started a mission to collect a lot of these older land races which had existed and farmers had worked with ever since we started cultivating wheat over 10,000 years ago. He collected these older land races by writing to various countries around the world and making this amazing collection of these highly diverse land races. And this has really nicely articulated the story of how he collected this collection of wheat varieties within this Genetic Society magazine article that was just recently published last year. But also what's nicely articulated within this article is the efforts of the whole community for a century after Arthur Ernest Watkins put together this collection to preserve it, to make sure that it still is with us today. And today, 827 accessions survive from this Watkins collection of wheat varieties, and these were collected over 32 different countries. And over the past 12 years, since the introduction of this first WISP program, this Wheat Improvement Program, this collection has really become the center of a national and international effort to unlock the diversity among these land races. And so some of the latest efforts to unlock this diversity have again been led by Simon Griffiths, working with his colleague Shifeng in China, where they aim to sequence all of the varieties within this collection, so all 827 accessions. But also, not only did they sequence all of these accessions within the Watkins collection, they also collected another 208 modern wheat varieties and sequenced those too, so then they could provide a comparison between these older land races and the newer wheat varieties that we're now growing today. And to cut a very, very long story short of more than a decade of research, what they found was that when they looked at the diversity among that Watkins collection, that rich diversity that was present in those land races, and they started to pull this apart, they realized this diversity could be grouped into what we call 7 ancestral groups, these AG groups. And then when they overlaid the modern wheat varieties and the diversity present in modern wheat varieties, well, that was actually collected in just 2 of these groups, this AG2 and AG5. And so what this tells us is that, yes, indeed, we have been through a massive bottleneck where we've reduced the diversity substantially of our wheat varieties, and actually over 60% of the genetic diversity that was once present in that Watkins collection, in the land races that our farmers relied upon, has now been lost in the development of modern wheat cultivars, and that diversity is now only present in those land race collections and remains untapped. But of course, the big question is that diversity actually useful? So is any of the diversity that was left behind in those land races actually useful for modern UK wheat breeding? Well, to address this, again, the large community of international researchers came together and they conducted over a decade of field trials assessing over 200 different traits. And this included a whole array of different traits, looking at improving, of course, wheat kind of yield, looking at improving grain quality, right through to addressing biotic stresses. And from this analysis, they found there were 1,700 genetic effects that they found that had the potential to improve modern cultivars. And 36% of these genetic effects were only present in these land races, so were absent from modern wheat varieties. And just as a very brief example of a couple of the traits that they decided to look at, particularly here in the UK, they decided to consider, of course, winter kill, which is going to become an increasing problem, especially as we start to see more and more extreme climatic conditions due to climate change. And then they also considered yellow rust resistance, and they identified 33 accessions within this Watkins collection that provided resistance against the warrior-type race isolates, which many of you may know are very prevalent across the UK and Europe. And so these could act as a really great source of new sources of resistance. And a lot of this work was conducted both at the John Innes Centre and Rothamsted. And so at the John Innes Centre, led by our amazing field experimentation team, and then at Rothamsted by Malcolm and Andrew, who have now identified thousands of genetic effects that might be useful for UK wheat breeding going forward. But of course, the work that's been done here, this is an example from the UK, and this Watkins collection isn't just valuable to the UK and UK wheat breeding, it's also extremely valuable for international efforts to improve wheat production. And so to really illustrate this, I wanted to bring you to an example of wheat blast, which is an emergent disease that is hugely damaging to wheat production. It isn't present in the UK, so it's not something for us to worry about here in the UK. And this disease is very new. It was first identified in Brazil in the 1980s, and then it very quickly spread to neighboring countries in the 1980s. And what's really problematic about this disease is it can create yield losses of up to 100%. And so it's hugely devastating when you have an outbreak of wheat blast. But up until just a decade ago, this was very much a problem that was isolated to South America. But sadly, over the past few years, in 2016, we saw this disease being identified for the first time in Bangladesh, and then in 2018 for the first time in Zambia. And what's very problematic about this wheat blast is we have so few sources of resistance against wheat blast, and so our wheat crops are extremely vulnerable to infection. And so there is an urgent need to identify new sources of resistance. And so to address this, Paul Nicholson, another group leader at the John Innes Centre, decided to look at the Watkins collection and see whether he could kind of look into that collection and identify any potential new sources of resistance. And to cut another very long story short, what he and his team found was that they identified a gene which could provide resistance. It existed in a number of different alleles, and a couple of those alleles could provide resistance at both the seedling and spike tissues. And these alleles, importantly, aren't present in modern wheat varieties. They are only present in these older land races that are part of the Watkins collection. And so I think this nicely shows, of course, how the Watkins collection has been coming to the rescue, not only through the UK work, but also through the work internationally. But this is, of course, just one example of the many, many traits that this amazing community that has come together have started to unlock through the diversity of this Watkins collection. And I'd just like to, of course, finish by acknowledging all of the amazing people who are involved in the work that I've had the privilege to share with you today. And I'd particularly like to acknowledge Simon Griffiths, who has expertly led the wheat community across the UK, bringing together all of us in this DSW program. But not only that, but also helping us to unlock that diversity within that hugely valuable Watkins collection that I'm sure will reap benefits for wheat production for many, many years to come. So thank you very much.
Brilliant. Thank you, Diane. What a fantastic project to be a part of, and yeah, really looking forward to hearing about more of the findings from that project so far. So thank you for sharing that with us. So up next we have Jed Clarke. Jed has just recently finished his PhD at the University of Leeds, where he's been looking at cereal lines that are good against battling blackgrass. And so Jed's going to share some of the findings of his PhD with us today.
So, hi everyone. As Henny said, my name is Jed Clarke and I've just finished my PhD. So today I'm going to talk about some of the main findings that have come from that. So the main aim of my PhD was to identify varieties of winter wheat that may be used as candidates for competitive cultivars in integrated weed management against blackgrass. So I'm sure I don't need to tell everyone what blackgrass is, but if you don't know, it is the grass weed that affects arable crops, and it is a particular problem in crops of winter wheat where it can outcompete the crop. And blackgrass is very good at becoming herbicide resistant, so we need alternative approaches of dealing with it. And one of those approaches is the use of competitive cultivars. Now, they have some benefits, so they're comparatively cheap and easy to implement compared to other IWM approaches. And we have seen from previous research that they have shown benefits in blackgrass control. However, research into this area is limited. That studies that have been done look at sort of small number of cultivars, and it's hard to then compare studies between each other because they look at different environments, different set of experimental conditions. So the uptake of competitive cultivars is poor. This is due to low confidence by farmers, basically because the cultivars that they will plant is based on sort of anecdotal evidence from other farmers. It's not based on any sort of scientific grounding. And currently we have sort of no quick and easy way of assessing what actually makes a cultivar more competitive than another. So we really need to understand how these plants, so how wheat and blackgrass are actually interacting with one another, how they are detecting neighbours in their vicinity. And what we know is that above ground plants can detect each other through multiple methods, through volatile stimuli, touch stimuli, and through light, so shading. And also below ground there's evidence that plants can detect each other via mycorrhizin through root exudate production and possibly electrical stimuli. So above ground traits that plants have that may increase competitiveness may include height, tillering, and the ability to shade weed plants. But below ground traits such as root growth, chemical exudation has not really been studied to any great extent, basically due to the difficulty of assessing stuff that's in the soil. So that's where my project focuses. So the first thing we kind of need to understand is how and when competition is occurring between wheat and blackgrass. And are there any below ground interactions which are influencing competition? We then went on to screen our crop varieties for variation in root growth. The next thing we did was we came up with a sort of a quick and easy method of assessing crop competitive ability, and then screened many elite wheat lines and also some landraces and barleys for their competitive ability. And finally, what we wanted to see is there a correlation between the root growth of crop lines and their competitive ability against blackgrass. So the first thing we did was went right back to the start of growth, and basically we compared blackgrass and 3 elite wheat lines, and we looked at these 5 different attributes, so germination time, time to produce a root system and a coleoptile. And basically we saw that blackgrass took far longer than the elite wheats to do these things. When we look at shoot biomass after 2 and 4 weeks, we also saw that blackgrass had a much smaller shoot biomass. And so this kind of indicates that in those first few weeks of growth, blackgrass is very unlikely to be gaining a competitive advantage, and it's therefore likely that competition is occurring at a later stage. So we then went on to obviously look at growth at a later stage, and here we grew barley, wheat and blackgrass in a hydroponic system. So this allows us to visualize both the root and the shoots. And what we saw, so these were grown in winter conditions, and what we saw was that blackgrass and barley were able to produce a much larger plant in terms of both shoot and root production than wheat was. So they had a much sort of a greater winter growth rate. So this may indicate an area where blackgrass is gaining a competitive advantage over wheat. You can see from the pictures the visual difference in the amount of roots that are being produced there. And also the fact that barley, which we know to be more competitive against blackgrass than wheat, the fact that barley is also having similar levels of growth to blackgrass may indicate how barley is able to counteract blackgrass competition. And if you look at the bottom right graph, we see the shoot to root ratio. So of the total plant biomass, the proportion of that biomass which is attributed to its shoots or roots, so the attributed to the roots is greater in barley and blackgrass than it is in wheat over winter. So that means that sort of blackgrass and barley put a greater emphasis on below ground growth. So the next thing we wanted to look at was when is that competition actually occurring? So here we have an experiment where we grew wheat with and without blackgrass competition, and we looked at crop tiller number over time. And basically what we saw is that it took to around the point on, you can see on the graph, 1,180. It took to around, that's about equivalent of 4 months growth to actually see any physical impact of blackgrass competition on crop tiller number. So this kind of indicates that either competition is only occurring at sort of when spring starts, at the point of booting, or is competition gradually occurring throughout the whole of winter, only it's unseen below ground, and that competition has actually already occurred and the plant has already won or lost due to the level of root growth that it has by this point. So this kind of led us to put forward our root growth hypothesis, saying that blackgrass gains a competitive advantage over wheat during the winter period due to increased root production and the prolonged winter season. And the fact that spring plantings are less affected by blackgrass is one, due to the low amount of blackgrass we see germinating in this period, but also the shorter growing season doesn't allow blackgrass to accrue the same level of root growth and the same below ground advantages in winter growth. So to test this, as I said before, we're going to screen for root growth. We're also going to screen for competitive ability and see whether there is a correlation between the two. So we had two ways of assessing root growth. One was riser boxes, so you can see a picture of the riser box there. And the second, so riser boxes were grown in spring conditions. And the second was the use of hydroponics, which were grown in winter conditions. So this is the riser box data, and we can clearly see that there's variation in root cover, so the percentage of that riser box surface that is made up of roots. We see that varies dependent on the crop variety that we're looking at. And the blue bars represent elite wheat lines, the sort of pink-purple bars are barleys, and then we have landraces in yellow and blackgrass in red. So we can see that the barleys tend to have quite a high level of root growth, and there is variation in root growth amongst elite lines. What was interesting is that the landraces and the blackgrass tend to have a fairly low root production when grown in the riser boxes in spring conditions, but when we grew these in winter conditions in hydroponics, the landraces and blackgrass had a much higher level of root growth, amongst the highest of all the lines, whereas for the elite wheat lines, the trend of root growth was pretty similar between the two methods of root assessment. So this brings us on to our main competition screen. So again, this is where we had a wheat plant grown in competition with blackgrass, and as a control we had the crop by itself. And what we did was we determined the percentage loss in crop biomass upon the addition of blackgrass. So how well is that crop variety able to tolerate the presence of blackgrass? So the top graph shows that data, so we can see that towards the left of the graph we have our barleys in pink and our landraces in yellow, and generally they are the best tolerators of all of our lines that were tested. And to the right of the graph you can see it's dominated by elite wheat lines. So obviously the elite wheat lines are struggling in their tolerative ability, but what is sort of positive is that we do see some elite wheat lines such as Cairn and Elation on this graph, which are showing sort of equivalent tolerance to some of the barleys and landraces. Then the bottom graph here is for the same experiments. This is the percentage blackgrass, so the percentage of the total pot biomass, in which when we have the competitive scenario that is blackgrass, and we can see basically a graph showing the suppressive ability of each of these lines. And generally you see the same trend as in the graph above. We see the barleys are the better suppressors, the landraces also do fairly well in suppression, and then we have this sort of variation in our elite lines, with again some lines showing promise. Now, because this competition screen was done in spring conditions, we needed a way of showing that the data we've got from this is transferable to the field. So what we did was we took 7 lines that we used in the lab and we grew them in container and field trials throughout the winter growing season. And I won't go through all the data, but basically what we saw was a similar trend in competitiveness of these lines between each of our environments, so we can have some confidence that the results we're getting for competitive ability in the lab are representative of what may be seen in the field. And the final graph, so this is showing root growth correlated to competitive ability. So on the bottom axis we have root growth, and on the side axis we have the percentage blackgrass, so the suppression, and we can see that there is a sort of general sort of negative correlation showing that as root growth of a crop increases, the percentage blackgrass decreases, so the suppressive ability of that crop is basically increasing as its root growth increases. Interestingly, we didn't see any correlations between crop tolerance and root growth. So just to conclude, we see that there is variation in root size amongst elite wheat lines. We see also variation in tolerance and suppression, and increased root growth may lead to increased suppressive ability. There's lots of sort of avenues that this PhD work could lead to. So further work could include assessing the impact of different environmental conditions on the performance of competitive cultivars. What one cultivar that's competitive in a certain set of environments may or may not be competitive in another. We need to look at the effects of early vigour and increased winter growth rate on competitive ability. It could be that that period during the winter where blackgrass is gaining its competitive ability might be a key area to focus on selecting wheat for, to give it the best sort of competitive outcome. Further work identifying the genetic basis for competition in wheat. Can we breed for competitive ability? We need to highlight the importance of the root system not only for competition but also for environmental adaptability. So with a sort of changing climate and changing weather systems, increasing that root system may make sure that that crop is going to survive a wider variety of conditions, and it also could help in soil health. And this is just sort of a small part of some of the work that I've been doing. And something else that we did do a lot of work on was assessing the role of chemical root exudates in cropweed competition. So I know rice and barnyard grass chemical competition has been seen to affect weeds in that system, and it's the possibility that in wheat blackgrass that root exudates could also be playing a role. So I'd just like to say thank you for listening, and thanks to my PhD supervisor Tom Bennett and the Bennett Lab back in Leeds, to Lynn Tatnell and the team at ADAS in Boxworth, and also to AHDB and University of Leeds for funding my project. Thank you.
Brilliant. Thank you very much, Jed. Thank you for sharing those findings of your PhD with us today. And it seems like it's really opened up even further questions for researching. So yeah, thank you. So we're about to head into our last presentation of this session. So just a reminder to all of you at home to please submit your questions online via Poll EV. And Jed's presentation just now actually leads really nicely into our last session of the day. So that's from Dominic Amos. So Dominic is a research agronomist and a natural capital specialist at Cope Seeds. And so Dominic is going to be talking to us today about breeding varieties for low input, or more specifically for organic systems. And that kind of, leads really nicely from Jed's, who's obviously been looking at early vigour and varieties that are good for weed control. So yeah, I'm going to hand over to Dominic.
Thanks, Henny. Right, so I'm going to talk you through basically a project that's been 7 years in the making, really. It's a farm-based organic variety trials network that we started back in 2017. Just quickly, some housekeeping. Those are the various guises that the projects were under. We have funders in there and our partners. I'd like to say a thank you to the breeders that have kind of collaborated in terms of kind of recommending varieties for us to try, but also in supplying the seeds. I don't know, you can imagine that getting these kind of farm scale quantities, 100 kilos of each variety out to 4 or 5 farms is quite a challenging task, so we really appreciate their efforts in supporting that, and the seed merchants as well that have also played their part. But the real heroes of the work have been the farmers, so we've had a real commitment from a group of organic farmers right the way through, lots that are taking part for the whole 7 years, and the ones that are underlined there are taking part this year. And I also want to give a big shout out to my old colleague and boss, Ambrogio Costanzo. So if this was the Manhattan Project, he's very much the J. Robert Oppenheimer of all this. And it was his ideas and inspiration that really got this off the ground. So that's a quick précis of what I'll be running through. Obviously a lot of relevance for organic farming systems, but I've tried to pick out some key points that I think are going to be relevant for the wider farming sector. So the background, I'm going to whistle through this because I haven't got much time and I've got about 50 slides. So in summary, organic winter wheat production in 2017 was in a bit of a state. We had a shrinking acreage. It was considered a really difficult crop to grow agronomically. I think if you went into a typical winter wheat field, it quite often looked like that picture. And we, the standard kind of testing and breeding is obviously not really geared up for that environment. So we thought, well, how are we going to tackle this challenge? So collectively, as researchers and as a group of farmers, we set up this kind of on-farm collective experiment to see if we could find out whether we could identify varieties that were more suitable for organic farming, because it absolutely was part of the issue. And we didn't really start with much of a hypothesis. We were kind of testing a model. We weren't quite sure what we'd learn or if we'd even learn anything. But having that really open-ended kind of approach actually has allowed us to learn a lot and create avenues for further research. We'd also kind of concurrently, we were doing a organic plot trial, and we felt that kind of lacks relevance. One of the really important things is obviously being able to assess the performance within the environments that you're going to grow them, and crop management is a really important part of that. And we weren't able to mimic farm practice on the plot trials, which is why we thought actually, farm strips are going to be much more relevant. I'm going to skip past that because I haven't got enough time. There's a quick overview of the network where we started in 2017. We had 7 farmers who we called the Magnificent Seven. Yul Brynner, Charles Bronson. I've got some quotes from them later. So as of 2024, there are currently 15 farmers taking part, and we're looking at 17 varieties. And across the whole 7 years, we've had 23 organic farms and 4 non-organic farms who would self-identify as regenerative, who were also interested in wheat performance under lower inputs. So simply strips of various varieties within the field, we'd collect, so the farmers would drill them and harvest them. They provide yield data and a grain sample, so we could analyse that for quality. And we were in the field doing various agroecological assessments, crop height, vigour, crop cover, weed communities, disease. So these are some of the selected results from the previous 2 seasons. As I said, it's gone on for 7 years, but obviously the commercial life of varieties can often be quite short, so I've tried to keep this as up-to-date as possible with the latest varieties that hopefully you will recognise. So the graph on the left is the 2 years combined, and the 2 graphs on the right are either the 22-23 season or last season. So a couple of points to highlight here would be 3 blends that we've tested across those 2 years have outperformed the average of their components, which is a really powerful result. And I'm going to talk a bit more about that in a second. And also, probably for the first time, we'd had a variety that had performed very consistently organically that had a really poor season, so kind of an interaction between genetics and environment. That variety was X-Days, but I'm going to go on to talk a bit more about why that was. And that brings me on to another really important point. So whilst we're testing all these varieties and they're named and have various breeders, really you need to think in terms of traits. So these varieties obviously have names, but it's the traits that are the really important thing. And there's no criticism or promotion of any particular variety. I think modern breeding is totally fit for purpose, and I think it produces good varieties for the environments that it needs to. But we also have some of those varieties that are able to perform under lower inputs and have the right traits that certainly organic farming systems need. So we just had Jed's presentation, and I think this kind of follows on really nicely. So a lot of the focus has been on weeds. It's absolutely the main production challenge in organic winter wheat would be weed competition. So we've been looking at trying to identify, the mechanisms by which the varieties can compete with the weeds, and if we can identify varieties that actually have those traits. So the three main mechanisms: early ground cover, hugely important. If the crop covers the ground, then you've got a great chance of suppressing weeds. That's really in the early stages of the season. Critical weed-free periods usually over winter. That's when most of your yield components are set. A lot of the yield potential is determined by the time crops go into stem extension. So that's really important. The early vigor, growth rate, can think of it as kind of biomass, partly linked to the phenology, the growth cycle. So obviously earlier varieties are going to have a lot more biomass by the time they get to stem extension, or let's say by calendar date, they're going to have more biomass than those later varieties. So they actually get ahead of the weeds. And the final mechanism is late height. So the tall-strawed varieties we find really do suppress biomass later in the season. But as I've mentioned, that doesn't necessarily, that's more of a whole rotation system thing, because that doesn't necessarily help with yield improvement, because the damage is often done early. So just to bring these kind of mechanisms to life. So there's an idea of early ground cover in February and March. So on the left, you can barely see any soil visible. So that's the variety Mayflower, really weed suppressive, and I'll show you some other photos of that in a second. But tillers really well, very prostrate. So over winter, it's smothering the ground, and it doesn't really leave any space for weeds to come through. And obviously on the right there, you can see a slightly less, a lower ground cover variety. And then a similar thing on the bottom as well. So that was the early season picture. That's what you get from a high ground cover variety later in the season. And the only difference, so if you split those two photos in half, the only difference between left and right, so everything else being the same, it's only genetics, the variety. So that's what Mayflower can do to a reasonably weedy field of charlock. Really, really suppress it. And I think if you were doing any kind of weed suppression assessment on that and a relative biomass, you would say you have 95% control of the charlock in that field, purely from the variety. So the idea that cultivars don't play a big part in weed suppression, I think really needs to be challenged. Again, on the left, you have the variety Mayflower for its early cover. Probably useful if you squint at this photo, but you can definitely see there's a lot less poppies on the left. In the middle is actually a blend of two varieties, and then on the right, a slightly less weed suppressive variety from last season. And obviously, we want more organic fields to look like that strip on the left rather than the strip on the right. And again, identifying the right weed suppressive cultivars has been a huge part of that. Similar thing again, on the right. Again, I've tried to draw a line down there and also kind of stylized the image because it was difficult to see that photo on the right. But basically, that weed suppressive variety with very high ground cover on the right has really helped to suppress wild oats. It's kept them small. It's reduced the number of spikelets on the panicles. Whereas on the left, you can see they're really growing very strong, very vigorously. So I even think against quite competitive grass weeds, varieties, weed suppressive varieties can be identified that are going to be able to make a difference, and they have to be part of the solution. Again, eagle-eyed amongst you will note that that's not wheat, it's oats, but it emphasizes the point really well. So this again would be in kind of March, April time. If I want to suppress weeds, I would much rather have that variety on the right than the variety on the left. So we really need that early vigor to allow them to be more competitive. And then the final mechanism, I said, late crop height. So again, that on the right was a modern cultivar on the RL that actually is one of the more weed suppressing varieties. On the left is a kind of heritage population, and you can see much less poppies, less annual weeds in there, and that's what late height gives you, a real reduction in biomass later through the season at heading. Again, slightly mixed picture. This is a variety that's late. It has a late growth cycle, and as a result, doesn't have very good ground cover coming out of winter, has low vigor, also a lower height through the season, and you can see, again, it's kind of lost the race. Now that's a strip of groundsel, sorry, a strip of wheat, but groundsel is the kind of main weed burden within that field. And you can see that it has really been poorly competitive. Those are the two other varieties within that field, one with high ground cover, one with high early vigor, that have both suppressed that weed. This looks very fancy, but the key take-home message really is that grain yield and ground cover stem extension are really closely linked. And unsurprisingly, those two traits are negatively correlated with weed cover at 32. So to delve in a bit more into management practice, there are two main methods for controlling weeds in organic systems. So you're either a farm on a narrow row system. Crikey, I'm running out of time, aren't I? Yeah. You're either farm on a narrow row system or on a wide row system. So we'd say there's more of a scale of sort of intensive weed management towards the wide row systems. So you're inter-row hoeing within those. Quickly, this is showing that whilst you might control more weeds with the hoeing, you actually tend to promote a relative abundance of more dominant weeds. Lots of these slides you'll have to go back through and look at and read on YouTube. Again, we found that the really pernicious weeds were promoted in 1 year in 2020 and 2021 within the hoeing system. So this image shows how the weed communities differ between the two management practices. So actually, you'd think they were pretty extensive weed management practices, but the weed management with wide rows and hoeing actually promotes a lot of the really pernicious, really nasty competitive weeds: blackgrass, wild oats, brome, couch grass, creeping thistle. So again, it goes to show that if you're implementing intensive weed management, you're often selecting the nastiest species. So the impact, again, very quickly, basically from where we started in 2017 to where we've arrived at now with the varieties that we're testing that are being grown commercially, we've actually seen a 33% yield increase in terms of the varieties that we're growing, which I think is a massive impact and has really changed organic winter wheat production. So again, fields tending to look like that on the left in 2017, and now much more typical to walk into an organic wheat field and see a picture like the one on the right. I haven't got time to read these, but these are the farmers that have taken part saying how great this was, basically. So wider relevance, climate extremes. Three main challenges, and these apply whether you're organic or non-organic. So we've got increased stress that the crops are under. It affects pest disease and weed pressure, and affects the efficacy of the inputs and the ability to apply them. Remind you of, I think spring is where we see the real impact. So we have very dry springs in 2020 and 2022. How do we potentially create some more resilience against it? So we've been looking at these two-way blends, which I showed you from the results last year. We think that if you combine an early variety and a late variety in a blend, it becomes more resource use efficient and it becomes more climate resilient. Again, we have RL scores, so we have some early varieties and the later varieties. The later varieties we think are less weed suppressive and also maybe more susceptible to spring droughts. If you're an earlier variety, you're more resilient against it. Where this is interesting, very, very quickly, is actually that very few varieties on the RL are early varieties. Now there's an obvious explanation for that, which is the longer a variety spends in the ground, the higher the yield is. And that's what generally things are being selected for in the RL. But it may be that those varieties are actually less resilient against drought. And I know that's a funny thing to say given the last spring that we had, but actually we've had 2 or 3 really quite dry springs. So that's just something to bear in mind. I don't think I've got time to say all this, have I? No, I'm getting shakes of the head. I'm going to just flick through. This is an idea of how we can categorize the traits more effectively and what could be added to the RL, I think. Very simply, very simple things to measure in addition to what's already on there, just to give farmers a bit more information about the varieties. And potentially a system where we can add, so we've done that organic farm trial network and proved its worth and had a real impact. Potentially, can we start looking at doing the same thing for regenerative farming systems and for the conventional farming systems? And where these trials kind of overlap with that mother trial, the RL probably is about the relevance. Less relevance for organic, a bit more relevance for regen systems, and a lot more relevance for conventional systems. Flick through. I also just want to make the point, I'm really sorry because I'm now 2 minutes over, that in the RL's own scoping reviews, scoping exercises, that these trial networks, these on-farm trial networks, address all the issues. So there's interest in more variety data, check. Help understand how to deliver information on variety performance under lower inputs, check. Complexities associated with testing varieties for reduced input performance, that's what it does. Demand for data on the performance of varieties closer to on-farm practice, that's exactly what it does. So in terms of development, we're really making progress on the genetics. Environment, kind of less so. So I think short-term and long-term, the genetics we've really addressed. It's the environmental stuff and the management stuff that we really need to develop now. Thank you very much. I'm sorry I've gone 3 minutes over. Could have talked to you all day about all this because we've got 7 years of data and loads of things to discuss. If any of you are interested in taking part in that kind of network, you can see the sense in it and the relevance, please get in touch. Thank you.
Brilliant. Thank you very much, Dominic. Like you said, it's been a really long-term project. There's a huge amount of information, a huge amount of data, and I appreciate you trying to condense that into what is a relatively short space of time. But all of the presentations that you've seen today will be available to you after today. So don't worry if you missed some of the information that was on there. We can make that available to you after the conference today. So that's the end of our presentations for this Sustainable Farming Varieties session. So I'm going to hand over to Anna Reynolds now, who is going to field some of the questions that you've been sending in to us.
Thank you very much, Henny, and thank you to all of you. Fantastic presentation, really complimentary. I think we're going to have quite a few interesting questions and a good discussion. A first question that we've got on the screen now, I'm just going to go to that. Slightly reductionist approach, but if breeders were going to focus on one thing to ensure that crop varieties are appropriate for the future, what would that be for each one of you?
Do you want me to shoot? Okay. Yeah, it is an interesting question, and you might be surprised for me to say that I think I wouldn't say the issue necessarily is with breeders. Like, I think from the work that we've done, it's identified that there are varieties out there with the right traits. You just need to find them. Disease is a distant third, I would say, within organic farming as an issue. There are 2 reasons for that, but one of them is because breeding has produced disease-resistant varieties. I think it's done its job. It delivers what it needs to deliver within the system that it needs to deliver it. So I think the varieties are there. I think breeding's doing its job. I just think we need to test them in the right environments, and I think we need to get them out in the real world and see what we can learn from their performance commercially.
Okay, thanks, Dom.
Jed? Yeah, just basically mirroring what Dom said, it's just finding the traits and testing them, basically. I've found that root growth might be having an impact on competitive ability, but we need to do further testing on that to actually see if it is a real thing. So.
Yeah, absolutely. And I think the 2 of your presentation, you've highlighted quite a few different traits. And we need to bring all of that together now and see, okay, what are the relationships like? Where are the effects really seen? So I think that's really exciting.
Yeah, so maybe I'm actually a plant pathologist, so I would tend to say that there's a lot more needed to be done when it comes to disease resistance, and particularly with the shifting climate altering the kind of seasonality of some of the diseases that we have become more used to at particular times of the year. Some diseases are becoming more prevalent than they were previously, and so ensuring that we have all of those resistances that are required in our varieties, both to protect them today, but also protect them in the next decade or so as the kind of disease landscape shifts as well, I think is really important.
I think probably the thing we're looking for now is more sustainability in terms of resilience to varying weather patterns. We've seen over the last 3 years quite a lot of extremes, extreme heat, extreme wetness in the autumn. Whether you can breed for that, I don't know. It may be that, as Dominic said, it's not the varieties, it's the systems we grow them in, so blends. And we know blends give greater stability in terms of yield. We've seen countries like Denmark, much more wider acceptance of blends. So it may be not the varieties themselves, actually the way we grow them, or the way we have to adapt to the future.
Can I just say something?
Thank you. Yeah, sure.
So I think, the breeders would test all these lines within quite extreme environments. I'm sure they get sent somewhere with really cold winters to test winter hardiness. I'm sure they get sent somewhere with really dry summers. So whilst you might not get the absolute best for any particular situation, I think you get a really good, solid level of performance across a range of environments. And then I think it's about blends in order to bring in multiple, because you won't get all the right traits in one variety, it's impossible. But can you select, carefully the right complementary traits from 2 or 3 that you put together?
Yeah, absolutely. Absolutely. Good. Thank you. Another question here, staying with Paul. Would there be value in working with the trial operators and managers to make sure the chemical programme applied is available and published every year? So that's the recommended list chemical programme. And a question also says, much of the commentary about the RL programme and being too much is hearsay and not based on actual evidence of what has been applied. Paul?
Yes, so the programme that we give the trial operators is changed every year. We've got a meeting tomorrow to look at next year's programme. It is published on our website along with all the protocols for the recommended list, so you can go on our website and see those fungicide protocols. In terms of what each individual trial will put on, there is some flexibility within the programmes to adapt to local disease pressure. We have 350 trials, so putting that sort of information out there would be a challenge. We are looking at potential ways of putting more information out about individual trials. So whether we'll get to that sort of detail, we'll have to wait and see, but it's something we're looking at.
Okay. Thank you. And a question for Diane. What are some of the key findings from the other work packages?
There are so many findings because it's such a huge programme, but maybe a couple of really interesting findings. I think one is actually the accessibility of the data. So this has been a huge push from the community to make sure all the data is accessible. And I think that's been a great thing to see. So they have this grassroots structure where all the trial data that's done is integrated into that system, and then people can access that data. And the system is also creating new tools. So we have mobile phone apps where we can then put in data as we do our trials or greenhouse studies. So I think there's also all of this other infrastructure that's being generated through the success of the programme and the science that's going on. I mean, other areas is, of course, looking at things like new ways of dealing with disease resistance. So working more on, instead of integrating resistance genes, but deleting or modifying or selecting mutants in genes which are targeted by pathogens to promote their success. So removing things that the pathogen requires from the plant to survive. So also looking at these other approaches and trying to integrate those into the programme. But there are so many successes within this project, lots and lots of highlights to share, definitely.
And where can we find all that information?
So there is a website there, and there is a website you can go to. If you Google it, you'll be able to find the website as well. And that should provide you some information.
Thank you. Thank you. A question for Dom. What were the reasons behind the performance of X-Days in 24? And do you think its agronomics held back the blend?
Yeah. So this was actually my presentation and I had to gloss over it. So thanks for whoever asked this because it's a really good question. Yeah. Okay, so my theory for X-Days is it's the earliest variety that we test. And it's had it's been one of the top performers for 3 or 4 years. So last season, I think it was purely a feature of early varieties being disadvantaged. So I said early varieties are at an advantage when you get a dry spring, but when you get the kind of winter and spring that we had, I think when X-Days really wanted to grow on, when it went to stem extension, when resource demand got higher, it was really struggling. You've got to remember that within organic farming, the crops are relying on what I would call biological fertility. You're not adding anything in. And if the soils were in the state they were in March and early April, then it really suffered. Did it impact the performance of the blend? No, because the blend of X-Days and Mayflower was the highest yielding of anything we tested. So it just goes to show, like it massively outperformed the average of those two varieties. So it just goes to show this way we're designing blends has this real resource use efficiency. That I think we need to just investigate further. But I think we've proven it over 3 years using this temporal complementarity to separate out resource demand. Looks like a really good way, not only to provide climate resilience, but resource use efficiency.
Thank you. Paul, do you have anything to add to that?
I don't think so. X-Days didn't do quite as well as it has in the previous years in 2024 in RL trials, but it's still one of the better performing varieties. So.
Okay, thank you. And you mentioned Wheat Blast. What is the likely chance of it making it to the UK? That's the first question there.
I mean, Wheat Blast tends to like moist, warm conditions and conditions that we don't always see here in the UK, and so the conditions and the environment are unlikely to support proliferation of Wheat Blast in the UK at this point. Of course, never bet against the pathogen. It could change, maybe our conditions could change and support it, but it's unlikely in the near future that it would establish itself here in the UK. And I think then we'd be in a very good position because currently everybody's looking for sources of resistance, so we would have them in the future if this ever did happen. I think at this current point, I don't see it as a problem for the UK, but you just never know with these pathogens.
Okay. And what about, I think the second part of that question was about brown rust. Has the project looked at brown rust?
So within DSW, definitely brown rust is in there, Septoria is in there. There's lots of different diseases that we do consider, and of course they are very, very important to the UK, and our focus is mostly on the UK. Some of the researchers obviously within the programme, like myself, do a lot of work overseas as well, working for the international community, but most of the output and the work done in DSW is done for the benefit of the UK.
Okay, thank you. And the lack of brown rust seedling resistance in the RL, is that likely to change? And if not, why not?
I think you'd have to ask the breeders that question. Yes, the brown rust, there isn't a lot of variation in resistance, and I think there's only one variety that has seedling resistance. But yes, I think it hasn't been a focus perhaps of breeding programmes over recent years.
Can I just validate that? Because it's probably the disease that's increased in abundance in the organic trials. Brown rust become like more and more of the issue, so maybe that kind of validates that if it's been less of a focus for the breeding.
Interesting. Thank you. And I think that's a question for Jed. Did you consider the interaction between cultivar and seed rate? And maybe perhaps any other traits that Dom touched on as well?
It's obviously increased seed rate would probably increase your control, but it's not, we didn't have the time and resources to test everything. So it's not something that we looked at.
Something for the future.
We've lost time for the Q&A because I overran. Sorry.
Actually, one final question for Jed, just staying with that, is root testing, obviously very laborious. How do you think we can integrate testing of root growth into plant breeding, so to make it maybe time and cost effective? Do you have any tips? Because it feels like it's linked to many things, including nutrient deficiency, which we'll be touching on next. Any experience tips?
I'm not sure, to be honest. I mean, we've got ways of assessing root growth, but again, it takes time for plants to grow, and then there's no sort of guarantee that plant is also going to be increased in competitiveness. But
I've got time for just one really salient point that I think across the whole thing is that we do need to consider much more genetics and management, right? Because I think there are two ways to think of a variety's genetic potential. The perspective I come from is plant the seed, don't put any inputs on it and see what it does. That to me is its genetic potential, but there's also a sense that it's throw as many inputs as you can at it, and that's its potential, and neither's wrong, but they're different ways to think about it. When you add inputs into the system, you change the response of the variety and its physiology. So absolutely, I think more work needs to be done in reducing nitrogen rates, because if you pull back on nitrogen, you're not feeding the weeds, you're not feeding the disease, and you're not feeding the pests. Things change, which again comes back to the varieties need to be tested under the management that they're going to be grown under.
Yeah, go back to importance of participatory research and trials. Absolutely, absolutely. Good, fantastic. Thank you very much, everyone, and thank you to everybody listening to us at home from office or kitchen table. We're now going to wrap up, and I think we shall be back in about 5 minutes for the last session of the day. Thank you.
Hello everyone and welcome back to our fourth and final session of the day at the AHDB Agronomy Conference. So we're sticking on the same theme today of sustainable farming, but this time we're going to move away from looking specifically at varieties and we're going to look more generally at sort of sustainable farming, but sort of environmental factors. And kicking off the final session of the day, you have another familiar face. So we're heading back to Anna Reynolds, and Anna is going to be talking us through the new AHDB letterbox, the new AHDB research priorities. So over to you, Anna.
Thank you, Henny. It's my pleasure to be here today to talk about how AHDB puts you at the heart of future research and innovation in agriculture. Our industry is grappling with rapid change in trade, policy, climate, just to name a few, and whether it's responding to pest and disease pressures, achieving sustainable yields, or meeting environmental goals, the challenges we face demand innovative, practical, and farmer-led solutions. So how do we do that? And as Henny just mentioned, what we've done recently is introduce a new process into AHDB, which is to have a single point where all research ideas, questions, queries, any discussions go in, and then from there can drive the research agenda. So really centralizing that function and bringing all those information flows together. At AHDB, we believe that research is most impactful when it addresses real-world challenges that you face. And this process isn't just about gathering ideas, it's about creating an ongoing two-way conversation, and that's really been a focus. So as Henny mentioned, it's a letterbox. We have over 1,000 inputs already that are being currently analysed, and they're being combined also with other assessments like industry horizon scanning, risk assessments, emerging policy trends, and also with perspectives from other initiatives, such as the recent UK Farmer and Grower Research Priorities project, led by the Centre for Effective Innovation in Agriculture, and also more focused reviews, such as the reviews of RB209, which you're going to hear about later, and others. By integrating these findings with the ideas submitted through the letterbox, we can create a more complete picture of what matters most. To connect the dots between smaller scale and more targeted consultations and the broader input from levy payers and others, including agronomists, researchers, and so on, ensuring that every voice is represented in shaping the research agenda, and also we can be as responsive as possible. It also ensures that AHDB investment aligns with immediate needs and long-term opportunities, and results in a research agenda that's not only responsive but also strategic and addressing today's problems as well as anticipating tomorrow's challenges. So as you can see on the slide there, you can submit research ideas. It could be through the form that's available through the QR code. You can speak to one of our knowledge exchange managers. You can phone us. Any interaction will be logged. And those are then analysed, as I already said, emerging in various topic groups. As you can see on the screen, some of the top ones are nutrition and crop protection, IPM, perhaps unsurprisingly, but these are very, very broad categories, but that's what we're starting to see from the data. Underneath that, we have subtopics. As you can see, some of the top ones currently include cover crops, biostimulants, and managing lower inputs. So those things are coming through currently. I mean, if you agree with that, that's fantastic. Add to the letterbox, add more questions to that. If you disagree, same action. Please put your voice in. And from all of that, identifying the right research priorities is just the beginning. To truly deliver impact, researchers must be practical, it must be relevant, and it must be accessible. And this is where we are putting those themes or topics into themes and then into projects. And we already started to commission quite a few, which is very exciting. And it's going to really result in some great findings in future. This is also where on-farm trials come in and participatory research, as we mentioned in the previous session, really highlighting the importance of those, as well as cross-industry collaboration. So one of the examples, for example, is the cover crops work. Through all the inputs that we've had through the letterbox, we've commissioned several projects One of those is currently up for request for quotation. It's being co-funded by several water companies, really leveraging the levy investment. We also have a cross-industry steering group for that particular topic, and a couple of members of that steering group will be presenting a little bit later. But we really want to replicate the success of that topic in other areas as well. And we need your help for that. It's also where AHDB strategic cereal farms come in. That's where research meets reality. As you can see, for example, NUE, we've got tailored nutrition, strategic farm trials flowing out of those topics. And participatory research and demonstration, as already mentioned, is another cornerstone of our approach. By involving farmers directly in trials, we ensure that what we deliver reflects the diversity of conditions across farms. Soil types, weather patterns, management practices, to name just a few. So it's really a shared responsibility. To keep this momentum going, we need your continued input. Research and innovation works best when they're collaborative. So I encourage you to share your ideas through the letterbox. They don't have to be complete research proposals. This really can be, as little input or as much input that you feel comfortable sharing. Please engage with broader initiatives such as the sector councils, committees, various projects that you see being advertised. They really do make a difference, and your participation makes a difference. Be part of participatory research and trials, always important. And if you want to see updates on where we are with this process, what's coming out, what research projects are being commissioned, what the outputs are, and what farmers are sharing on the ground, then please subscribe to Agronomy Focus. It's a research and agronomy newsletter that comes out monthly. So it gives really great updates across the board on all current issues. Together, we can maximise the impact of research and ensure that every pound of levy money is an investment in a stronger, more sustainable future for farming. Thank you.
Brilliant. Thank you very much, Anna, for giving us that overview of the new letterbox process that we have at AHDB. And as Anna said, we really do encourage you to get involved. So submit your research questions. It really is incredibly valuable for us to hear what you'd like us to research. So without wanting to bombard you with QR codes, just a reminder to submit your questions via Poll EV. It's really important that you kind of share what you'd like to ask our panel at the end of our session. So up next, we're hearing from another member of AHDB. So we're hearing from Amanda Bennett, and Amanda is going to be talking us through the results of the RB209 review and where we're going to go next with nutrient management.
Brilliant. Thanks very much, Henny. Good afternoon, everybody. So my name's Amanda Bennett. I'm a Senior Environment Manager at AHDB, and I've got about 10 minutes now just to give you an update on the progress of the review of RB209 that we have been carrying out this year. But don't worry, we're not starting on a blank page. This time last year, we were celebrating 50 years of RB209 being recognised as industry guidance around nutrient management. But we also asked the question, what's next for the Nutrient Management Guide? We want to make sure it stays relevant and is useful for all of our levy payers. So AHDB committed to carrying out a strategic review of RB209 this year. Now, we had different aspects of this. We looked at the purpose and the role of RB209 as industry guidance. We wanted to look at how it's currently used and also look at some of the technical aspects around the data that we have currently, and also identify gaps and where we might need new research to update the technical guidance. Don't worry too much about what's on the screen there. This is really just to flag up that we have a dedicated web page about the RB209 review. So I would invite you to go and have a look at what we've got on that web page. It gives you a bit of background and also introduces you to the RB209 Review Committee. So this group was put together. We've got AHDB representatives from farming systems, agronomy and engagement, and the environment teams. And we have farmer representation from across our sector councils, cereals and oilseeds, beef and lamb, and dairy as well. So the focus areas for the review this year focused around three different areas, really. The first of these was looking at the governance of the UK Crop Nutrient Management Partnership. And I'm not going to go into a huge amount of detail about this today, but again, I invite you to look at our web page there that's highlighted on the screen. And this will give you the background around the steering group and the technical working groups that make up that partnership. And you can see which organisations are represented there as well. So it's across the industry, across the 4 nations. So we've done some work looking at the structure of that partnership and the membership, and also working with cropping sectors where AHDB doesn't collect a levy to ensure that we have a sort of a route for updates to those sections of RB209 as well. For part 2 and 3, we were very keen to get industry feedback, and we had a series of activities across the year to gather that from levy payers and from a range of stakeholders. So just for the next few slides, you'll see at the top there's a sort of a progress little flow chart just to take you through where we are. So this time last year and the beginning of this year, we started to look at what the initial views were. We asked the Crop Nutrient Management Partnership members for their input. We had a questionnaire that went out in January and February time with a very broad, open question around what does RB209 do well and what would improve it. And in the background, you can see sort of washed out there just an example of the number of comments that we had back from that questionnaire. So all of that free text that we were able to get everybody's feedback from. And if you attended the Agronomy Conference last year, you'll remember we had a poll and we collected some views as well from people who were putting their ideas forward for what would improve RB209. The next step really was to take these initial views and to try and identify some bigger themes. I haven't put them all up on the screen here, but I'm sure these will be very familiar to you in terms of the sorts of big topic areas where we need to delve into a bit more detail. And that's exactly what we did. We looked to explore those areas further. So over the summer, we had a main questionnaire that went out, and this was promoted on social media and at a number of events. And I'll just pause here to say it wasn't just me going out to do all of this, and there was a team of people, so thank you to everybody who was involved in that. And obviously highlighted in our own publications and newsletters. And if you're FACTS qualified, you'll have seen it in the FACTS newsletter as well. We did have a prize draw for a hamper, and just to show that we did have a winner, and our lucky recipient received their hamper earlier on this year. As well as the main questionnaire, we had a number of stakeholder consultations. These were conducted by Grounded Research, and they took place also from April to September. A little bit more in-depth interviews, a bit more targeted. From that, we've taken all of the feedback from those consultations and the main questionnaire, as well as additional feedback that was either emailed directly into AHDB or from conversations with staff across the year as well. Again, on the right, don't worry too much about the detail. This is just to show that we had a lot of free text comments that came in as well, with lots of suggestions and recommendations for what people would like to see develop for RB209. And we had some more directed responses as well from the questionnaire. So the bar chart there, for example, with some improvements to the presentation or use of RB209, people were able to rank what their preferences were. So we'll be able to prioritise some of that, taking it forward. The pie chart is just an illustration that we also looked at segmenting some of the answers so that we were able to see if it was an agronomist or a farmer who was had that input. And we also looked at whether it was a main arable production or grassland production, just to get a different feel for what the priorities were for the different respondents to the questionnaire. So we've got to the point where we have a very long list of topics, and our next steps really are to prioritise and plan what the next activities are going to be. So I guess the spoiler alert is we're not going to get to the end of this pathway today, but we will have RB209 updates coming in the future. But just to give you a feel for some of the headline topics, we had clear feedback to strengthen the position of RB209 as industry guidance. So it is not a regulatory compliance document. That was a very, very strong message. To focus on communication and knowledge exchange to improve the uptake and the use of nutrient management planning, using RB209 as the core foundation technical guidance for that. We had a lot of feedback about improving the user experience of RB209. Now, this can take many different avenues. We can look at the format of the hard copy publication itself, the digital version, the PDFs online. We can look at the API, the digital version of RB209 that third parties can access for their software, and talk about the development of an app. So there's lots of different areas that we can explore. Soil came through as a very strong theme as well, to reinforce soil health as a starting point for crop nutrient management. RB209 takes it as read that the soil is in good condition before you start looking at nutrient management planning. And just a little segue that if you're interested in our soils information, please do go to the Great Soils website, and we have information on there around soil management, as well as measuring and monitoring soil health indicators. Obviously, the key part is improving the technical information and the guidance. We have a very long list of topics, a wish list, if you like, that we could be focusing on for future developments. Some of the areas that we are particularly have come through particularly strongly are around nutrients across the rotation, release from soils. Anna mentioned the project about nutrient release from cover crops, bringing that sort of information in. And very strongly it came through about grass and forage crops. Section 3 of RB209 needing another update in terms of the content and the technical guidance there. And also because farming systems are changing and grass and management is changing, and there's quite a lot of arable farmers who are incorporating grass lays into the rotation as well. I won't go into too much more detail now. I don't have the long list to go through, but just moving on to my final slide, we are partway through this prioritisation process and looking at what we're going to plan and go forward with. The key bit is that we want to make sure that what we do has the best value for our levy payers. So we are going to be looking at technical updates, we are going to be looking at format and the presentation of the guide, and also working very much in partnership with the members of the Crop Nutrient Management Partnership, and looking more widely at engaging across the industry to help inform good quality nutrient management planning. So again, I invite you to look back at the RB209 review webpage for any further updates, and hopefully next year I'll be back again to tell you what we've been up to in 2025. Thank you very much.
Brilliant. Thank you, Amanda. Yeah, we look forward to seeing you again at the Agronomy Conference in 2025. And yeah, thank you for showing us what we're doing with those RB209 review findings, and that we're really taking on board what levy payers are asking us for and trying to then put that into practice. So up next, we have Danielle Roche from Sarah's research. So Danny has just very recently finished her PhD looking at biostimulants. So Danny's going to be talking us through some of the findings from her PhD and yes, what we need to look at in terms of biostimulants. So over to you, Danny.
Thanks very much, and it's a pleasure to be here today. So I'll give you a little bit of background of me as I have more of a presence in this sphere. I'll talk a little bit about what are biostimulants and how are they supposedly working, and I'll talk through what evidence we have for those effects, and then touch a little bit on regulation and future prospects of biostimulants. So my background, I did an undergraduate in environmental science and a master's in climate change at the University of East Anglia, where I focus a little bit on fish migration and rising sea surface temperatures and consumer and social science studies. I then moved on to Cranfield, where I've just completed my PhD, as Henny says, and this PhD really focused on biostimulants and soil health. It was partnered with Sainsbury's, and they were really interested in how fresh produce can be grown on better managed soils, and I'll talk a little bit more about my PhD in my later slides. I work for Series Research and Knowledge Exchange, and we're really trying to drive that gap between at the academic insights and actually what's happening on the ground, driven by the data. And I'm also heavily involved in the British Society of Soil Science and Women in Food and Farming Network in communications and their early career memberships. So what are biostimulants? Generally, we've got kind of two broad categories. First, microbial biostimulants, looking at those plant growth-promoting fungi and bacteria, and then the non-microbial biostimulants, so humic acids, those humic and fulvic acids. Within the category of humic substances, we've got protein hydrolysates, which encompass your amino acids, seaweed extracts, biopolymers such as chitin, and inorganic compounds such as selenium and aluminium. And this is not an exhaustive list. During my 4 years of my PhD, the last 4 years, different products have also come onto the market under these different categories. So biostimulants, what are they supposed to do? Well, inevitably they're there to improve crop productivity, whether that's the yield of the crop, quality in terms of those vitamins, mineral content, for example, the uptake of nutrients of the plant and their use efficiency of those nutrients, and the tolerance to abiotic stress like drought, salt pressures or temperature, for example. But what I'm really interested in and what my PhD focused in was around, well, how do these biostimulants supposedly improve crop productivity, and how do they differ from conventional products that we have? So fundamentally, biostimulants are not supposed to be a fertilizer. They are not supposed to be available forms of nutrients that you add like you do a fertilizer onto your soil. They are also supposed to be from natural resources and applied in minute quantities. So my research kind of highlighted that some products can be applied supposedly at 250 grams per hectare, which is really, really small. And they're fundamentally different in terms of that promoting innate productivity of your plant or your soil. And so these mechanisms can be broadly grouped into that sort of crop and soil area. If we look at the above ground mechanisms, these relate to stress-induced responses. These biostimulants can act as plant hormone-like activities, for example, regulating genes for nutrient transporters and metabolism of the plant, allowing that plant to take up more nutrients. On the below ground mechanisms, this is much less widely studied, and this is really where my PhD focused on. But there was suggestions that these biostimulants may be able to perhaps neutralize soil pH, improve physical characteristics such as soil aggregate stability, soil water retention, and also stimulate soil biology. But I'll come back to that in a future slide. So if we have a brief look over the above ground mechanisms first, I've picked out 3 example studies from the literature. The first was looking at, so that top one there was looking at biostimulants based on plant flavonoids, enhancing potato growth and commercial yields. And this study compared potato production in containers and commercially grown in the field. They used 3 different varieties, and they looked at both soil applied biostimulant and foliar applied. And on average, the tuber yield increased 33% in the containers and 5.2% in the field above those controls. But in terms of dry matter, there was no effect. And obviously, dry matter is quite important in your potato production and where that potato is going once it's out of the ground. If we look at the second one on the right side of the slide, this was a grain yield and quality study looking at wheat in response to a seaweed or humic acid based biostimulant. And this study looked at 3 different applications, foliar applied, and the key quality here was grain yield, and they found an increase 3.8 versus 4 tonnes per hectare of both the seaweed and the humic compared to the controls. But in terms of the other qualities that they looked at, being protein, bulk density, gluten, there were no differences. If we then look at the final study at the bottom there on my slide, this was a meta-analysis looking at yield effectiveness in field trials covering 180 open field experiments and looking at grain yield predominantly. And across all of those studies, the average yield benefit was close to 18%, but this was hugely variable depending on whether what kind of cultivation and what climate. So the greater performances in vegetable cultivations and in arid climates, more efficient in low organic matter soils, saline, nutrient deficient or sandy soils. And whilst this is a very brief overlook of these studies, what I want you to take away from this is that biostimulants have a very varied effect through these above ground plant mechanisms, and there's so many different variables here of when you apply them and what climates you're applying them to, what kind of soils, that it's quite difficult to determine an overall effect of these biostimulants. If we then focus on biostimulants and those below ground mechanisms on soil health, this is where my PhD really focused in on. The start of my PhD really looked at what evidence is there of soil applied non-microbial biostimulants. So I focused on the seaweeds, the fulvic acids and the protein hydrolysates. And we were looking at whether they how can they influence these different soil properties? And you can scan the QR code there for my review paper on this. And what we found was there was only actually 10 studies that were within the scope of this review looking at those soil-applied non-microbial biostimulants on soil properties. And there was a range of different effects. However, despite varying effects and to degrees of what soil properties these biostimulants impacted on, there was a general sort of reference to how they might work, which was to do with this water retention, aggregate stability, soil pH, inevitably impacting soil biology. And these studies, they all differed in terms of their spatial scale, so whether they were small pot trials or field scale experiments, whether they applied one biostimulant or a combination of, and what properties they were looking at. In terms of the soil biology, this was about the evidence and the theory suggested that it was the biostimulants acting as an energy source for microbes to utilise in mineralising organic matter in the soil and making nutrients available for crop uptake. So if I move on to sort of the aim of my PhD work, it was to understand the effectiveness of biostimulants in improving soil health and corresponding crop productivity. And this was split into 3 phases. The first was to quantify the effect of soil-applied biostimulant. So in the literature, there's very little to suggest what kind of applications are required, how frequently. And so the first was to understand how much do we need to apply in order to promote soil biology in terms of their community size and their biomass and their respiration and activity. The next phase was to look at looking at different combinations of different types of these non-microbial biostimulants on different soil properties, and that's where I covered a bit of soil biology, the soil physical properties in terms of aggregate stability, water holding capacity, and in terms of the chemical side, it was about that nutrient availability. And that led on to the third phase when we were looking at these biostimulant mechanisms in soil and relating it to crop growth, in particular spinach. Because spinach have a high requirement for nitrogen, so if we could increase the sort of innate productivity of the soil microbes, making that nitrogen available for crop uptake without additional fertilizers, that would be quite useful. So just to kind of reiterate that sort of hypothesis or theory behind these experiments, it's about trying to stimulate the microbiology and corresponding soil properties to increase the nutrient bioavailability for spinach uptake and corresponding quality parameters. So if we look at an overview of my results then, firstly, in terms of the quantification study, and what I should, as you'll see from the picture there, this was a small-scale pot experiment in a polytunnel because I wanted to control things like temperature and moisture as much as possible to really look at the biostimulant mechanisms without additional noise in the system as much as possible. So when quantifying this effect, we compared the recommended field application scaled down to the surface area of my pots from one of the biostimulant suppliers we worked with, and we compared it with 100 times that rate to see what was needed to really boost the microbial community. And we found that yes, there was an effect at the 100 times recommended application rate in terms of boosting that microbial community in terms of size and also their respirational activity. So using this application rate, we then moved on to compare the different combinations of biostimulants. So I looked at for example, seaweed applied on its own versus a seaweed and a protein hydrolysate. And the overall amount, the quantity stayed the same, but it was the different ratios of these types. And the theory here was that if we can provide multiple resources of energy for the microbes, we can improve their activity throughout the community and end up with greater nutrient or nitrogen availability. However, that's not what we found, and it didn't matter whether they were singly applied or applied in combination. There was no difference, neither from the control. So at this point, it was experimented on without a crop because I wanted to look at the native soil microbial response to biostimulants without additional variables. But actually, biostimulants were never intended to be applied on bare soils. It was always in a cropped system. And so at this point, I thought, maybe without that crop and without that microbial community in the rhizosphere, we're missing the amount of microbial community in the first place to be stimulated by the biostimulant. So my third phase was evaluating these different combinations alongside crop growth and whether there were differences between that native soil microbiology on the bare soil versus the rhizospheric microbial communities around the crop roots. But we didn't find any effects again between those single and combined treatments, nor compared to the control. So at this point, as a researcher, I'm thinking, well, why not? How am I going to explain this in my PhD thesis? So there are multiple reasons here. Firstly, the dose rate, the application, the frequency of how much I applied. We had that sort of proof of concept quantification study of how much to apply in the beginning, but in terms of frequency, I only applied it once at the beginning of my experiment because there wasn't enough evidence to suggest when and how many times I needed to apply this biostimulant on soil or in terms of spinach growth. So I didn't have any evidence for when to apply it more than once. And if we could apply it once and see over the growth stage where we need to apply it more often, that's quite useful. And if there was evidence already out there for the dose rate, the application, the frequency, and the understanding of the mechanisms behind why that's needed, I wouldn't have needed to do my study. We would have already known. Another aspect is I only measured these effects in the spinach system in a polytunnel in smaller pots. And of course, if I'm looking at the soil rhizospheric microbial communities, these differ between crop species. And so there may not have been an effect in spinach, but there might have been an effect in a different crop type. Soil type and texture is really important. My soil, my sandy loam soil, generally reflected one sort of set of biological, physical and chemical properties, and perhaps there wasn't a big enough community to start with or enough organic matter for the microorganisms to mineralise, or it could have been to do with the other physical or chemical properties. And so I think there's a lot more work to be done on how these biostimulants may differ in their action upon different soil types and textures. Moving on to the next one, climate and baseline nutrient conditions. So I grew my spinach in suboptimal nitrogen conditions because I wanted to look at firstly the effect of these biostimulants in those conditions, which is probably more common, and if there's a possibility of those biostimulants to negate the use of nitrogen fertilizer. But actually this might have inhibited the spinach growth to begin with, and if we're thinking about those spinach roots and the microbial community around those roots, if that's inhibited to start with, then my biostimulants may not have had as much effect because of this. Lastly, the mode of application. I applied my biostimulants to the soil surface through a small sprayer as reflective of, say, nitrogen in the field. But if we're thinking about trying to boost microbial community and that access of biostimulants to the microbial community, perhaps it's needed to be more incorporated into the soil. So there are many reasons why my results are not as clear of a biostimulant effect as I had hoped or hypothesized. And really what this shows is there's so many different types of crop and soil types, different biostimulant combinations, the amount, the frequency, and each one of those variables could be a PhD in itself, really. So there's a lot more work to be done to understand how these are working. So I just want to kind of give a bit of a reflection on the regulation and the overall lack of definition and mechanistic understanding of biostimulants really contributes to the disparate testing and labelling of these products. If we're not quite sure how they work, then we don't quite know when and where to apply them for the best case scenario and best agronomic improvement. And so how do we then test for them? How do we label them? And what do we need to measure as a response to these biostimulants? So just a brief overview of where we are with regulation. And it is quite mixed. So in the US, they've got a 2020 plant biostimulant guidance, but this is a fragmented regulation, and it requires compliance with a fertilizer law. And if you remember from a few slides ago, biostimulants are not supposed to act as a fertilizer. They're supposed to be fundamentally different. So there's a little bit of confusing boundary there with the fertilizer laws. In terms of the EU, they've got a fertilizing product regulation effective from 2022. And this does require certification, assessments for safety, efficacy and claims. And those efficacy and claims, I think, are really, really important. And the UK are creating their sort of amended version of this regulation. In terms of India, China and Australia and the wider global sphere, it's really a mix. Some have draft regulations, some are unregulated, some are as a biofertilizer. So it really creates problems when we don't understand fully how they work and where best to apply them to then be able to regulate them. So onto my last slide, science-backed tools or overhyped products, what do we think? Well, I'm going to remain a bit in the middle here and say it really depends. It depends on the product, it depends on the formulation and how it's intended to be used. It depends on the plant properties, what variety are you looking at, what kind of crop and what growth stage. And in particular, if you're applying to the soil, well, what kind of soils are you applying them to and what is the mechanism behind what you're trying to do with that biostimulant? And in terms of what's next, well, hopefully I've highlighted that there is a real need for an improved understanding of these biostimulant mechanisms, which comes from more research and more trials and creating an improved evidence base for these applications. If the manufacturers that I worked with had this evidence base of why we're applying this amount, when we're applying it, and the effect, I wouldn't have needed to do this piece of work. So really, biostimulants, they're not a blanket fix for all situations. They are so dependent on the situation, and I think it's where we are at the moment is needing more research to understand those niche pockets where biostimulants may be able to have an effect. So thank you very much, and I look forward to further conversations.
Brilliant. Thank you so much, Danny. What a fantastic PhD project. And we get so many requests from levy payers about more information about biostimulants. How do they work? You know, how do I know what's cost effective? So I think your presentation there really highlighted, you know, that there are still so many questions that need to be answered around biostimulants, and it is, yeah, such a varying topic. So yes, thank you for such a fantastic overview of your PhD work there. So we're about to head into our final presentation of the day, but for one last time, I'm going to ask you to submit your questions via Poll EV using the QR code. This is, one of your final opportunities to submit some questions before we hear from our final panel of the day. So our last session, looking at sustainable farming, we're going to be deep diving into cover crops. This is another topic that we get so many questions from levy payers about. It really is a hot topic at the moment. So we're really pleased to have two of the best names in cover cropping with us today. So we've got our final double act of the day. So we've got Toby Simpson, who is a farmer and Nuffield scholar, who's going to be talking through his experiences in his Nuffield scholarships, sort of what he found out about cover cropping through that and through his own experiences as a farmer. And then we're going to be hearing from David Purdy, who has also recently finished his PhD, who is a soil health specialist. So he's going to be talking a bit more about the sort of soil health mechanisms that cover crops can give us. So on to our final session of the day, and over to you, Toby.
Thanks very much, Henny. Thanks so much for having me at this really incredible conference. My name's Toby Simpson, and I'm just going to really quickly take you through some of the practical challenges and opportunities we've found with cover cropping on our own farm and in our own farming system. This is where we farm. It's a bit of context that I like to really sort of start with. We farm in the dry east, Cambridgeshire. In an average year, we might get 500 mil of rain. In a dry year, it could be half that. In a wet year, still nowhere near as much as some of the parts of the country. And even this year, we've had a lot, but not as much as some. So our challenge, though we get the rain over the winter, our challenge is actually holding on to it when the crops need it most in the spring. And we farm a range of soil types, mostly clays, clay loams, but a little bit of fen peat too, which provides its own challenges. And we grow a range of combinable crops, mostly cereals, some beans. We've recently stopped growing oilseed rape due to the challenges that we faced. And over the last 25 years, we've moved through a sort of min-till establishment system up to about 5 years ago, where we moved more into a no-till establishment system. And it was at that point that we were experimenting with not just the establishment method, but the whole systems approach to soil health. And it led to us trying cover cropping for the first time, really, and experimenting how we could make that work in our system. About 5 years ago, really wet autumn of 2019, quite similar to this, we planted some catch crops and we planted the wheat straight into them on the green. Got on really, really well, actually. Where we thought the slug pressure would be really high in the heavy clay soils, we found that actually the presence of the catch crop led to the slugs eating it and leaving the wheat sort of get away scot-free. We tried the same thing, actually, in the following spring, very dry spring of 2020. We tried some different termination methods on the overwinter cover crop that we'd grown, oats and oil radish. And what we found was that where we drilled on the green, the soil was in the best condition. It was the most friable, sort of best structured. But actually, when it came to harvesting the spring barley, we found over a tonne a hectare yield deficit in that strip that we had terminated post-drilling, probably due in hindsight to the allelopathic effect and perhaps some nutrient lockup too. But it really got me questioning how we were going to make this fit within our system. We really wanted to understand that. And that led me to apply for an Nuffield Farming Scholarship, which, for anyone who's not aware, is 8 weeks independent travel, researching a topic of your choice in return for a report, video and presentation. And I got to go to some incredible places. I focused mainly on the sort of Northern Hemisphere, USA, Canada, Scandinavia, Europe. And I met some amazing people along the way, and I'm always grateful to the Nuffield Farming Scholarships Trust and my sponsors, McDonald's UK and Ireland. And what I came back with was the one of the sort of key learnings for me was thinking about how we implement this in reality. And I like to think about FDD, functionality, diversity, and density. So when planning a cover crop, setting out with clear objectives and a management plan, and that will inform how we best achieve those outcomes. So when it comes to the cover cropping objectives, it's really important, I find, to think about what we want that cover crop to achieve, and then how we're going to go and manage it. So are we thinking about building carbon in the soil? We might want a higher carbon-nitrogen ratioed cover crop, or are we thinking about green manuring? Are we wanting to return more nutrients faster to the next crop to be available? So we might look at lower carbon to nitrogen ratioed mixes, using maybe more legumes, but actually probably importantly thinking about the termination time, the maturity of the cover crop at termination. Are we trying to get weeds to grow within it? Are we wanting to plant a thin stand, or are we trying to get them to smother? Do we want a dense stand? Do we need to think about nematode reduction if we're growing root crops in the rotation, sugar beet, potatoes, and there's a nematode problem? We need to think about perhaps using certain types of radish to reduce those nematode numbers rather than multiply. Or actually, are we in a situation where we can increase the diversity of the cover crop, maximizing the number of species and varieties that we have within it? And then are we grazing it, or are we not grazing it? And if we are, we need to think about not just the variety, but the species that we're choosing. And is it a catch crop or a cover crop? So thinking about the maturity timing, do we want that catch crop to sort of grow quicker, mature quicker, whereas actually if we're wanting a cover crop to survive the winter or to sort of last a lot longer, we don't want it to go too soon. So choosing the species is really important. This is just an example of a cover crop chart from the USA. It's very specific to the context of their soil and climate, but it's quite a good way of illustrating that. And actually, definitely a shout out for the UK Cover Crops Guide, which is being looked after by the AHDB now. Really, it's a fantastic one-stop shop for all things cover cropping, really good sort of soil health guide. Helps out with species selection and things too. And I'm a big fan of the visuals, anything that sort of shows the kind of rooting and shoot ability of the cover crop. And so we've got to think about species and the varieties within them like we would with our cash crops, what are those individual varieties able to achieve in sort of helping us achieve our objectives? What's the provenance of the seed? Where's it come from? Has it been grown to the same sort of standards that we'd expect in the UK? Are we potentially bringing in a weed burden by bringing in some sort of cheaper seed that hasn't been tested? What's the germination and 1,000 grain weight? Really important when working out plant density. And actually that'll change year to year. So having that sort of level of detail in the cover crop mix is so important. And trying to home save as well where possible, keeps the cost down, but also those seeds are primed with endophytes, so they're going to be used to the soil that you're growing them in. So growing the sort of easier to harvest stuff, like the bigger seeds essentially, and then mixing it up yourself on farm. This is an example of a very sort of technical farmer who went into a lot of depth mixing up his own cover crops, but my system was a bit more simple back home, just using a tote bin, layering it up like a cake mix, and then mixing it between two and then adding it to the drill during harvest, but keeping the cost down in various ways. And when it comes to establishment, there's no hard and fast rules, whether we're broadcasting, drilling, raking. Really it comes down to good seed-soil contact and moisture. And there's challenges at that time of year with moisture availability, labour availability, herbicides still active within the soil, residues not necessarily sort of distributed properly behind the combine, seed separation at different depths. Are we struggling to get clovers to grow at the same depth as beans? So thinking about perhaps applicating them in different ways. Raking and rolling, we definitely find that having that operation after is a really useful way of managing the slug populations, but importantly, treating it like any other crop. The photo there really clearly illustrates that even in a wet year where the catch crop took really well, where the trees have taken moisture away, it's really not achieving its job at all. So there are many challenges, no right answers, just sort of different ways to approach it. And the ongoing management is equally important. I've mentioned slugs, but also volunteers in barley and oats, for example. They can really smother out the effectiveness of a cover crop, and that's sort of illustrated in these two pictures. And when it comes to termination, again, there's sort of no right or wrong way. It'll feed back to what we want to achieve with our cover crop. Do we want to graze it down? Do we want to try and roll it, kill it on a frost? Or actually, more often than not, perhaps using herbicides. Recent sort of study found that actually herbicide-terminated cover crops provided more nutrients to the next crop, probably because they had a broader kill across the board. And it's a balance. It's a fine balance between termination timing. If we go too late, we sort of face the potential of nutrient lockup, high carbon-nitrogen ratio breaking down, locking up nutrients, the allelopathic effect. Certainly on heavier soils, clays, we find that where the sun and the wind isn't able to dry that soil out as quickly in the spring, we can really suffer with the next crop. But if we go too soon, we can lose that physical soil cover, the loss of the living biological roots feeding the sort of soil biology, and the nutrient loss too, if there's nothing there to be sort of mopping it up. It's a fine balance, and it'll depend on what you're trying to grow afterwards. If we're growing beans or maize, for instance, then perhaps we can afford to leave the cover crop longer. If we're on lighter soils, maybe it's less of an issue, but it's all down to the individual context of your own situation. And I really like to take biomass cuttings just for my own sort of sanity to work out what sort of performance the cover crops have done. So I can take fresh weight, I can make sort of dry weight analysis, separate out the different samples. It's just a really useful tool to sort of analyse in a little bit more detail about how well the cover crop's done. Sometimes we send it off for a bit of lab analysis, but I have recently been using a Mercy method, which is French sort of orientated, so based on a fair amount of research over the years, have to sort of input a bit of data to assume that we've got similar soil and rainfall. But in doing so, just by some simple input of fresh weight biomass cut downs of individual species, it's able to feed back the sort of potential nutrient availability to the next crop, which is really, really useful for ongoing management and understanding the sort of the long-term benefits of cover cropping and where we can maybe start to make savings in the sort of nutrient use in the next crop, but actually just accounting for the value of that cover crop all the way through its life cycle. And there's further opportunities too, public financial opportunities. The SFI is now hopefully up and running, mostly available to all, and funding overwinter cover crops and summer sown cover crops and spring sown, in fact. And then there's private opportunities too. The Lens network is a really good, if you sort of happen to be within one of the regions, a really good way of accessing funding for all sorts of different outcomes, cover crops being one of them. And water companies too are really starting to recognise the importance of treating pollution at the source, so using cover crops to keep the nutrients where they should be, to avoid sort of soil loss and pollution within the water courses. So within different catchments, there's many opportunities. Supply chains are also starting to fund this sort of thing. Livestock integration opportunities are equally apparent if we're able to work with neighbours. And I finished with the sunflowers. A farmer I met on my travels was using them alongside a road and was putting up a QR code essentially. People would come, see the sunflowers, pick a few, pay him a few pounds. It was on a patch of failed oilseed rape. And actually, he rather embarrassingly said that he'd managed to save up a few £1,000s by doing it. Felt so bad he had donated it to charity. But just really good public engagement opportunities alongside all the sort of wider benefits. But I'll hand over to David now for a much more in-depth look at cover cropping. Thank you.
Thank you, Toby. I thought I'd look at some more key science around cover crops and some of the data and some of the results that we've found. And I've entitled this little section, 7 freebies that cover crops provide. We all accept that, of course, cover crops take some money to absolutely establish, but then once they've established, what do they really provide? Well, of course, they provide energy into our system. As you'll see in a minute, energy is really, really important. They can deliver carbon into our soils in the form of carbon stocks, long-time stored carbon, that is. They can create, very importantly, very good soil structure. And I think for most people, that's the thing that they can see more than anything. And we define soil structure by aggregation. And aggregation in the soils provide many of the important processes that many of you will be very familiar with. They also increase porosity, or another way of looking at that is reduce bog density. Quite hard things to do, but cover crops are very able to do that. And we'll look at some data in a minute. Just to prove that. They improve hydrology, and of course water management within our production system is very important, so we need to have soils that can manage heavy rainfall events but also provide moisture in more drier periods, and cover crops help the soil kind of deliver those two very important things. They very importantly feed soil biology. We're learning lots more about soil biology, but cover crops certainly deliver and feed the biology, and of course the biology in turn provides many important functions within our soils. And then finally, well, it's not finally, but finally in this presentation they provide very important nutrients and potentially certain species fix nitrogen. Of course, this is not an endless list. There are many other things that cover crops provide. For example, they help with weed control, and they certainly provide a level of increasing biodiversity, for example, and of course there's many other things as well. So let's just look at how they capture carbon. Of course, the one very important, how they capture energy. The one very important thing is energy in a sort of global sense. And we can look at it like this. Earth is a chemical battery and photosynthesis is the trickle charger. And of course, over the last few years, human life has exploited that stored energy, primarily with fossil fuels, but also in the loss of our soil organic matter. And it might not feel like it, but that stored biomass, that biomass that's created by photosynthesis is critical to human life. There is no plan B. So getting energy into our soils is really important. Of course, cover crops help through this unique pathway we call photosynthesis, and they produce shoots, biomass above grounds, in other words. They produce roots, biomass below grounds. Of course, much of it leaks out of the soil as something we call exudation, and they feed all these little critters in the soil, and in turn, we haven't got time to explore that, provide many important processes to our soil. So as that energy flows into the soil, it's not a one-way process. Energy will leave the soil, and of course, what we want to do is create production systems where ultimately we bring more into our soils than we lose, and certainly cover crops are one of the very important mechanisms, and research has shown one of the most beneficial ways of actually delivering in this area. We can also look at it as an electrical system. I like to look at an electrical system where our plants become our solar cells, if you like. They charge the battery, and of course, depending on what sort of soil type you've got, if you've got heavy clay soils, you have a bigger battery if you've got lighter sandy soils, you have a small battery, and the amount of stable carbon in that is like the compartments most people can identify with how a battery works. And the charge level is the level of biological activity within our soil, and that's where cover crops really come in, because as you'll see in a minute, they increase that kind of biological activity and essentially our charge, it will increase the charge into our battery. And of course, as humans, and our activity, particularly in fossil fuels and soil organic carbon and tillage and things like that, we will also discharge some of that battery. And it's important, as I said already, to produce a production system that stores more than it leaves. And here cover crops play a really important role, particularly when they're combined with zero-till as well. And we'll look more at that closely in a minute, because they increase the biological activity, they have the ability to increase stable carbon, they help buffer things like pH, and of course zero tillage reduces oxidation, which is essentially energy leaving the system. And very, very importantly, probably the most critical thing, they improve soil structure. And soil structure, as I've mentioned already, drives many of the important processes within our soil. So it's a simple bit of physics at the end of the day. Energy in, work is done, it's like us as humans, and then functions are performed. And I'll touch on a few of those very important functions. So one of them is the ability to deliver carbon into our soils. And we've looked at photosynthesis already. This is the process of photosynthesis. You'll be familiar with it when you went to school. Very importantly, one of the things that cover crops do is increase the porosity within our soil, the amount of space, if you like, within our soil. And you'll see how that happens in a minute. And that allows more air and more water, and they are critical elements to photosynthesis and the plant development. They also deliver more minerals into our soils, another critical element to photosynthesis. So cover crops are a critical role. When I mean cover crops, I don't just mean a cover crop. Any opportunity to get a living plant into our production systems really constitutes a cover crop. And this is just a picture from a farmer in North America. It's a little microscope device. I've got one myself where you can fit to your mobile phone. It's amazing what you can see in your soil, but this is a plant root with its hairs delivering this kind of liquid carbon pathway into our soils, which in turn delivers many of the important processes and functions that we've already heard about. So this is work that we've done at Project Lamport near Northampton, where we looked at many different things. We looked at carbon. We had 96 cores down to a metre deep, so quite a lot of work we measured. Broke those down into 4 sections, so we had bog density and we had organic matter, and we can do some maths on that and create carbon, what we call carbon stock. And what we found when we looked at tillage systems versus no tillage systems, and no cover crops involved in that, is over 5 years we lost 17 tonnes of CO2 equivalents. I won't go into too much detail of that. But the left-hand plot you see there is just the actual carbon stock. The right-hand is the CO2 equivalents. In our zero-till work, where we looked at cover crops versus no cover crops, which is our control, then we saw further down the soil profile, which was quite an important thing, that actually a lot of this carbon is delivered further down the soil profile with some of these cover crop species. Quite an important thing just to remember. We actually, over 5 years, we actually drew down 27 tonnes of CO2 equivalents, which is an astonishing achievement for what are relatively small biomass cover crops growing for relatively short periods of time. But the combination of zero-till and cover crops can deliver that really important carbon stock, not just increasing organic matter, but actually increasing the weight of carbon in our soil. So that was an 18% gain, and much of it comes from this kind of root exudation that I've mentioned already. That's kind of one of the primary pathways. And of course, we know as we increase organic matter within our soils, lots of other things happen, and I haven't got time to go into all of those. But one is water holding capacities. There's many studies around the world. I've just picked up on a couple here that as you increase organic matter, of course, the availability of water and the storage ability of water in our soils goes up quite significantly. So next one, number 3, is they create aggregation of structure, possibly one of the most important things, because we know if we create aggregation within our soils, many of the other important functions that we strive to achieve in our soils follows on from creating aggregation. So this is work again done at Project Lamport, where we looked at lots of different treatments, but the treatment on the far right is our control. You'll have to accept my word for this, but when we started that work, those were big, blocky, angular bits of soil, and we just left it alone. We never had a cover crop in it, but we just left them alone, and now they break down into quite nice structured soil. It's not highly aggregated and biological, but just leaving soil alone on its own does a good job. The next one is every single year, whether we need it or not, at 25 centimetres, we drop a fairly intensive tillaging system into it. No cover crops, And you can see these two plots are actually right next door to each other, not even a metre apart. And you can see the effect of intensive tillage has on our soil. It's big lumps of soil. When you throw them on the ground, they do not break up. The next one is we've added to the tillage, we've added cover crops. That's the middle one. And you can see cover crops are having some benefit, but still tillage, especially at depth there, is still having an overriding effect and creating these large, very ugly, denatured soil. And the one second in from the left, or fourth from the right, is our cover crop plot. It's never had tillage, and it's had 3 species in now for a few years. And hopefully you can see a colour change, and that does represent, I've already mentioned, an increase in carbon stock as well. But you can see that soil is much darker, and you have to take my word for it, but it's lovely aggregated soil. And this is challenging silty clay loam with some challenging soil chemistry to go along with it. And the last one on the left is the field margin. And we've still got some way to go. We're 40% below the organic matter of our field margin, but we're heading in the right direction using cover crops and zero till. Of course, if you perform an intensive tillage event, that will destabilise soils. And what I say to everybody is you must follow up really with a living root. We can call that a cover crop, to build aggregation and not leave the soil bare. So this is another piece of work where you see we tilled the soil at the end of August, both years, quite intensive tillage. But on the right-hand side, we planted a cover crop and overwintered it. On the left-hand drawing, we didn't do that. And you can see quite a substantial difference in the way that soil kind of responds to having a living root in it. And it makes a lot of common sense, because as we till, we oxidate more carbon out of our soils. And of course, the plant is there ready to capture that carbon, deliver it in a carbon cycle back into our soil, bringing all those functional benefits that we're aware of. And so what's the, how do we get from, if you like, this was our soil when we started the work, big, angular, blocky soil, taken at exactly the same time of the year. How do we get from our left-hand diagram to the right-hand diagram? And I firmly believe it is fundamentally the power of photosynthesis, 5 years of work. But lots of other people, anecdotally, and there's many different sources of information, but can see that benefit is when we put a plant into our soils and that carbon gets pumped downwards, what an amazing effect it has on aggregation and soil structure. Of course, soil structure is one of those things that whatever resolution you use, whether it's the naked eye and a spadeful, or whether you stick it under a microscope, or whether you stick it under an X-ray machine, it kind of looks the same under whatever microscope level, whatever degree of scale you look at. And it's a phenomenal medium for looking at that. So lots of space, whether it be little space, whether it be big space, lots of really important space within soil for air and water, and they're really important. So now I look at soil health, if that's the right phrase, not from just looking at the soil, I look at it through the lens of photosynthesis. Whenever I pass a field and I see a field bare soil, I get troubled by it almost, because I think photosynthesis is so important. So having a plant there feeds the biology, feeds our root systems, creates structure and aggregation, and ultimately creates a healthier plant, hopefully more yield. If you manage cover crops well, I'll come back to that in a minute, and that increases photosynthesis. So it's that kind of making life from light process that photosynthesis is so important in doing. What's another freebie? Well, they reduce bulk density. This is no mean feat. Bulk density is a very damaging element to our soils, and cover crops, as you'll see in a minute from the work we've done at Landport, reduces bulk density. As we reduce bulk density, we will increase in porosity, as I've already mentioned, and that increases the amount of water you can have in your soil, but also increases the air in our soil, really vital for plant health. And it also reduces the need for intensive tillage as well. Sometimes eliminates, but certainly reduces the need for intensive tillage. So this is a big study looking at how bulk density has gone up over time. And I've just zoomed in on the UK here. The left-hand one is bulk density. The right-hand one is packing density. That's just taking a weighting from silt and clay, but it's essentially a very similar story. And you can see in our intensive arable areas, we're increasing our bulk densities. So for all the stuff that we do, we're increasing bulk densities, and that's reducing porosity, reducing air and water in our soils, and causing us all sorts of problems. And one real benefit of cover crops is to help us readdress that, help us reduce our bulk density. So again, this is a bit complicated, but this is work from looking at 3 cover crop systems. We have different mixes and different types of cover crops, but they all produce the same result in this end. The light green is our cover crop plots. The dark color, black, is our control plot, and you can see our 3 cover crop systems there all reduce bulk density almost to a similar level as the red line in the top left-hand there, which is our tillage system alone with no cover crops. And that's no mean feat. The very important thing to notice there is that that bulk density is being dropped at depth. It's not actually being dropped at the surface, which can cause a few challenges. And now we just focus our tillage on the top 80 millimetres of soil. Anything more than 100 millimetres we consider now deep tillage and consequently avoid. So cover crops are reducing bulk density, and of course that improves porosity in our soil as well. And cover crops, if you've got damaged soil, I've just looked at compaction here. This is a study done at Nottingham University where it's looked at different cover crop species, 3 of them, I've just pulled 3 out. It's changed the bulk density halfway down the soil profile, so it's 1.2 at the top, 1.4 at the bottom. And you can see after 58 days, the top one's after 20 days, after 58 days, you know, our radishes and particularly our black oats there are pushing through quite high bulk densities. And of course, that's where they're delivering carbon and improving structure and helping us in those kind of more difficult areas of the soil profile. So certain species of cover crops can really deliver in quite compacted, high bulk density environments. Of course, they feed biology, and this is a study of worms at Project Lamport, and you can see the red line is our mean of our controls, and over the years, we've dramatically increased the amount of worms in our soils. And we've gone from having 2 per spadeful now to having anything like 20 or even 30 per spadeful. And so that's been quite an exciting journey. And anybody who can appreciate earthworms, they bring many different functional, they're ecosystem engineers, many different functional benefits. This is 2 studies looking just at the ability for them to cycle nutrition and that nutrition contribution to grain yield. And 6.5% globally and 8% in Europe. And of course, this is from an AHDB monitor farm in Norfolk where cover crops were growing versus no cover crops. And you can see deep down in quite tight, compacted subsoils, you can see all these earthworm channels from these aneic earthworms. And that's where the roots of the plants also follow those channels because that's where a lot of rich environment is as well. Stop working. Okay, so looking at another biological response, this is something called PLFA, which is the fatty acids in microbial membranes. And I just pulled out 2 of the biomarkers. We actually measured 68 of them. I pulled out 2 because they're very interesting, which is the fungal biomarkers. One is saprophytic fungi, one is mycorrhizal, and you can see all the green plots. The far right-hand one is our field margin. We always reference everything against field margin. Again, the work's done at Lamport. You can see the amount of fatty acid as a result of those 2 markers, nearly double of what in our control plots, which is our grey one. So it's very importantly changing, say, earthworms, but also changing much of our microbial life, particularly in our fungal. Of course, we want more fungal activity within our soils. Of course, it improves hydrology. Very important that we manage water in our soils, and cover crops can help our soils deliver. This is a moisture release curve, and what we want in a moisture release curve is large pores, but we want a whole spectrum as pores. And one thing that biology does when you combine plants with soil, it really creates a wide spectrum of pore sizes, so that absolutely soil drains very quickly after a drainage event, but actually holds on to moisture very importantly when it becomes more restricted and you're in dry periods. So we want that phenomenon. It's only created biology, and it comes as this result of having energy in the system, work done, and functions are performed, is better moisture. So this is the infiltration rates at Project Lamport again, over 4 years, and you can see the first 2 years we didn't really affect infiltration rates. The 3rd year we started to see our cover crops, the green bar plots there, increasing infiltration rates, and then almost in year 4, it went exponential. And you can really see a difference as you walk the field of the cover crop plots now after a big rainfall event, how they're handling water so much more effectively. Again, it's about energy in, work done, and functions performed. And finally, it's just about providing nutrition. And I've given a few examples here. The right-hand one is from a farm in East Anglia, and you can see, I won't read them all out, but you can see the list of nutrients that are potentially available. And these are dry weights, cut down dry weights, similar to Ollie's already mentioned. And then the Project Lamport, I've just looked at nitrogen here. You can see how much potential nitrogen is available to the following crop or to the same crop. And we'll touch on that in a minute. And this is wet weights from Mike Powley up in Yorkshire. So it's going to be higher because they're wet weights. But again, you can see potentially how much nutrition might be available to the next crop or even crops further down the line. And this is work we've done at Project Lamport, where we had 2 years of cover crops and spring cropping. Then we went back into first wheats. One of our big objectives is get back into first wheats as we possibly can. And then we split the plots, so we had no N and full N. And hopefully you can see, without me going into too much detail, where we had no N, following the cover crop plots, delivered nearly 1.5 tonnes more yield than the same plots, the same treatments, but with no N. So our cover crops in the following crop, it's happening, further down the line, if you like, are really delivering nutrition into those following crops. So these are these are 7 freebies. They capture energy, deliver carbon, stocks, create aggregation and structure, increase porosity, reducing bulk density, improve hydrological effects of our soils, particularly infiltration rates, feed our soil biology, very important because they provide much of the benefits and provide nutrients and fixed nitrogen. However, as we've heard already, cover crop success depends on management. So how do we manage the system? Very quickly, we keep feeding the system, more energy in, more work done, functions performed. We put more in than we take out. That particularly is relevant to carbon. We encourage diversity. That's where cover crops, catch crops, companion crops in space and time are really, really important. But management is key, and we've already heard that. And early planting and early termination, we can talk more in detail, and the questions are critical to the success of cover crops. And finally, keep the habitat healthy. Don't smash the architecture. We have a philosophy of just stretching it, maybe at a very shallow depth, and manage this pulp density or packing density very, very carefully. And that's it. My quick run through cover crops.
Brilliant. Thank you very much to both Toby and to David for just a fantastic deep dive into cover crops. I mean, you covered so much information there, and yeah, really looking forward to sharing those slides with you afterwards, because there was tons of information in there that you simply can't cover in one session. So thank you very much to you both. So that is the final presentation for our final session of the day. However, we are going to move over to our speakers from this session to field them some of your questions. So we're going to head back to Ollie Johnson, and Ollie is going to be sending some of your questions to the panel for the final Q&A of the day. So over to you, Ollie.
Thanks, Henny, and thank you for sending those questions in. Keep them coming. And thank you, panel. Some really interesting topics there. This first question's around biostimulant efficacy, Danielle. And we're being asked, can you make some broad conclusions on biostimulant efficacy? If you have well-structured soils, strong earthworm activity, crops not under stress, would you say that many biostimulants are less likely to provide significant crop yield or quality improvement?
Yeah, good question. And firstly, I'll say these are my own thoughts, of course. I think biostimulants, as well as other amendments, are, I described it the other week as this is a system like a watering can with a hole in it. And amendments and biostimulants and things that we are applying onto our soils or into the top of our watering can are trying to sort of promote that soil health at the surface level, but we've still got that hole in the watering can feeding out, whereas good management practices over a long period of time, building up our soil health, however we define that, is then patching up that hole in the watering can. So to directly answer your question, I think if you've got good soil management practices, I don't know whether the addition of a biostimulant would significantly enhance your yields. I think it is really about that critical background management of your soils.
So it's about getting the background right, and then almost the biostimulants contribute to that as almost a magnifying effect. But if the fundamentals aren't right, the biostimulant won't necessarily help.
I think so, and also there's another aspect of it is it may not have a greater direct or significant effect on your yields, but they may have other biostimulants, may have other effects in terms of if you could still get the same effect of, say, nitrogen mineralization from a biostimulant, a natural resource, compared to a fertilizer, which is probably more expensive as well, there might be greater economic benefit in terms of what you're applying rather than the yield benefit.
Okay, thank you, Danielle. We've got a question here, this will be for you, Amanda, around RB209. We've obviously done the review and we've got some interesting bits out of that. Are there any bits from that review that we think we can implement straight away, that with the research is already there and we need to just put it into the guide, the low-hanging fruit, so to speak?
Yeah, definitely. I think we've had such a long list of things, fed back to us about what we can do. There are definitely areas where it's apparent to us that we just need a bit of signposting. Perhaps, the content is there in the guide, but as anyone who's familiar with RB209 will know, it can be a challenge to navigate it occasionally. So there will be definitely bits of work that we can do to help with signposting to where that information is. And there are definitely some sort of quicker wins that we can implement over the next 12 months. But the longer term technical updates might take, a few years if we need to do some research. But obviously we'll keep progress updated on that.
Yeah, thank you, Amanda. And I guess this question is related to that, and it's probably maybe for both of you to touch on. And it is around, in terms of research for AHDB, how do we make sure that we get that research from all the stakeholders and we're getting a good representation from across the industry?
Yeah, really, really important. There are many ways in which we can do that. And I think the main one is by collaborating across the industry with other stakeholders that also have interest in collecting that information and making sure that it's all fed in and analysed as part of the process. Because otherwise, as this question implies, we could fall into a trap of bias where we're just asking the same people for the same kind of input. So yeah, cross-industry collaboration is key, really, really consciously seeking out people that we're not currently engaging with on that level. And there are lots of ways of doing that, and that's a really key focus for us as we broaden this initiative.
Thank you, Anna. And this is on a slightly different topic. This is in relation to cover cropping now. And we're talking about critical numbers of species within a mix. And certainly from my experience as an agronomist, I've heard conflicting things, keeping it nice and simple, perhaps 2 or 3 different species, a broadleaf and perhaps a cereal-based mix. Let's start with David first. Is there a critical number of species? Are we looking for a greater diversity or keeping it simple?
There's a lot to that question. I mean, the first one is to ask what will actually grow where you are, because you've got to focus on what will actually grow. The second thing that's really important is what time of year you're actually planting it, because if you're planting cover crops slightly late, say into the beginning of September, many of the species just won't grow. So you've got to decide what will grow, and largely it's going to be determined by when you can actually plant them. After that, then you're looking for, it depends what you're looking for. If you're looking to capture nutrition, or then something will create a lot of biomass like a brassica crop, or if you're looking to fix nitrogen, obviously nitrogen fixing elements to that. It's always good to have a nitrogen fixing element in your cover crop. If you're looking to get through compaction, that's where I've mentioned already, where black oats or where a tillage radish or something like that. So you've got to kind of decide what you're going to do. What we find is that actually you can aim to have a really wide level of diversity, but actually what happens is a few species become very dominant, and many of the species don't perform very well in that kind of environment. So, but there are really good species. Black oats are good species. Phacelia are really bomb-proof little species. We're learning a lot about buckwheat, especially as a companion crop. That's a great species. Having a clover in there, and veggies are really good. Those sort of things really all grow really, really well and do a great job. But the important thing is try and get it in a little earlier. Get something in the ground that you know absolutely grows, and then you'll get many of the benefits that we've spoken about.
Thank you, David. And this is on a similar point, Toby. I'm interested in, from your experience, has there been an occasion or a situation where you would do something a bit different, where things perhaps didn't work, maybe a mix wasn't quite right or a species didn't establish properly? What would it be, and what would you do different this time?
I think it comes back to getting the basics right. So actually just paying attention to detail during a really busy time of year is really important. When we sort of started out, we were quite guilty of just firing and forgetting. It's just a cover crop, it doesn't really matter, we'll chuck it on and won't pay it much attention. But actually giving it the due attention that it's worth within the rotation is so important. And there's plenty of time to plan before harvest, to get the mixes right, to have it all ready to go, to work out how you're going to establish it. Being flexible as well, as David says, what time of year you're getting into a later harvest, then actually using sort of more appropriate mixes. And actually just thinking about it has probably been the thing I would have started out with, giving it sort of the attention that it needs.
Thank you. That's really good insights there. And we've got a question that's just come through around the credibility of biostimulants. So I guess this is for you, Danielle. I understand that most trials are conducted at 85% confidence rather than 95% confidence, probably for reasons we've spoken to about the variability. Does that undermine the confidence compared to say conventional chemistry on the fungicide dose response we've talked about this morning, for example?
I don't think it removes the credibility completely, and I think we need to be open and honest when we reduce our confidence levels. But I think that perhaps biostimulant, there may be a place where they are improving significance or they have an effect in such a small niche place in our fields or our trials, that perhaps there is a place for reporting at a slightly lower confidence interval. But again, I think it's always about being open and honest about those confidence intervals so that we can understand where and when they aren't working.
Thanks, Danielle. That's useful. And I just wanted to come back a little bit onto, because you showed a really great slide earlier, David, about the different structures of the soils, the different cover cropping systems and tillage and no tillage. And I wonder, this is a question for you actually, Amanda, is around the soil health scorecard that we have, and how might a farmer use that to be able to do that sort of thing on their farm?
Okay, so for those who aren't aware of the Soil Health Scorecard, it's a very simple on-farm way to check a variety of physical, chemical and biological indicators of soil health. The intention of it is that you would choose a place in the field and go back to it on a rotational basis and take those in-field measurements of a visual evaluation of soil structure and an earthworm count, and then also take a sample to send off for laboratory analysis around nutrients and pH and soil organic matter. There are other optional microbial indicators as well that you could tick the box for the lab analysis. I would say for cover crops, growing them once, you were probably less likely to see a change in those things. I'm interested of the timeline, David, of some of the results that you showed, because I think it's something that those improvements in soil health you would see after multiple occasions of growing cover crops, for want of a better term. But yeah, so not maybe immediately afterwards, so before and after you might not see a difference, but I think over repeated use over time is something you would definitely start to pick up.
And that's a resource, they can access that through the website. Yeah.
On Great Soils.
Yeah, on Great Soils website. Great, thank you for that. We've got a question here. We're still sticking on the subject of soil health. This person says, we can all see the benefits of cover crops to soil health, but what impact did they have on the following crop performance on heavier soils, later drilling, reduced yields, higher slugs, etc.? I suppose this comes back to the point earlier about perhaps what things can go wrong. Either of you want to pick that one up?
I'll handle a little bit of it. So yeah, we've got all these kind of functional benefits that can happen to soil or soil health. What impact do they have on the following crop? And that's largely going to depend on management, which is why I touched at management at the end. So if you establish a cover crop early, before the end of August, you terminate it before the end of January, there's every chance that it could, and I'm not saying it will, but it could improve yield of the following crops. If you plant late into September and you terminate, say, at the end of February, there's a very high chance, not guaranteed, that it will have a negative effect on the following crop. So management is the key to its effect upon the following crop. If you want to be bomb-proof, plant it early, terminate it early. It might not increase yield, but it will bring all those very important functional benefits into the soil. But you might just get a yield benefit as well. That's very seasonal, rainfall, weather, everything plays an important role in that. So that's our experience.
Yeah, thank you for that, David. And I suppose, from my experience as an agronomist, where I've seen people get into cover cropping with the greatest of intentions, and sometimes it works and sometimes it doesn't. I've got some ideas for me that where things have been going wrong, and that is around perhaps destroying too late and locking the carbon up, and then therefore not seeing the benefit. And it comes back to your point about they see that yield hit and would never do it again. Is that something that you see as you go around farms? Do you see that people, what are the common things that people get wrong?
I think it's a little bit about nitrogen management. So if you terminate a cover crop, say late, then you get a very, very dry period for the following crop, and the nitrogen just sits on top of the soil, doesn't get into the soil. That's the kind of worst of both worlds. You're terminating late and you're not getting the nitrogen into the following crop. If you can terminate early and get the nitrogen in, then there's every potential to improve yields in our experience.
Okay, thanks, David. Obviously a hot topic today, biostimulants again, Danielle. They're often marketed with the idea of having an effect in the season they're applied. But could we see gradual benefits if they're used from year to year? I guess a compounding effect.
Yeah, there's a really great question. I think it highlights actually that I haven't found products with enough evidence behind their applications, when, how often, etc., that would suggest they are either seasonal or long-term. However, the work I've done with biosims applied to the soils, if we're applying them with the mindset of trying to improve physical soil properties and that biological activity, that could definitely be something that could have a more gradual benefit from year to year.
Thanks, Danielle. I think that's all we've got time for today on that. But it was really great to hear your presentations and have those questions. Thank you so much for that. And thank you for joining us today and for listening to what we've been talking about. I hope you found it really interesting. It's been great to have you with us. Let's say a few thank yous, not just to these speakers, but the speakers that have been with us all throughout the day. Henny, who's actually behind the camera at the moment and doing some of the tech work, the tech team, who you haven't been able to see, but we can see at the moment, that have been helping to put all this together. And for the venue as well, for keeping us all together. It's been really good. Few final things from me. We've heard some great speakers today, from fungicide dose response, cover crops, environment, IPM, environment, soil health, the list goes on. What I would say is that AHDB has a really important role to play in the industry in collating some of these stakeholders together. And what I would ask is that please do use the information that we've been putting together today, but please do reference us to and reference the speakers if you use their information. Really, really important to show the value that we've been doing. You'll shortly, after this event, be receiving an email. In that email, there will be a feedback form. That's really important for you to fill that in, because it gives us a real clear guidance of what went well today, what things you enjoyed, and things you would like to see next year too. So please do take the time to fill that in. And don't forget, Anna has been talking about the letterbox. If you've got really great ideas that you want us to focus on, please do let us know what they are. But once again, thank you so much for joining us, and hope you'll have a lovely Christmas, and we'll see you next year.